Good morning, everyone. It's really a pleasure to be here and see you all. I see a few people coming in from the waiting room. David Ackerley, Dean of Rausser College. Of course, I haven't seen essentially any of you in person this year, but I did put on a jacket just to be a little more deanly, which I also have not done very often this year. I just want to start off and welcome you all to the annual Honors Symposium. And maybe we should start, Sophia, on slide 2, and then I'll say a few more things. I do want to start with a land acknowledgement that we recognize that Berkeley sits on the territory of Huichin, the ancestral and unceded land of the Chochenyo Ohlone, the successors of the historic and sovereign Verona Band of Alameda County. The land was and continues to be of great importance to the Ohlone people. We recognize that every member of the Berkeley community has and continues to benefit from the use and occupation of this land, and that includes its use for research and student learning. Before we continue, I want to offer my great thanks to Sofia and Anna for organizing, as they've done for several years now, to all of the advisors who have guided you all as students, and we have an extraordinary advising team, which we're very proud of in the college. To all of the faculty mentors who have guided projects in this year, especially doing this in this most unusual year. And most of all, to all the students who are presenting for all the extra work it took to both finish your studies and do independent research and do it under COVID. And we do hope that this is the last of our remote online honors symposia and that things will be returning to normal. But for those of you who are graduating, a very special congratulations, and we'll see you online. And I hope you get a chance to walk across the stage of the Greek Theater, those of you who are living in the Berkeley area, even just for the pleasure of walking across that stage. So again, thank you all for being here and all the hard work. And I'll be in and out a little bit during the day, and to hear a couple of them, unfortunately not as many as I'd like. And I'll turn it over to Sophia.
Thank you, Dean David Ackerley, and I extend also my amazing thank you to Anna Hoehenrieder for all that she does for the students and keeping the Honors Symposium with the quality that we have. I will just here to give you some background on logistics, and then we'll just move to the first presenter starting sharply at 9. Anna and I will keep you on time, and hopefully we will make this a family-friendly experience. I hope some of your friends and family are watching, and if they're not, this will be recorded and posted on the YouTube channel for the college. Welcome, and a brief history. The honors program was started by our colleague, Professor Tasios Melis, and he thought about it in 93 and 94 academic year. And you know, as academics, it takes us a year or more to think. And but anyway, it took some time to get it started at Berkeley. And the first honor students to graduate with honors from our college was in 1995. As you know, and for those that are listening, students enroll in 2 semesters in an honors class. And this is a 4 units class. So they do a lot of work with their advisors and independently putting their research projects together that you will see today. And one of the requirements is that you present your work in a symposium. And the first symposium was in 96, and we are now, I should have remembered, but we're having a lot of, actually, because now we also have symposiums in the fall, but we do this every fall and every spring now. And just to give you an idea, this is the first cohort of 1 student in 1996 spring symposium, and this is all of you. There are 63 of you this year. Today and tomorrow, 45 honor students will present their thesis. 18 have already presented in the Environmental Science Symposium, and they are part, all of you, of the 63 spring 2021 honor students in CNR. And together with the fall ones that did in fall 2020, it's the honors class of the graduating class. And we are so proud of you. And the benefits of being in the honors program is that you will receive honors in your major at graduation. The student that, because the judges believe the presentation and the project was meritful will receive a medal in the honor of Professor Melis. And the judges that are judging today and tomorrow will be Ellen Bruno, Leila Safavi, Lynn Hunsinger, Vincent Fon, and Chloe McCollum. And please join me in thanking them for coming, listening to your presentations, and judging your work. But also, they will give you feedback on suggestions for improvements, and that feedback can be asked either from me or Anna. We are welcome to send you then that individual feedback on how your project pushed the frontier of research in their opinions. And you, of course, will have bragging rights. And today is my honor to present the cohort. And if you wonder what number you are, the first student presenting today, Claire, will be number 777. And the last student with the environmental science students will be, so the last student on Friday, sorry, will be 822. And then with the environmental science students, that puts us into 840 honors students since 1996. And this is all of you. And thank you for sending me your photos. And this collage, with your permission, will then be proudly displayed in our college website so that you can see, yay, that's me over there, spring 2021. The logistics are as follows. If we were in person, your advisors will be proudly sitting in the audience holding this. What is this? These are the cords, the honors cords. And they would just be holding them and waiting for you to be done, and then would come up and give you the cords around your neck, and you would then wear them for your commencement on top of the black robe. And this is presently being delivered by mail to your house, address of record maybe that you have. So if you're have it already, yay. If you don't have it, it's coming. So look for a surprise in the mail. And Anna also sent some details for other things that you are expected to do to complete the process. Thank you again for the faculty mentors. Thank you for those especially that are today joining us and supporting your students. You are spectacular. Thank you to Anna. And with this, I will ask, we are going to start a little early, if that's okay. And I will ask Claire, I think it's Claire, right? To kick us off. I was sharing my screen, so I lost the schedule. Yes, Claire Perrin, that is going to take the stage and present her work. Claire, you have 10 minutes. And maybe I should give Anna some opportunity to also say something before Claire starts, because she's probably going to talk about the timer. Do you have that somewhere, Anna, or no?
Yeah, so we don't have the timer in the back, but I'll show also, if you want to look at the hands, I'll show when you're down to about 5 minutes, and also when you're down to 1 minute. So
yeah.
So
Anna will, and then when you're done, we'll do this. And for those that came in late, I shared a link to participate in this event, and I should know better, but apparently I'm not very good with Zoom. So as you can see, there's many of me now. And if you see my name under your photo, there is a way to change that so that you have your own name and can show up with your own name in the photo. Anyway, Claire, take it over and welcome everybody.
Thank you. Okay. Good morning, everyone. My name is Claire. My project is on identifying drought resistance genes in CRISPR-Cas9 transformed tomato plants. And I am in the molecular environmental biology department. And I want to give a huge thanks to Professor Dodd for sponsoring this project, and Dr. Marty for all his mentorship and support. So I used a wild drought-tolerant tomato species endemic to the Atacama Desert as a model for CRISPR transformation. So the Atacama Desert is the driest non-polar desert in the world. The average rainfall is below 10 millimeters per year, and there are periods of up to 4 years with no rainfall. And Solanum sitiens is a wild tomato species endemic to the Atacama Desert, so it tolerates extreme drought and salinity conditions and provides a great model to identify genes involved in drought resistance. So the abscisic acid stress ripening, or ASR, gene family is involved in response to abiotic stress, such as drought and salinity. And Solanum sitiens, which is the drought-tolerant tomato found in the Atacama Desert, and Solanum lycopersicum, or the domesticated tomato, show stark differences in the ASR4 gene. So this figure shows basic local alignment search tool, or BLAST, results for Solanum lycopersicum and Solanum sitiens in the bottom row. As you can see, Solanum sitiens has 2 deletions, which are shown by the dashes. It has 1 deletion of 56 base pairs, followed by another deletion of 36 base pairs in the ASR4 gene. So CRISPR-Cas9 technology was used to knock out these 2 regions in domesticated Solanum lycopersicum tomatoes. And CRISPR uses small guide RNAs, or gRNAs, to direct Cas9 endonuclease to specific DNA sites. So 2 different gRNAs were used to create 2 different constructs of transformed tomatoes. So construct 1 plants have the first 56 base pair knockout, and then construct 2 plants have the second 36 base pair knockout. So after Dodd Lab sequenced Solanum sitiens from the Atacama Desert and transformed plants using CRISPR-Cas9, I sequenced plants to determine that the 2 CRISPR constructs were successfully incorporated into the transformed plants. So this involved germinating and transplanting tomatoes as they grew larger. And then I collected samples from the greenhouse, homogenized samples using a bead beater, and then performed DNA extractions using a CTAB procedure. Next, I ran polymerase chain reactions, or PCRs, with primers targeting the areas in the ASR4 gene that were knocked out using CRISPR. And here's a photo of a gel I ran using the PCR products. And then samples were sent for sequencing and analyzed using SnapGene, which is shown here at the bottom, and BLAST, which I mentioned earlier. So next, after confirming both CRISPR constructs were successfully incorporated into transformed tomatoes, I tested the plant's phenotypic response to drought and heat stress. So the experimental design involved 4 treatments using 2 growth chambers in the UC Berkeley greenhouse. So one growth chamber had a temperature of 26 degrees Celsius, and the other had a temperature of 36 degrees Celsius. And within each growth chamber, half the plants received water and half did not. So there were 3 genotypes used in each treatment, 2 CRISPR constructs with different regions of the ASR4 gene knocked out, and then a control with no CRISPR transformation. And plants were left in the growth chamber for 8 days and then returned to the main greenhouse for recovery at ambient temperature and with water. So I expected both CRISPR constructs of the transformed tomatoes to withstand drought and heat stress better than the non-CRISPR control, since the transformed tomatoes were modeled after the wild, drought-tolerant Solanum sitien species, which was native to the Atacama Desert. And the results of the stress test with the 4 different treatments were very striking. So all plants from treatments 1 and 3, which were watered, survived the 8 days in the growth chamber, but construct 2 showed drought tolerance in treatments 2 and 4 without water. So just as a quick reminder, both constructs had different regions of the ASR4 gene knocked out. So these graphs show the number of plants alive versus time. So the number of construct 1 plants alive is shown by the yellow line, construct 2 is in red, and control plants are shown in this mint green color. So the y-axis shows the number of plants alive, and each genotype starts with 3 plants. And then over the 8 days in the growth chamber, plants begin to die without water. So looking at treatment 2, the top graph, by the end of the 8 days, all construct 1 and control plants died, but 66%, or 2 of the 3 plants from construct 2, survived 8 days without water. So for treatment 4, which was 36 degrees, we see similar results. Since the temperature is hotter than treatment 2, the plants did begin dying faster than treatment 2, but 33%, or 1 of the 3 plants from construct 2, survived 8 days without water. So construct 2 is showing remarkable drought resistance in both cases. And here are some images of the stress test. So I'm focusing on images from treatment 2, which was the top graph in the previous slide, to highlight the differences between the 3 genotypes, since treatment 4 was quite aggressive due to the 36-degree heat stress. So these images are from 3 days of recovery after the 8 days in the growth chamber. And construct 1 and control plants, as you can see, completely died, whereas construct 2 was very much alive, healthy, and even grew new tomatoes and flowers. So it's showing really incredible drought resistance. And this brings me to my key findings and conclusions. So I was able to successfully use the CRISPR-Cas9 protocol to knock out 2 regions of the ASR4 gene. This was shown by sequencing of the ASR4 gene of the transformed tomatoes. The seeds gathered from the transformed tomatoes and then germinated had the transformed gene. So that's showing that transformation was stable across generations. And the stress test results strongly support the role of the ASR4 gene in drought stress response. So the transformed tomatoes with CRISPR construct 2 showed drought tolerance, unlike the other 2 genotypes, which could imply that the 36 base pair deletion in the ASR4 gene may confer drought resistance in CRISPR-Cas9 transformed tomato plants. And this could provide a means of introducing increased drought resistance in tomatoes as well as other crops. And this has profound and exciting implications, especially when applied to agriculture and food production. Some benefits could include heightened drought tolerance, which is particularly important as climate change progresses. CRISPR-Cas9 transformed tomato crops could also increase the stability and resilience of food systems and decrease agricultural water use. And this would contribute to a more sustainable system of food production overall. And moving forward, I will verify the results by conducting another stress test. I will also conduct amplicon sequencing to verify results and determine how strong inheritance is across generations. And finally, I will search for other genes that may be involved in drought resistance. Thank you. That's the end of my slides. Please let me know if there's any questions.
Thank you, Claire. I thought that was an excellent presentation, very professional. Well done. You've done great work during the semester. So again, congratulations for that. Do you think that you're going to one day come back and look at other plant systems and perhaps drought resistance, climate stress response? What are your thoughts?
Definitely, yes. Thank you so much, Professor Dodd, for the opportunity to research with your lab. It's been incredible for the past 2+ years. So thank you so much. I definitely very interested in pursuing more research on drought resistant crops. And it's really exciting, you know, applying new technologies like CRISPR and, you know, especially with climate change progressing, it's super important and relevant. So it's super exciting to do research on this. So thank you.
Great. Thanks. Thanks to you.
Thank you.
Did you, Professor Dodd and Claire, do you also consider other variables that could be affecting this treatment, such as temperature, degree days? Is that something that, so you could do like a design that would be kind of not just 2 by 2, but it's just a thought, because you have such rich possibilities in the lab. We social sciences are so jealous of you guys because you can control everything.
Yeah.
And
well, the third, I guess, Dodd, you can also speak to this, but we did do like the 36-degree growth chamber was very aggressive. The 36 degrees Celsius was pretty aggressive. So a lot of the plants died. It was kind of remarkable. Even one of the construct 2 plants survived. So I think in the future we could consider doing another growth chamber with more variation in degrees, but maybe not, you know, 26, 30, 34 degrees Celsius, or something like that. And we did originally, we were only going to have the plants in the growth chambers for 5 days, but we didn't. We wanted more plants to keep dying, so we kind of kept them in there longer to make sure, you know, we weren't just we were adequately stressing them in terms of heat and drought stress. But yeah, we could definitely talk about that, Professor Dodd, and consider, you know, some tweaks on the experimental design in future experiments.
And Michael Black asks a related question: soil quality. I assume everything's been randomly assigned, so it's the same soil quality, but maybe you were proposing a variation along those lines, Michael?
We used Sunshine Soil Mix number 4 for all of the tomatoes. I don't know. I don't know if I didn't have, I just, that's the mix that we used. We didn't really consider mixing the soil since we were mostly trying to focus on drought and heat stress. But that could also be a consideration, interesting factor. But yeah, we controlled the soil, so that wasn't a factor in the experiment. Thanks for the question, Michael.
I would just like to add that actually our original project here was on pine, but because the germination and developing the protocols for CRISPR were taking so long with pine, then we were using tomato as a sort of pilot study just to test our system. So no, we haven't tested all the other possible factors that could be having some influence.
Right. Yeah, tomatoes grow a lot faster than pine trees. Makes it easier.
Well, thank you. I will ask Katrina Black to start transitioning to her slides. And I again thank Professor Dodd for being part of our attendees. I hope you can stick around and see how spectacular the other students are, but welcome everybody again that's showing up now. We are in the Honors Symposium and Katrina Black has the floor to present her work.
Hi, yes, thank you. What a great presentation, Claire. So yeah, hi everybody. I'm Katrina. And today I'm going to be discussing my research on self-incompatibility in the hexaploid Prunus domestica. So I wanted to give a quick thank you to my advisors, Professor Dodd, as well as Dr. Marti, for all of their guidance and encouragement they've given me throughout this entire project. I'm going to start with some background on what makes this species so unique and why it should be studied. So the Prunus domestica, also known as dried plum, is a popular snack that contains many nutritional benefits and serves as a source of fiber, iron, and many other vitamins. Additionally, the seed oil is often used in skin and hair products. So the fruit originated from the hybridization of the diploid species Prunus cerasifera, which is pictured in the middle of the bottom row, and tetraploid species Prunus spinosa, pictured on the bottom left corner. The hybridization results in the hexaploid species that I'm studying, which is pictured at the center and labeled a European plum. The specific trait that I'm studying is self-incompatibility, and self-incompatibility is an important evolutionary mechanism that prevents self-fertilization. So in nature, this mechanism promotes genetic diversity through outcrossing and cross-fertilization using pollinating insects. And these traits are evolutionarily beneficial because they can reduce the risk of certain diseases and genetic abnormalities. However, the opposite of self-incompatibility, also known as self-compatibility, is the most important thing to look at from a breeding perspective, a plant breeding perspective. And self-compatibility speeds up the process in order to produce the highest yield of fruit-bearing plants in a harvest. Furthermore, self-incompatible plants rely on pollinating insects, whereas self-compatible plants do not. And this is a really important thing to look at, as pollinating insects like bees are declining due to climate change. And currently, all of the species being farmed in California right now are self-incompatible, so they rely on the presence of pollinators, which is concerning for the future of this species. The diagram on the right gives an overview of the compatibility system. And I'm just going to describe it a little bit. So in order to create a successful pollination, the pollen alleles, which are indicated on the top and are numbered S1 through S3, need to be unique from the pistil alleles, which are indicated on the bottom. In the figure on the left, you can see that S1 through S3 match on both the top pollen genotype and the bottom pistil genotype, meaning that the plant is self-incompatible and the pollen tube stops growing. Whereas in the second figure, all of the pollen alleles indicated at the top differ from the pistil alleles indicated at the bottom, meaning that the plant is considered self-compatible and the pollen tube can reach the ovule, which will eventually produce a fruit. The self-compatible allele that I'm studying results in the non-functioning of the entire self-incompatibility system. So the self-compatibility allele that I just described has been identified and exclusively studied in over 100 papers for all other diploid prunus species, such as almond, apricot, cherries, plums, and many more. However, there have been less than 5 papers published on the Prunus domestica because it is difficult to study a hexaploid. A hexaploid contains 6 copies of each chromosome, which is pictured on the right, instead of like the typical 2 copies of a chromosome in the diploid species, which is on the left. You can see that there's a lot of room for variety then in a hexaploid, as the genome can have allelic variation at each of the 6 chromosomes, which makes it even more difficult to find self-compatible fruit because each of the alleles have to be unique. The goal for my project was to identify the self-compatibility allele that results in the non-functioning of the whole self-compatibility system, like I mentioned, among different Prunus domestica plant samples. Additionally, I was also identifying allelic variability with samples in order to provide more information for breeding programs. So here's an overview of how I plan to test my hypothesis. I began with sample collection, which included picking leaves off of the trees, and then I extracted DNA from those plants and then amplified that DNA using PCR, and then lastly, amplified the results through gel electrophoresis. Initially, I extracted DNA from over 130 plants last winter using a CTAB procedure. However, the DNA was pretty difficult to isolate due to contamination from phenols and polysaccharides because I initially extracted from buds, which are pictured in the center, and they're kind of harder and have a pretty thick coating. So it can be difficult to get really good quality DNA. So I ultimately went back to Winters, California, which is where the orchard was for all of the trees, and collected the leaves from the trees when they had grown on. And I ended up collecting those leaves and then performing DNA extractions the same day in order to produce the best quality of DNA in order to move on to the next step of the project. So the next step after producing the good quality DNA is to run PCRs with different primers. So we began with a universal primer that works with the entire Prunus family. So it gives us a good overview of all of the alleles in each sample. And so we use the Pru C2 PCER primer to identify the variability of alleles for the different samples, specifically looking at the SI gene. So each lane can have up to 6 bands, which represents each different allele. And the lanes are indicated, they go across and they're numbered at the top, and each number represents a different sample. And then the bands, which are the green bands in the center of the gel, each of those bands represents a different allele, and all of the numbers represent, like I said, the different alleles. So we found that S17 is associated with self-compatibility, and this allele is located in samples 10 and 14. And we confirmed this in the field at the orchard based off of the phenotypic data from the trees. So we began with this overall picture, and then the next step is to narrow the scope, and we were looking at that specific S17 allele. And so the next step was to design a primer to do that. And so that's what this primer is. It was identifying the S17 or SC allele in certain samples just to figure out this was like I said, associated with self-compatibility. So with the presence of a band in this gel, it meant that the sample had the self-compatible genotype, whereas the absence of a band indicated that it did not have, it had the self-incompatible genotype. So overall, interpreting the results from the gels, the first picture of the gels, we identified the 17 alleles, and this variability is super exciting and will be helpful for future plant breeding. Like I said at the beginning, currently there's only one variety of Prunus domestica being harvested in California, and this can be an issue as it can lead the species to be vulnerable to widespread disease, pest outbreaks, and annual and statewide variations in yield caused by variable weather conditions that negatively affect the fruit and can lead to crop failures. So having a wide variety of alleles is super exciting and helpful to address these threats, which is very important to ensure the future of this plant. Additionally, in the second gel I showed you, we found that at least one of the 17 alleles we identified is associated with self-compatible plants. And this is exciting because, like I said earlier, again, as pollinating insects become more scarce, it's important to have self-compatible plants, as this is kind of the future of agriculture. So following up, we're going to create these self-compatible plants using CRISPR-Cas9 technologies to transform the Prunus domestica in order to knock out the self-incompatibility alleles. Ultimately, our findings give us an improved understanding of the sexual reproduction mechanism among the Prunus species. Additionally, our findings will create faster next generation breeding, as breeders can now use molecular markers for early detection of self-compatibility rather than having to use phenotypic data, which would be having to grow the trees, and it just takes a long time. And then they'll have to see, like, based off of how the trees grow in order to decide the next step. We can do this process a whole lot faster with the molecular markers that we found. And so these findings will have a major impact in the agricultural field and potentially beyond. That's the end of my slides. Please let me know if you have any questions. And thanks to everyone for showing up and listening to my presentation.
Okay, thank you very much, Catriona. That again, like Claire, you've done great work over this last 18 months, I suppose it is, and a great presentation. Thank you.
Thank you, Professor. That means a lot coming from you.
I would just like to add that you are dealing with a complicated system. This is a hexaploid, and in this case, the putative parents are a diploid and a tetraploid, and so that adds a lot of complexity to the problems of allelic variation, and of course the role that it will play in self-compatibility or incompatibility. I'm pleased that you mentioned that it was an association, this S17 allele, because anyone who knows about the mechanisms of self-incompatibility will no doubt know that it is quite a complicated system, and so we don't understand exactly what's making, transforming, if you like, organisms to self-compatibility, and it could be lying at two different regions within the self-incompatibility gene locus.
Mm-hmm. Yeah. Yeah, it's a pretty complicated thing, but it was Professor Dodd did an amazing job helping me through this whole thing and really breaking it down step by step, because it was pretty complicated. But yeah, thank you, Professor Dodd.
Thank you, Katrina.
So how was this research affected by the pandemic research away? It looks like a lot of lab and manipulations needed to be done, and I praise you both for this, but I'm just kind of curious how it all worked out.
Yeah, that's a great question. We were lucky enough to have all of the lab members were very cooperative in all of the COVID restrictions, and getting into lab was kind of took coordinated effort in order to figure out timings and stuff. But luckily I was able to get into lab and do a lot of my DNA extractions and all of that type of work by myself, like, and I was able to be in the lab by myself, which was super, super helpful. And then, yeah, when I have to like collect, when I had to go collect the samples, it was all outdoors, which was helpful. And so luckily my research wasn't too affected by COVID. If anything, it gave me a little more flexibility with having classes being remote, and I had a little more flexibility with my schedule. So yeah, we were able to make the best of it with a lot of like help and coordination from all of the lab members.
That's spectacular. I have a question from Ciara. I hope I'm not assassinating your name. Do you think you could use your technique on a plant with a higher pairing of chromosomes, is asked.
Yeah, well, as you get higher with, as you have more chromosomes, it gets a lot more complicated. Polyploidy is a difficult thing. So definitely, I think that we could use some of what we've learned through this, especially as we've learned more about like reproduction mechanisms with a hexaploid. It definitely could be, it could provide information for working with a species with more chromosomes. But yeah, great question.
Well, again, congratulations. And I will ask Olivia, if Olivia Yeeun, please, could take the floor, the Zoom floor, and share your slides.
Yes. Hi, everyone. Thank you so much for being here today. I'm excited to talk to you about my research, which is on the spatiotemporal patterns in wildfire connectivity. And then I also just want to thank my mentors for this project, which are Van, Diego, and Ariane. They provided so much help along the way. So I wanted to focus on wildfires within California for my thesis, because I was just interested seeing their significant increases in size and frequency in recent years, which you can sort of see in this graph below. But then they're also really important. They can be really detrimental for human health, but also incredibly essential for ecological health. So that really interested me. And then ways that we sort of commonly talk about and measure fires include in terms of the area burnt, the number of fires, their relative destructiveness. So I was interested in something slightly different, which is their spatial connectivity to each other. And so to study this, I used percolation theory, which is this method that helps you understand the connectivity of a network. And so if you look at this diagram below, how that works is you have this grid with all of these different cells, and cells within it can be occupied, say, by fire randomly with a probability denoted as P. And so P can increase then from 0 to 1. And as it is increasing, you have this sort of transition point where before you had many small isolated clusters within the network, and then you eventually get this dominant interconnected cluster. And this transition point is called the percolation threshold, or PC. And you can see here that it is this constant value of 0.59. And so this theory has been applied across multiple fields like geology, forest fragmentation, urban planning, but then I wanted to apply it to wildfires. So my questions were then, how can I apply this, and what would this add to our understanding of fires? Has the spatial distribution of fires changed over time? And then what would be the percolation threshold value for real world fire data, and would that be different from the theoretical value? So this is an overview of my methods. I first obtained spatial data on all recorded fires within California for this time period from 1950 to 2019. I then rasterized this data, which basically means converting it into this grid pixel format. I then created different time aggregations from the data. So taking the 70-year time period and then dividing it into a series of 1-year chunks, 2-year chunks, etc. I then apply this clustering algorithm, which assigns fires 2 clusters together based upon their distance apart. And then I use these two methods to characterize what's known as the critical distance, which helps measure this transition as modeled in percolation. And so this is just sort of demonstrating what these fire rasterizations look like. So in the top left corner, you see the state of California, and then these purple shapes are all the recorded fires from 1950 to 2019. And then I also wanted to show what I mean by these different time aggregations. So you see within this period, then each decade of fires laid out separately. And you can sort of see how when you split into smaller and smaller chunks of time, you get sort of sparser fires that are more spread apart. So then the question was, how do you measure connectivity when fires are sort of spread apart across the landscape like this? And that's why we use the critical distance. And so if you look on the left-hand side here, this is the 70-year fire data altogether. And I've assigned fires to clusters based on this distance of 7 kilometers. So the clusters are in these different colors, and each cluster fire pixels are 7 kilometers or less from their nearest neighbor. And you can see here that there's sort of these two very large clusters, there are a couple of smaller ones, sort of disconnected across the system. But then as you increase this distance, so on the right-hand side, if you go up to 9 kilometers, then you sort of get this much larger sort of system spanning cluster of fires across the state. And the question is, at what exact distance does that transition occur? So I use these two methods to sort of estimate that critical distance value. On the left-hand side is what I call the derivative method, and this basically takes a weighted average of all of the jumps in size of the largest cluster. So for this data, that's 7.85 kilometers. And then the other method I use, I call the jump method. And this again looks at the size of the largest cluster, but just assigns the critical distance to the point where the cluster grows the most in size. So that was 8.5 kilometers here. So you can apply these two methods to all of the fire data and across all of these different time aggregations. And you can make these graphs of critical distance by time. And so what you might notice at first is that the two methods are very strongly correlated with one another. And so they're showing the same general trend in the data, which is a good sign, but the jump method does have sort of higher variability. And then also what I was sort of surprised to see was that there wasn't necessarily a clear increasing or decreasing trend over time in the fire data. So that prompted me to look at connections of critical distance to other variables. So what I ended up finding was that there's this really significant connection between critical distance and the total area of fires. So you can see these two plots here, again, showing these two methods, derivative and jump, and those values are expressed on the y-axis. And then on the x-axis, you have sort of this proportion of the state covered by fire. And so as you create larger and larger time aggregations shown in these different colors, you're adding more fire area, which makes sense. And then you're also proportionally decreasing the critical distance values. And so I use this relationship to then predict the point at which the critical distance would be equal to 1 kilometer, which is the resolution of the data. And that would sort of mimic this true percolation theory where all the grid cells are directly interconnected. And so doing that and some mathematical transformations, I found these values for PC, which are 0.86 and 0.89. And so in conclusion, these critical distance values can help us approximate the percolation threshold. And the values that I found are greater than this theoretical value, which you may remember is 0.59. And that's because this theory is based upon randomness. So 0.59 reflects if fires were sort of randomly spread across the space. That's what you might expect to find. But of course, fires are not spread apart randomly across the state, and they're concentrated in certain areas and certain ecosystems. And so this sort of quantifies that degree of non-randomness. And then what was also interesting to me was that even cumulatively after 70 years, fires were still far from this predicted point of percolation. And since fires can be such an important ecological force, their sort of fragmentation across the landscape could then have impacts on sort of habitat fragmentation and things like that. And so in the future, I think it'd be interesting to more closely analyze this data in relation to different ecological and socioeconomic factors. And then this could also be applied to other types of landscape phenomena as well. So yeah, that concludes my presentation, and I would love to take any questions.
I have a question. I was just trying to understand. Can you go back to the slide where you try to predict the PC level with the two?
Yeah.
So the colors depict different levels of aggregation, and it looks like the right graph is kind of less variable than the left graph is less variable than the right graph. And it's hard to see the colors, but is that because of the aggregation method, or is?
Are you talking about like these?
Yeah, they're
these
just like
over here.
What's going on over there? That there's some right down there that don't show up over there, and I was just wondering if it's driven by the method, or
yeah. So it is by. So these are the two different methods to characterize the critical distance, and this method on the right is just looking for where the largest jump in cluster size occurs, but that we sort of found was not always necessarily the most accurate, because you may by chance sort of just have really few fires and a large jump in size might occur before you might theoretically call that like the transition point. So it just tended to have a couple more outliers than the other method. But yeah, that's a good question.
So we have a question from David. Do you want to unmute and ask the question, or should I read it? David, feel free to unmute. All right, I'll just read it. Really interesting work, Olivia. As you looked at the maps, did you have a sense that the percolation threshold may be reached sooner in some parts of the state over others if you did the analysis regionally?
Yeah, that's a good question. Let me actually go back to the maps. So that was actually one of the ideas that I sort of wanted to investigate, but I didn't quite have time to do. But yeah, if you look at where the fires show up in the state, first of all, they don't show up in all regions. So like the Central Valley area, the deserts to the south, don't have very many fires. And so I think it would have been interesting to also be able to split into different sort of ecoregions and analyze those separately, just because it's such a large state with so many diverse types of land that it's sort of difficult to maybe like generalize about. But I would say that especially the South Coast has really very intensive and like heavy fire seasons that cover a lot of the area there. So that would probably look closest to me to percolation at the moment. But yeah, the regional analysis is definitely something I'm also interested in.
All right. Any other questions? Olivia, what's next for you?
That's a good question. Finishing this thesis write-up and yeah, enjoying my summer in Berkeley.
You deserve it.
Thank you.
All right. congratulations once again on a spectacular honors project with huge application to where we live. And that also brought to the attention David's question on applicability in other settings with fire, similar issues. I know Portugal has a lot of similarities with that climate thing. And I was wondering if you thought about thinking about beyond California in any of your results. Is there external validity, or is it very specific to California?
Yeah, I think it could definitely be applied to other sorts of areas that do have these really prevalent fires. I think it would depend on sort of the availability of data. I don't know too much about like how other areas collect data, but I know that California like has these pretty complete and pretty far back reaching records of fire. But I think that it could definitely be applied to other areas, yeah.
You should definitely share your work with our former dean, Keith Gillis. You will probably find it very interesting. He's been very involved with California fire.
Oh, very cool.
All right, Lily, feel free to take the stage and you have the floor. Lily Levesque-Eichhorn is gonna talk about her work. And I think you have to share, yeah, perfect.
All right, can you see everything normal? Okay.
Beautiful ball.
Perfect. All right, thanks everyone for being here. So today I'm going to be talking about some work that I've been doing in the Niogi lab over the past 2 semesters. So the title of my talk is A Genetic Analysis of Endosymbiosis in a Novel Algal Ciliate System. So I'm just going to start off with talking a little bit about what the system is. So why is this system important, and how can it help us understand secondary endosymbiosis? So Stentor pyriformis is a large single-celled pond-dwelling ciliate, and interestingly, it hosts hundreds of mutualistic endosymbiotic algae. So on the right here, we see this one giant cell, which is our stentor. And then all of these purple dots are endosymbiotic algae of the genus Chlorella. So what we are interested in doing is developing this as really a model system for studying secondary endosymbiosis. And there's a few reasons that this system in particular is really suited for this type of study. So the first thing is that stentor really exhibits very interesting and complex behaviors. So for example, they are able to phototax. Additionally, they are able to regenerate. So these are really interesting behaviors that we would like to understand more. So why study endosymbiosis at all? Well, endosymbiosis is a mechanism for great evolutionary change. A classic example of that is primary endosymbiosis, which is thought to have led to the great oxygenation event of the world's atmosphere. Additionally, endosymbiosis is a mechanism for increasing biodiversity in otherwise unsuitable habitats. So you can think of like, for example, coral reefs. So just to get into the project components, there are a lot of components to this project because it's a really new system. We know virtually nothing about it besides what we can observe visually. So we have this new system and we have some observations, which I'm going to circle right back to. And then the other really piece of data that we're working with here is we were able to sequence the genome and transcriptomes. And these aren't completely complete and like perfect sequences yet, but we have like the preliminary data as of yet. So what this allowed us to do is to look for things like genome statistics, GC content, genome size, chromosome numbers, things like that. Additionally, another part of my project was looking for specific genes involved in specific functions. And this is really important because it allows us to understand what is the genetic and molecular basis of some of these observed behaviors. Additionally, we looked at the presence and absence of these same genes and other ciliate and algae, some of which were symbiotic, some of which were not, in order to understand how our system may be different from these other similar systems. So today, I'm really just going to focus on our photoreceptor story because there's too much here to present in 10 minutes. And this is really motivated by this observation, and I'm going to get into that right now. So Stentor pyriformis, our ciliate, exhibits phototaxis in response to blue-green light, and this obeys a circadian type pattern. So we can see in this video here on the right, each of these cells is a stentor, so they're really, really large. And you can see that they're moving directly across to the left where there's a light source. And this really begs the question, is the stentor or is the chlorella responsible for this behavior? So we hypothesized that chlorella, which is an symbiotic endosymbiont, is a photosynthetic endosymbiont, may be sensing light via blue-green photoreceptors, and then signaling to its host to respond accordingly. So kind of picture-wise, this is what this would look like. So we have a light source, we have the stentor in green here, it's endosymbiont in purple. The endosymbiont senses the light and tells the stentor move towards the light. The stentor does this, and because they have a mutualistic relationship, this benefits both the host and the endosymbiont. So how did we test all of this? Well, to test all of this and try to investigate this hypothesis a little bit more, we needed to look for the presence or absence of photoreceptor proteins and genes in both of our organisms. So we looked at 4 main groups. And what we did was we first compiled a list of these photoreceptors from really well-studied organisms. So we had a bunch of sequences that we knew had photoreceptor function. And we blasted these against our 2 genomes, both from the stentor and from the chlorella. And then what we did was we confirmed these hits by reblasting them at a bunch of different stringencies with different matrices to really confirm, okay, these are for sure good blasts. So what we're really interested in is which organism has more green or blue light receptors, because these are the receptors that could potentially be involved in this phototactic behavior. So what we see here, first, we're going to look at aureochromes. So aureochromes are really classic blue light receptor. And when we look at our results, we see that stentor has weak homology with 1 aureochrome reference gene. This is the data here. So we have a pretty good percent identity, not a great E-value. And then when we run this gene, the actual sequence from the stentor genome through a domain finder, we find these domains which have function related to aureochromes, but not directly aureochromes themselves. Additionally, we have, when we look at chlorella, we have a multiple gene homology. So more genes are homologous with both of our aureochromes. Again, when we run this through a domain finder, we find this PSA domain, which is not directly an aureochrome, but it is related to signaling pathways involved in photoreception. So the takeaway here is that while stentor pyriformis may have 1 aureochrome related gene, chlorella likely has multiple genes involved in aureochrome function. We looked at channel rhodopsins. First, we found that stentor pyriformis has homology with 1 channel rhodopsin gene. However, when we run this through domain finder, it kind of looks like this might be an error because we get this WWE domain, which is not at all involved in channel rhodopsin function. It's involved in like ubiquitin stuff. Interestingly, chlorella does not have homology with any of the channel rhodopsin reference genes. So moving on to cryptochromes, we see that stentor has no cryptochromes, but chlorella has multiple gene homology with all 3 cryptochrome reference genes that we ran. When we run this through a domain finder, we actually do find a cry domain coming up. So we can say with pretty high confidence that these chlorella do have cryptochromes. Again, the takeaway here is that stentor does not have any cryptochromes, but may have 1 channel rhodopsin potentially. And then chlorella appears to have multiple cryptochromes, but no channel rhodopsins. Okay, so I've included phytochromes here, which are actually in the far red/red spectrum, which is not likely to be involved in this phototactic behavior because, again, phototaxis was only observed in response to blue and green light, not in response to other wavelengths. So what's interesting here, though, is we see that both the stentor and the chlorella have pretty good homology and multiple gene homology with both of these, with the reference phytochrome genes. So for stentor, there's multiple gene homology with the 2 phytochrome genes. Here are our 2 top hits from that. And then for the chlorella, we have multiple gene homology with all 4 phytochrome genes. So when we run these through domain finders, we do indeed find the phytochrome domain for chlorella, which really, makes us feel, okay, this for sure has, this organism does have phytochromes. Again, this is not necessarily related to phototactic behavior, but it's definitely interesting to think about. So again, the takeaway is both stentor pyriformis and chlorella likely have multiple phytochrome-related genes, although chlorella does appear to have more genes involved in phytochrome function. Okay, so conclusions. We can see here from this Venn diagram that we have cryptochromes only in the chlorella domain, so only chlorella has cryptochromes. Only stentor has channel rhodopsins, though that has pretty low confidence in that result. And then both of our organisms have aureochromes for pretty low confidence, especially for stentor. So what we can say here is because chlorella confidently has cryptochromes and more likely has aureochromes, chlorella may be better equipped to initiate phototaxis in response to green/blue light because it contains more blue light receptors. And this really, is interesting because it shows that, this molecular analysis supports the idea that there's communication between the host and the endosymbiont in exhibiting these behaviors. So chlorella may be signaling to its host, move in that direction towards the light so that we can both benefit from optimizing photosynthesis. So with that, I just want to acknowledge the CNR Honors Program and thank you to everyone in the Niogi lab, especially Vincent Bordeaux, who was my mentor. I could not have done it without him, especially with having to transition everything with COVID. And then thank you, Professor Niogi, for being my faculty mentor and all of your support. With that, I'm done.
Well done, Lily. I will ask, I see that your mentor's in the house. So if you have a question, feel free to unmute and ask. Otherwise, just put them in the, oh yeah. You want to unmute? Krishna? Oh, sorry.
I didn't have a specific question for Lily. I just, I was just clapping and nice job.
Thank
and
you.
I think Vincent is also in the attendee list. So he's also here.
Great. Yeah, thank you for this opportunity. This was a very exciting thing to be able to study for a little bit.
So I'm looking for questions in the chat. Feel free to start them in. We're running a little ahead of schedule, but feel free to write them. And Vincent, if you have any comments to make, I see you have a fantastic background.
Thank you for having me, and well done, Lily. This is a real tour de force this semester or this past year in being able to transition from what was supposed to be a wet lab science project to a completely computational virtual science project. So well done.
Thank you. I couldn't have done it without all of your help.
So Lily, what's next?
Well, I'm graduating, which is very exciting. I'll be in Nevada for the summer doing some conservation work. And then I'll be looking for more jobs. So
yeah. No grad school in the horizon?
Probably I'll be applying either this year, so this fall or the following fall, which is a little bit more likely because I want a bit of a break. So
very good.
Yeah.
Well, you make us all very, very proud. Thank you for being part of the honors program, and we wish you all the best. And thank you for the mentors that not only attended, but also supported your work. You make it all possible. So thank you so much, Krishna and Vincent. All right, without further ado, I will ask if Elizabeth Ordeman wants to take it over and start presenting. And I have to say that Elizabeth is welcome back.
Thank you.
You were in the fall, so double kudos to you. Does that mean you're like double honors?
I'm not sure, but I'm glad
I think you are.
Thank you. Okay, so today I'll be presenting on my work on the milkweed butterfly lineage, and really looking at what evolutionary forces are driving toxin resistance in these butterflies. So big shout out to Professor Noah Whiteman from IB and Professor Ben Blackman from PMB for mentoring me along this process. So it's important to get this background in first of that there's multiple trophic levels involved in the story of the monarch butterflies and the milkweed lineage of butterflies. So the entire lineage of milkweed butterflies has evolved target site insensitivity to cardiac glycosides that are found in the milkweed plants that they eat. And so really what I wanted to ask was why this target site insensitivity has occurred in this entire lineage of butterflies. Was it because of the benefit of herbivory, of being able to eat the milkweed plants and access that food source? Or was it because some of these butterflies, such as the monarch, are able to sequester the toxins and thus gain protection from predators? So this is why you see the bird throwing up after eating the monarch, because it stored toxins that it has eaten as a caterpillar. So the sodium potassium pump plays a key role in this, as it is the place where mutations occur that provide the target site insensitivity, as cardenolides target the sodium potassium pump. So it's the sodium potassium pump generally maintains the concentration gradients of sodium ions and potassium ions across the cell. And so if that's blocked by the cardenolides that are in milkweeds, then it can no longer function, and the cell will no longer have the proper gradient of ions, which is very bad. So we found that the butterflies in this lineage have mutations in this pump in particular in order to have resistance to the cardenolides in their food source. So very specifically, the mutations are present at amino acid sites 119, 111, and 122, as found by previous work from others in the lab that I worked with. And Wabane was used in their experiments as a way to see where this binding pocket would be. And it was found that Wabane and other cardenolides bind to the potassium binding pocket. And so the mutations that are in that potassium binding pocket are thus what confer resistance to the cardenolides and milkweeds for these butterflies. What's really interesting in this lineage, and I found very cool, is that the butterflies themselves across the lineage vary in resistance. So those at the bottom, which would be the genotypes QAN, LAN, have are very low resistance, and LSN as well. And it goes up to VSN and VSH being the monarch butterflies genotype that have the highest resistance. And so then typically in our first experience, we just looked at Wabane. And so in this project, we wanted to look at how these butterflies would respond to different milkweeds. And so a different mix of cardenolides, as milkweeds themselves vary in toxicity. So doing this entire project with a lot of butterflies would be somewhat difficult due to the long life cycle of butterflies. And so we are able to use CRISPR knock-in Drosophila lines in order to isolate the sodium potassium pump and those mutations. And it was really important that we were able to do this in order to test the sodium potassium pump's resistance itself, as the milkweed butterflies exhibit other adaptations to their toxic milkweed diet, such as an iron gut, which helps them not absorb the toxins in their food, and also the sequestration aspect exhibited by some, such as the monarchs. So in order to test things at the organismal level, Nama Wexler, who I worked with, we collaborated on larval feeding assays, and she did her honors thesis in MCB on these assays in particular. And so we were able to gauge resistance at the organismal level because we used 6 lines of Drosophila knock-in flies and then tested them on milkweed media. And so the way we made this media is we grew up multiple different lines of milkweeds with varying levels of toxicity. And we mixed the milkweed into media in which we grew up second instar larvae of the Drosophila lines. And then we monitored survival, and we were able to quantify this survival against the different levels of toxicity in the milkweeds, and also use a control of SY media and also a lettuce control in order to test for any effects of having plant material in the media that does not have cardenolides. So my thesis really focuses on the physiological side of this, which is looking at the sodium potassium pump in isolation from the flies themselves. So we're aiming to gauge the resistance of the pump at the physiological level. And the way that we're able to measure this is that since the sodium potassium pump is an ATPase, that means that one of the products of its function is inorganic phosphate, which can be quantified using colorimetry with ferrous sulfate molybdate reagent. And so what we did is we took the 10 different lines of milkweed and created, as you can see in the figure to the right, we did a separation and extraction of the actual cardenolides that are in different mixes in these different milkweeds, and then placed those pumps in isolation in the solution and measured the ability for them to react and continue functioning in the presence of cardenolides. So at the organismal level, there is an interesting result because we expected there to be 3 different levels of resistance, but there was only 2. And we expected 3 because there's 3 different levels of toxicity in the milkweeds. And so what we found very interesting is that on the far right, where you see the names perennis and asperula, which refer to the milkweed species, the VSH lines, which are the monarch butterfly, way outperformed all of the other lines. And so that implies that there's benefits for outside of herbivory for the VSH lines, as they have side effects from the mutations that must be countered out by the fact that they're doing something else. So what's interesting is that VSH is a line that is the monarch, which sequesters the toxins. And so this provides evidence that the protection that monarchs get from predators of sequestration is a driving force for their additional levels of resistance outside of the other lines. So the next steps is for the physiological assays. We've collaborated with a professor at Cornell, and so we're waiting for some of the results from them just due to delays in COVID. And what we expect now that we have the organismal level results is that VSH will really outperform all other genotypes at the physiological level as well. And we also expect that there will be a wider difference in ATPase activity, so that being that they'll not only outperform the other lines, but by a larger margin than we first expected. And this figure on the right shows one of the graphs that we will be creating once we get our results from our collaborators. And so we'll be able to map the amount of resistance based on the different levels of cardenolides in all of those milkweeds. So just wanted to give a great thank you to, again, Professor Whiteman and Professor Blackman, also Dr. Mariana Carriagorgia, who mentored me over the summer, and also Susan Bernstein, Nama Wexler, and Derek Yip. So thank you all so much. And I'll take any questions now.
Well done, Elizabeth. I was wondering if you can go back to the slide where you put the survival rates for the different butterflies.
Yes.
And I assume the one on the right is the one that you're interested in, the monarch. And so that I understand correctly the slide, when you had the lettuce part, or you had some that had no problems, right? So are those the ones that are furthest to the left? Is that the control?
Yes, so we have control here, which
why
is...
are the monarchs so variable there? I was just trying to see why you see what I'm saying? That thing is huge for
on the lettuce.
Yes.
Yeah. And so the lettuce was interesting. So we use lettuce as a control for, I guess, just having plant material in the media. And that was actually like pretty interesting to us. So we are thinking that it's possible that it's kind of like I guess just an artifact of the media being different, because there's possibly other secondary metabolites in the lettuce that possibly interact poorly with the monarch lines. And that's definitely something we have thought was interesting as well. But the thing is since we didn't so we have essentially for all of the milkweed lines, HPLC analysis to see which cardenolides are in those plants and which other secondary metabolites, but we did not do that for lettuce. So that's where it's possible that there's some other outside secondary metabolite that could be affecting that line.
So did you run your comparison with and without the lettuce when you have your final result? That probably should be something you should be doing.
Do you mean like between the control and the lettuce?
I was assuming you were also using the lettuce as a control in addition. The control has nothing, right? The lettuce also looks very harmless, but maybe you think it has something, right? So that when you look at the treatment effect of what you're doing, considering with and without the lettuce, or you don't pull everything, you just do pairwise comparisons. Just wanted to understand the design.
Yeah. So overall, what we did was essentially looking at each line's survival individually. So,
okay, got it.
Yeah.
All right. Any other questions? Did you consider other outcomes other than morbidity, mortality? So, because
so we also tracked the rate of eclosion, which is just essentially a fancy word to say when the flies themselves go from larvae to adult fly. And so what was interesting is there's a bit of a delay for some of the lines between their rate of eclosion and their survival. And so that's like a further analysis that we're interested in for actually possibly publishing, is that there is this different delay also between not just survival, but between the rates of going from pupa to adult fly in some of the lines.
Excellent work, Elizabeth. Congratulations. And I'll ask Daphne Yusi to take the floor and present her work.
Thank you. I'm just going to share my screen. Can you see my screen or no? Okay. So hi, everyone. My name is Daphne, and today I will be talking about conditionally active CRISPR-Cas enzymes and their role in genome and transcriptome editing. I want to say a huge thank you to my research mentors, Brady Kress and Professor Jennifer Doudna, and my faculty research sponsor, Professor Britt Glonsinger. So CRISPR-Cas proteins are revolutionary genome editors because they offer us programmable DNA manipulation. The guide RNA that you see in orange here guides the Cas protein to a specific site on the DNA, and the endonuclease, which are basically molecular scissors, cut or cleave the DNA on that exact location. So changing the sequence of the guide RNA readdresses the protein elsewhere. And this really offers us very specific, highly efficient, and an easy to engineer way of genome editing. However, we sometimes see off-target effects, meaning Cas proteins will cleave at unintended locations, and we don't really have a lot of temporal and spatial control. And to tackle these challenges, we use CRISPR switches, and these are specially engineered Cas proteins to be conditionally active. So when we add a ligand, meaning a small molecule or chemical that binds to a protein domain, we can turn Cas activity to be on or off. And Cas activity turning on basically means we see genome editing. And there are several different ways to engineer CRISPR switches. I classify them based on when we see ligand effect. And as with all proteins, Cas proteins are made as described by the central dogma of molecular biology. So DNA to RNA transcription, and then RNA to protein translation. And we can conditionally control Cas activity during the different steps transcriptionally, post-transcriptionally, and post-translationally. In the first part of this presentation, I will talk about the different CRISPR switch constructs, and some important properties of these will be listed on the screen. However, I won't really have time to go into details, so I'd just like to give you some context beforehand. Reversibility means once you turn the switch on, can you turn it back off? And fold change refers to the magnitude of change between the on and off states. In an ideal CRISPR switch, we want to have tight control, meaning no activity without ligand and high activity when ligand is present. And we also want to have a tunable dose response curve. As I mentioned, I'll start with an extensive review on CRISPR switches and then move on to current limitations and the unexplored potential of these switches. But before moving further, I'd also like to add that it's really difficult to compare the different CRISPR switch constructs and their properties, and to say one is better than the other without benchmarking these against each other. So first step in the central dogma, DNA to RNA transcription and transcriptional control. The first checkpoint in expression of the Cas gene is transcription of the mRNA. If Cas mRNA is never produced, no Cas enzyme can be made. And here on the diagram here, you see that the transcription factor only binds to the DNA if the inducer or ligand is present, meaning if there's no ligand, then we won't see any Cas mRNA produced. So in post-transcriptional control, guide RNA control is one way that we can do this. And there's no translation of the guide RNA. Guide RNAs are considered non-coding RNA because they don't code for protein, but we can control how accessible the guide RNA is. If the guide RNA isn't accessible, so if it has like a bulky part here, as you see on the picture, Cas won't show any activity because it will be non-functional. So one example to guide RNA control is through an aptazyme insertion on the guide RNA. Aptazymes are self-cleaving ribozymes, and the bulky part that you see here is only cleaved off when the ligand is present, and only then the Cas protein will become functional. And on the right side of the slide, you see that I provided the number of primary literature reports by ligands, showing which ligands have been used to control each of the mechanisms. And the next, oh yes, okay, so each color means shows a different ligand, and there's also numbers, but I'm guessing you can't really see them. But it's really the count of how many literature reports use those ligands. And you'll notice in the next slides that you'll see the same colors, meaning the same prototypical ligands are used over and over to control CRISPR switches. For example, 4-hydroxytamoxifen is used in almost every single construct that I will show here. And okay, so this moving on to the second step in the central dogma, RNA to protein translation. When we insert a destabilizing domain to the Cas protein, the protein will become unstable, and like all unstable proteins, it will get degraded, meaning it will be turned off. When we add a ligand, this protein will be stabilized and it will turn on. For degron-based systems, it's quite the opposite. You insert a degron domain and the protein is active, so it's on. But when you add a ligand, it will be marked for degradation and it will be turned off. Another post-translational control mechanism is allosteric regulation. And here we insert a ligand binding domain to the Cas protein. And when there's a ligand present, we will see a conformational change and it will become active. And this looks very similar to the previous two slides, but I think there's a key difference here. Unlike the previous two slides, we don't really have a stable versus unstable state. Instead, we have two stable states, only one of them is active. And the last mechanism that I'll talk about is dimerizing domains and split Cas9. Here, the protein is practically broken into two parts and often have dimerizing domains attached on each piece. And the ligand effectively restores the Cas protein by gluing Cas each of the pieces back together through the connection of the dimerizing domains on the Cas protein, producing a full-length Cas9 and turning Cas activity to be on. And you might have noticed the lack of color in those diagrams that I showed earlier. And that is because we currently only use 17 ligands to control Cas proteins. But how many other ligands could we use to control these? Could we potentially study hundreds or thousands of intracellular metabolites? And I want to say that perhaps for some therapeutic applications, for example, loss of function studies, creating conditional knockouts for behavioral neuroscience, and so on, the current ligands might be enough. But what if we want to use CRISPR switches for intracellular recording to track disease onset mechanisms or small molecule interactions or cellular environments? Could we potentially study hundreds or thousands of metabolites using CRISPR switches? And you might be wondering, what kind of molecules can we use? Are there any constraints on what we can use? And here you see the structures of the 17 ligands that we currently use, and they are very, very diverse from each other. So the key thing here is that there are no structural limitations on which ones we can use. And in theory, we should be able to use any ligand as long as we know the receptor or ligand binding domain pair of that ligand. So after a systematic search, I discovered that so far, all of these ligands listed in their red box here have been used as sensors or switches in other proteins. And this means that there's a really good chance that we can engineer conditionally active CRISPR enzymes using these ligands. And this also suggests that in theory, we could engineer Cas enzymes to be activated by any molecule in nature, and especially those that already have known ligand binding domains. So one application for CRISPR switches in this case would be molecular recording. And the key innovation here is that the Cas protein or Cas9 would be converting the amount of molecular signal, for example, concentration of metabolites, into a proportional amount of DNA editing. So we don't have to break open cells or tissues or animals to take measurements over time. We just read out the edited DNA at the end of the experiments to infer information about the metabolites over time. So it's basically a genetically encoded measurement device that facilitates longitudinal studies in a way that hasn't really been possible until now. So molecular recording could really help us understand important biological questions. And you can see some example on the slide here: tumor microenvironment dynamics or cell differentiation dynamics, bacterial community signaling, and so on. And with that, I'll just go through the key takeaways from today's presentation. Conditionally active CRISPR-Cas enzymes offer temporal and spatial control for genome and transcriptome editing. Currently, only 17 ligands are used to control Cas enzyme activity. And by analyzing previously established receptor-ligand pairs, we've generated a list of potential candidates to control Cas enzyme activity. And we believe that this could significantly expand applications for CRISPR switches. And conditionally active CRISPR-based molecular recorders can allow scientists to record frequency and intensity of metabolite interactions over time without disrupting biological states. And lastly, our future directions include engineering conditionally active allosterically regulated Cas enzymes based on the list that we've generated. And I would like to thank my research mentor, postdoc research mentor, Brady Kress, our principal investigator, Professor Jennifer Doudna, and my faculty research sponsor, Professor Britt Glonsinger. And I'll go back to the conclusion slide to see if anyone has any questions.
Congratulations, Daphne. So go back to the slide, please. For those social scientists like me, any of these processes, are they linked to some kind of fundamental implication of the cell function, so that, for example, a disease, so is there something that, for those that don't quite understand the terms, that you could kind of have as a takeaway? Thank you.
So I think I'll try to explain it better, maybe. So basically, CRISPR-Cas proteins are really used for genome editing technologies. And here we can program these CRISPR proteins to only be active when a specific molecule is present. So for example, when we only have this one specific ligand, it will show genome editing. And if we can engineer CRISPR proteins to work with several different intracellular metabolites, by reading the DNA editing, we can track how those metabolites change over time. So this could potentially or ideally be applied to any metabolite that is active, for example, in a disease or in a bacterial community. I don't know if that made it clear or even.
I believe so, because I was just thinking what the main contribution was, so I think it became much more clear to me. But also Claire is asking a question if this could have applications to cancer treatment and prevention.
For cancer treatment, I'm not entirely sure, but I think I personally think that definitely to track disease onset mechanisms and to see, for example, tumor microenvironment signaling and to understand those better and perhaps to derive, I don't know, therapeutics according to that. I think it could definitely play a role, but it's also, I think, important to note that it's still a very new tool. So that's sort of what we're aiming at, I think, with a lot of these technologies.
So this also required you to be green badged and on campus doing the research?
No, actually, I was able to do all of this remotely because it was mostly a meta-analysis of literature reviews and then taking it sort of a little further by finding other ligands and pairs and analyzing them to generate the list. So yeah, it's sort of changed the process a little bit for me, but yeah.
So you had to pivot. Yeah.
Yeah.
Very cool. Once again, congratulations on honors from our college. And I would like to give the floor to our last presenter before we do a short break. It's Lily Klinek. And I will ask you to please share your screen.
Hi, everyone. Can you guys see this okay? Sweet.
Yes.
Okay, so today I'll be presenting my thesis, analyzing green-up phenology of North American forest ecosystems with Ameriflux and PhenoCam data. And my faculty mentor was Professor Dennis Baldacchi, so I'm very grateful to him for his mentorship. So as we know, forest ecosystems play a really important role in the carbon cycle, and they're actually one of the largest terrestrial carbon sinks. But there's still a lot that we don't quite understand about the land sink in particular. And one of the reasons for this is the variable seasonal flux of photosynthetic activity, which can in part be attributed to the cycle of green-up and senescence of deciduous trees. So on the bottom right, we have a graph showing three mixed forests, or two mixed forests and one aspen forest, and their photosynthetic activity over the course of the year. So they all undergo a huge seasonal flux, and in the late spring and early summer, there's kind of a skyrocket of photosynthetic activity as the forests put out their leaves for the spring. But then the photosynthetic activity sharply declines as the forests drop their leaves in the winter. And so the seasonal flux falls under the discipline of phenology, which is the study of cyclical or seasonal natural events. And in particular, my study focuses on the green-up, which is the date when forests put out their leaves for the spring. It's also the start date of the growing season or the photosynthetic season. And additionally, it's the date when forests transition from a net carbon source during the winter to a net carbon sink. Green-up is often a trigger for ecological events as well, so such as migration patterns or breeding patterns, and its timing also can control predator-prey interactions. But along with these ecological impacts, green-up also has effects on albedo and evapotranspiration, trace gas exchange, and cloud formation as well, due to its effects on the planetary boundary layer of the atmosphere. So the green-up date, especially in recent decades, has been used as kind of a proxy for climate change itself. And it's a really good metric for examining how climate is impacting an ecosystem's biology and metabolism, which would then go on to control the ecosystem's productivity. And many recent studies have shown that green-up is shifting progressively earlier as climate warms. But as can be seen from this figure, this kind of causal effect varies really unevenly across different latitudes and different ecosystem types. So it would definitely require closer study. But the earlier green-up and the delayed autumn senescence that we're observing could lead to an overall increase in carbon uptake with a longer growing season, which would be a good thing since it would help mitigate some of the detrimental effects of our anthropogenic emissions. But on the other hand, longer growing seasons could cause an earlier depletion of plant available water, which would cause water and drought stress on the forests. So that might decrease overall growth and productivity. So it's kind of a complex and multifaceted issue. Green-up is also an important parameter in biogeochemical and agricultural models. So for example, the length of the growing season, which is influenced by its start date, plays a large role in determining crop yield. And additionally, the presence or the absence of a forest canopy controls latent and sensible heat flux. So being able to understand what's driving forest green-up and what's influencing its timing would be really important for improving the accuracy and the implementation of these kinds of models. So that brings me to our hypothesis, which is that the green-up date corresponds to the date when the soil temperature at a site equals its mean annual air temperature. So on the bottom right is an example plot of what we would kind of expect to see with our hypothesis. So the soil temperature in blue is warming in the spring, and when it equals the mean annual air temperature of the region, that's when we would expect to see the green-up date occurring. So this was originally observed anecdotally in Tennessee, and then was tested across many other sites in North America and Europe, and was found to explain 61% of variance. And the mechanistic explanation as to why this hypothesis kind of makes sense is that plants have evolved to use temperature cues to time their leaf out. So they're essentially balancing the potential rewards of an earlier bud burst, like increased growth and productivity, with the risk of frost damage if they leaf out too early. So plant phenologists have modeled this idea with the concept of heating and chilling units. So plants must accumulate a certain number of chilling units from the winter to measure how long the winter was, and heating units in the spring to measure how long the start of spring has been. And together with enough chilling units and heating units, these will trigger bud swelling and burst at the time of least risk. So we're using soil temperature as a proxy for these heating and chilling units due to its thermal inertia, which is its tendency and ability to hold heat. So that kind of explains why this correlation makes sense and why such a strong correlation has been found. So for my study, I wanted to retest this hypothesis with over 50 new site years of data in North America specifically. And I sourced this data from Ameriflux and Phenocam, which are two networks of sites that continually record environmental data. And in particular, Phenocam provides green-up estimates at each of their sites. So by resampling and then analyzing this data, I was able to test the strength of our correlation across the entire site group, which is listed here in my site table. And so across this group, we were able to kind of find examples where our hypothesis worked really well and then examples where it broke. And so one of the latter was at Willow Creek, Wisconsin, where, as we can see from these graphs, the soil temperature is crossing over the mean annual air temperature consistently early compared to when the green-up is occurring, which is happening later. And a potential explanation as to why this is happening here could be due to the higher latitude and the colder winter climate. So out of the entire group, this site was the one with the lowest mean annual air temperature, followed by the University of Michigan Biological Station, which also fared pretty poorly. But sites in lower latitudes had much stronger correlations. And the best example that we found was at Missouri. And we can see over many years that the green-up date is occurring very precisely when or very close to the date when the soil temperature is crossing over the air temperature. And sites in other similar latitudes, like in Tennessee and Indiana, also did very well. So that suggests that climate and latitude plays a large role in determining whether our hypothesis works or whether it breaks. And when I considered all my sites, I found that our phenological rule only explained 11% of variance. And so the highest latitude sites here were kind of causing the problem, as well as one California grassland site, which we think was experiencing like more of a warmer spring and more effects of drought and warming. So by removing these sites, the R squared was allowed to increase to 76%, which shows a really strong correlation. And there are two potential sources of error that could be at play here. One of them was that the different sites in our study measured the soil temperature at different depths in the soil. And so that wasn't standardized across all of our sites. And then the other was that PhenoCam's green up estimates are only kind of a proxy for the actual quantifiable green up date. And so using them here definitely increased efficiency and accessibility for testing this hypothesis, but it did kind of sacrifice some of the precision that you would gain from using actual carbon exchange measurements. So my main conclusion was that the higher latitude sites, though they're not actually in the boreal forest zone of North America, they could represent or be creating a boreal adjacent zone where the more extreme winter temperatures and the snow cover that's kind of lingering later into the spring would render our phenological rule kind of inapplicable. But my findings do suggest that this rule can be applied to a vast number of other sites, especially with the implementation of field validation to correct for some of the errors that could be at play here, like the varying depth for recording the soil temperature or the estimates that GreenUp is, or that PhenoCam is providing for the GreenUp. And in particular, with the lower latitude eastern deciduous broadleaf sites, this method could be really helpful in generating wide GreenUp estimates, which can then be used to refine biogeochemical models or agricultural models that might require phenological input, or to estimate and ultimately predict forest carbon cycling. And yeah, thank you so much for coming. I would like to thank my faculty mentor, Professor Baldacci, and everyone in the Biomet lab. And I'd love to take any questions.
Lily, I was wondering if you considered any characteristics of the forest other than latitude. And
yeah, so if I can go back, let's see. I'll go back to my site table. So the different forests that we were considering here, along with latitude, they also had kind of different content in terms of the actual species of trees that were most common. So a lot of them had oak as like the most common species of trees, but most of them were mixed forests, which means that there are many different species of trees. And so obviously these different species could be responding really differently to climate cues. So I think if I were to kind of like repeat this study, I would want to focus in on that and really do some more studies with different species that are the most common in these sites. But generally, I was kind of just trying to test this really broad hypothesis and consider how like latitude and the spatial like variation between the sites was affecting how the hypothesis worked. Because I think kind of the main end goal for this like correlation or this rule would be to just use it really broadly across many sites to generate like broad estimates for when greenup or predictions for when greenup is occurring. So I was kind of focusing more on latitude rather than like focusing in on the tree species. But I think that would be really interesting to do for like a separate study.
We have a questions from Professor Baldacci.
Oh, very, very nice job. So quick question, at the high latitude, do you think day length could play a role? And did you look at day length?
No, I didn't, but I think that would be really interesting to consider too. I know like there are some studies going on that are testing phenology of greenup at even higher latitudes. So like latitudes in Canada, and those are actually in the boreal forest zone. And so I think testing that there, even though we kind of have seen from this that it wouldn't work as well, that would be really interesting to kind of develop like an alternate phenological rule for those kinds of sites where this might not work. And that could factor in like other factors like day length to try to, yeah, predict greenup better at those types of sites.
And other than the count of the number of days of temperature, soil temp,
greater than 75, did you also do consecutive days other than the number on that graph that you plot?
Let's see.
Was that the consecutive days with that temperature, or was it the number of days with the R squared, the one that you have the?
Oh, right. Okay. Yeah. So with this graph.
Yeah, on the x-axis, are those consecutive days or just the count?
Those are not consecutive days. That kind of refers to the day of the year that cross date occurs at.
I see. Would you consider just doing the cumulative days? Does that affect growth?
Like the cumulative days that those temperatures equal each other?
Correct. Or
yeah, so I think for our study, we were kind of focusing more on that first cross date to show like when the soil had warmed enough for maybe the trees to have accumulated their chilling units or their heating units. I think if you were considering like cumulative days when the soil temperature equaled the air temperature, I think that would be different and more testing like how long the climate stays the same. But I think we were kind of focusing on this cross date as like a cue for when the soil had warmed enough to reflect that those heating units had been accumulated, if that makes sense.
It does. Thank you.
Sure.
And you also mentioned that it varies on the depth of the readers for the temperature. Can you control for that if you know which ones are deeper than others, and you could put an indicator for when you know these units come from these kinds of, because I think you have a panel data set, right? By site, by year.
Yeah, so yeah, that definitely could be something to try to control for. I could have like just eliminated the sites from my study that were kind of missing some of the soil temperature depths. Some of them kind of provide like multiple depths, like 2 centimeters, 4, and then so on. So you can kind of select which ones you would rather study. So I could have eliminated sites that like didn't have the soil depth that I was looking for. But I think I just wanted to get a really broad and multi-site study. So I didn't eliminate those sites and I decided to just keep them in regardless. But yeah, definitely that could be something to control for, to either remove those sites from the data set or to do some separate study to see if that could be causing the errors. Yeah.
Thank you. I will ask if you want to just socially just talk a little bit among yourselves, panelists, and then we'll make a break at 10:38, and everybody has a time to stretch. And also the students, the honor students that are coming after the break, during the break, if you want to try sharing your screen, that would be fantastic so that we know we're good to go. Congratulations, everybody. Sarah, do you want to try sharing your screen, or who is it now? Oh, okay.
Okay, should I share? Sorry, Anna, I know that you're
fantastic. I think you're good.
Wonderful. I think I'm good. Yeah.
Can you scroll down just to see if you can switch the? Yay!
Yep.
Fantastic. Michael, do you want to try too?
Yeah, sure.
Welcome. All right, welcome back, everybody. We have the pleasure to kickstart our second half of the morning session with Sarah Shu, and Sarah is mentored by our very own Professor Brian Wright. And so we want to have Sarah, you have the floor.
Awesome. Thank you so much, Sophia. So I did my project on an analysis of energy consumption during COVID-19 in the PG&E service area. And before I continue, I would like to thank Professor Brian Wright for being a phenomenal mentor and all of my friends and family who supported me through this process. So some background on COVID-19's impact and the energy consumption in the state. As evidenced by us all being online, COVID-19 has really changed how we live our lives. California saw high unemployment rates throughout 2020, and the California Employment Development Department reported that each month in 2020 saw higher rates of unemployment than the 2019 average. California also saw high rates of infection. The bottom right graph shows the number of cases in March 2020 till now by county in the San Francisco Bay Area, and you'll notice that there's the initial increase in cases is July through September 2020. We also know that low income and communities of color face inequities in energy costs and consumption through previous research. Historically, research has shown that communities of color are energy burdened, meaning they pay a higher percentage of their income on energy. And furthermore, reports and research that we're already seeing about the pandemic's effects has indicated that disadvantaged communities, and specifically communities of color, have been disproportionately impacted by the pandemic in terms of unemployment, infection rates, and some research out of Illinois is already showing this disparities in energy consumption. With this in mind, I set out to answer the question, how has energy consumption changed during the COVID-19 pandemic? This question was broken down into two main parts. The first one is what are the impacts specifically on commercial consumers? And then the second one is, did the pandemic disproportionately impact disadvantaged communities? So a bit on data sources and my methodology for this research. I obtained data on energy consumption by PG&E customers, which is publicly available by customer type. There are three main ones: commercial, agricultural, and residential, and by zip code. The PG&E service area goes as far north as Humboldt County and as far south as Santa Barbara County. And this data is aggregated by zip code to report average and total energy use by kilowatt hours. Furthermore, the data is only reported if there's at least 100 residential customers or 15 non-residential customers. Otherwise, the customers get counted in surrounding zip code data. And since we're looking at energy consumption for a zip code in a specific month and not day by day, we don't actually expect to see the widespread impacts of the pandemic in California until the April 2020 reported data. This data also, since it's PG&E service area, does not include those who generate their own power, such as households with residential solar on their rooftops. The other data source that I used was CalEnviroScreen. CalEnviroScreen 3.0 is a mapping tool. This tool generates a score on socioeconomic and environmental factors such as birth weight, pollution burden, education rates, and water quality for each census tract in the state. And this score uses 20 indicators to map California's over 8,000 census tracts. The higher the score, the more burdened a community is. And the score is important because the state uses the score to help distribute cap and trade funding. The census tracts that score in the top 25% are officially considered disadvantaged communities. And these communities are marked in this map as the dark orange and red census tracts. For this research, I combined these two data sources, and when zip codes had at least one census tract listed as a disadvantaged community, I designated the entire zip code as a priority community. With this data, I conducted a time series analysis as well as looked at the mean changes each month by month on the reported average kilowatt hour for each groups of customers and conducted a t-test when appropriate. So we'll move into findings for commercial customers. On the left hand, you'll see an average energy consumption by commercial customers. And on the right hand is the total use. And as you can see, for both average and total energy consumption in the commercial customer group, they had similar energy consumption trends in 2018 and 2019. That is the red and blue lines in both of these charts. And there's a repeating pattern throughout the seasons, similar variability and trends. However, in 2020, there was a huge drop in energy use starting in March 2020 and slow increases, but not to the original energy use levels throughout the year. And interestingly, the total energy use shows that there was a large drop in energy consumption again in November 2020, which is not something that we see when looking at just the mean energy consumption. All right. So moving on to residential customers, the graph on the left, so the violet graph, are the priority communities. So these are communities, again, with at least one census tract that is considered disadvantaged. And on the right are the non-priority communities. And we see a pretty distinct seasonal pattern. The y-axis is the same kind of length. And we see a distinct seasonal pattern with overall pretty similar trends and variation across all of the years. The darker line for both, so the dark red here and then the dark green here, indicate 2020, which you can see is higher than previous years for most of the months, though not as striking as commercial energy use. The seasonality is also that we see with 2020 is found in other research about energy consumption in California. Specifically, priority communities consume more energy in the summer months and less in the winter months compared to non-priority customers. And this is like I said before, in line with previous research that finds that richer households are more likely to have energy efficient appliances, generate their own solar electricity, especially in summer months, which we see in these two graphs. So what the previous graphs did not really show is the strength in difference and how much more energy was used by residential customers in 2020. So this table shows the month-by-month mean energy use change by customer type in the time periods. Red and orange denote strong and medium negative change. Yellow indicates little change, and green and dark green indicate strong positive change. The top rows here show the change between 2018 and 2019. As you can see, throughout the months, the change is kind of mixed in strength and direction, whereas for 2020, from between 2019 and 2020, there's a strong overall positive change starting really in April 2020. The months in which change are not significant in this table are marked in purple. And with this month-to-month change, I found that priority communities did slightly increase energy consumption more than non-priority communities. This brings me to kind of the conclusions. So the COVID-19 pandemic caused many of PG&E's commercial customers to ramp down their energy consumption in early and mid 2020. Total energy consumption by commercial customers began to decline again in October 2020, unlike the average energy consumption. And I think therefore it suggests that the growth in average energy consumption in October is not an indicator of businesses returning to business as usual, but rather customers shutting down and leaving the market overall. All residential communities increased energy consumption in 2020, with priority communities increasing consumption slightly, but also statistically significantly more than non-priority communities, which would indicate a disproportionate impact of COVID-19 or some sort of difference happening between these communities. And historical residential energy consumption patterns, both the seasonality as well as the differences in energy consumption, still hold true in during the COVID-19 pandemic. I think it's really important to address some of the limitations of this research. The first one is the data that was used is not very granular. It's a month average or month total. It's not day by day or individual customers. And that data would be interesting, would be very interesting to have in an avenue of further research. But this data used for the research was aggregated as an average of an entire zip code. And the second limitation is communities aren't weighted by the population size of the disadvantaged communities. This could be masking energy use changes in disadvantaged communities that are in larger zip codes. For example, in Berkeley, where we have disadvantaged communities in a certain census tract, and right next door, a census tract that has really high income, but it's all in the same zip code. Many other variables can also explain these findings, including COVID-19 rates, employment metrics, geographic location, and variations. These limitations could also open up further research pathways, though. So as mentioned before, granular data would open up research about the immediate impacts of shelter in place, or we could regress household characteristics with energy use. Additionally, the California alternative rates for energy, so the CARE program customers, could have been differently impacted. The CARE program provides a discounted rate for electricity to qualifying low-income households. And also energy consumption does not necessarily translate into energy kind of burden. So it'd be really interesting to look at both the CARE program and also energy burden as two factors that can explain higher energy use in priority communities, especially since PG&E had a moratorium on utility disconnections during this time. So there could be a high utility debt right now that we aren't seeing translated into consumption. I think it would also be really interesting to see for further research. Does energy consumption show a compliance with shelter in place rules, which really varied by county? So further research is definitely needed and important to truly understand how businesses are doing in the pandemic and inequities in energy use in California. But that is my research. So thank you so much for your time and for listening to this presentation.
Spectacular, Sarah. We'll open the floor to some questions.
Hi, Sarah. Yeah, very nice presentation. I think one of the interesting things about this is I think you showed a difference between the average consumption and the total consumption for commercial in October, right? So could you just go over that again? I think you might have said that the total rose, but it's actually, I think what you meant to say was that the one on the right is falling dramatically, right, in October.
Yeah.
Which may be the most interesting thing. It seems to suggest that some businesses are just stopping, right? The average is not moving like that. I think this is very interesting, and it may give a sort of an indirect insight into something that really bad that was happening for commercial customers at that time. So thanks very much for that.
And
thank you so much.
You don't have more granularity, right? You only have commercial. You don't have by type.
Yeah, by commercial type is not something that is reported, but it'd be really interesting to do like individual outreach and research with businesses. I think it could be done in a smaller scale.
I know the zip codes are very big, but could you interact a zip code with the commercial consumption and a zip code that has a lot of commercial stuff with one that has less, or commercial with more I just trying to see if you could at least try to proxy for some zip codes that had a lot of them, and then you can pick up closures, because if you see less consumption for those that have a lot of businesses, it's probably consistent with closures due to the pandemic.
Yeah, I would imagine so, that there are proxies for figuring out kind of different commercial types and how they've closed over time.
I think some of the zip codes with relatively small populations, it might be possible almost to guess what kind of businesses they are, right? Like you probably know in some of these rural zip codes, they're not going to be like manufacturing enterprises or something. They're going to be probably, you know, just the downtown. They may only be like, I think the minimum is 15 commercial customers, not to be anonymized, right? So
well, 94720, it's UC Berkeley, right?
Yeah, that's right. Yeah, but I mean, in the countryside, there must be some with relatively small population of businesses, and you probably can guess that they're more likely to be the sort of lightweight commercial, more like consumer sales and things like that. I'm guessing, maybe hairdressers, maybe.
Definitely an avenue for further research.
Yeah.
Well, thank you again, Sarah. Congratulations, and I'll turn the floor to Michael Quiros, who is going to present his work, also in energy. So let's see. Do you want to share your screen, Michael?
Yeah, thank you. Can you see the slides full screen? Cool. Trying to get Zoom out of the way now. There we go. So hi, my name is Michael, and my project was on evaluating the efficiency and distributional effects of net metering policies and alternatives in California. So originally, my research goal was to quantify the effects of net metering policies on electricity rates in California's 3 investor-owned utilities, using difference in difference econometric regressions. But I ran into a few problems. Even after having designed like a quasi-experiment, I realized there was a lot of data missing from a FERC database that I was going to use to inform my target variables. And it was going to generally be kind of hard to control for a bunch of omitted variables. So the research goal kind of shifted halfway through this semester to summarizing existing academic literature to sort of detail the regressivity of net metering policies in California, evaluate the validity of a few different pro-net metering arguments I heard from the solar industry, and then analyze some potential policy alternatives that might be a little more effective than the current retail rate M tariff. So to start with, what is net metering? How does it work? Net metering is a billing mechanism that credits solar energy system owners for the electricity that they generate and sell to the grid. So for example, if you put rooftop solar on your home, your utility would purchase any excess energy that those panels generate and then redistribute it to other customers. There's 3 ways that these policies are usually differentiated state by state, and it's in how the generators are compensated. They're usually, for each kilowatt hour generated, you can be compensated at more than the retail rate, less than the retail rate, or at the retail rate of energy in that area. And 40 states plus DC and 4 territories, all of the dark blue states in this diagram, have mandatory net metering policies, most compensating at or above the retail rate. And a reasonable line of thinking might be, if we're buying energy that would otherwise be wasted and encouraging renewable development with no direct government subsidy, what's not to like? But a common critique of this policy is that there's a pretty regressive cost shift inherent in it. And that's because of how utilities in California recover costs. Generation, transmission, distribution, pollution, etc. All of these things are recovered through volumetric per kilowatt hour charges. And because of this, retail rates are usually much higher than wholesale rates. And when utilities are required to buy energy from net metering customers at the retail rate, they lose revenue that they would have earned if that kilowatt hour was purchased at the cheaper wholesale rate. And if utilities are unable to recover the costs of operating and investments, all of these things, they might raise electricity rates for all customers, including non-solar owners, to compensate. And this is truly regressive in both senses of the word. It benefits the wealthy. Solar owners are wealthier than non-solar owners. Rooftop solar has recently become a financial benefit, at least in the past 5 years. And some studies have shown that solar subsidies usually are distributed to wealthier areas. And this policy can also hurt the poor. Non-solar owners typically pay higher rates to recover these costs imposed by NEM customers. And they also spend a greater portion of their income on energy and thus the NEM imposed costs. So what's going on in California specifically? In 1995, California established a statewide retail rate compensation for net metering. And there were some incremental changes between 1995 and 2016, but the most significant changes to this policy were in 2016, when things like the capacity ceiling were removed, all generators were moved to a time of use rates, and they were charged a small interconnection fee. But throughout this entire process, that retail rate of compensation has been maintained as the norm. But in 2021, the Public Utilities Commission expects to update the rules concerning customer compensation, which makes the debate over net metering policies both timely and fierce. So in this plot, all I did was sort of correlate the cumulative net metering solar capacity in California, these like aquamarine bars. They've been increasing over time. And as you might expect, as they've been increasing, the median residential solar price has been decreasing. And at the same time, the average retail price for energy in California has been going up. This definitely doesn't imply causation, but there's like a pretty clear correlation between these three variables. So yeah, onto the crux of my paper, pretty much evaluating some of the various arguments that the solar industry has put forward. One common one is that increasing the capacity of distributed resources, such as rooftop solar, with net metering policies will not only decrease the costs associated with losses, but also the need for expensive transmission and distribution investments and maintenance. And this is partially true. If you site generation closer to load, that's going to result in lower transmission losses and congestion, which in turn could potentially reduce the need for grid maintenance and capital investments. But these benefits really aren't enough to justify the cost recovery issues that have been found at the moment. There was a 2016 study, it was peer reviewed, that found these benefits to be less than 0.4 cents per kilowatt hour when they were levelized. So it's just not nearly enough. Although it is possible that electrification will amplify the magnitude of these benefits, and some degree of electrification certainly seems to be in the cards for California. So yeah, at the moment, the benefits associated with reduced grid maintenance and investment do not outweigh the costs of net metering. And I don't expect them to in the near future until there is some degree of significant electrification. Moving on, there's another common argument that the environmental benefits of rooftop solar, being a non-fossil fuel generating source, might outweigh the cost recovery issues. One issue with this is that other policies already internalize the benefits of solar, and methodologies like that incorporate these environmental costs often double count the benefits that are already accounted for in state and federal tax credits, stuff like that. Also, rooftop solar, if you look at this like levelized cost graph, out of all of the renewable generating types, it's got by far the highest levelized cost of energy. So not only is it already subsidized, but it's also possibly the most expensive way to decarbonize the grid, and net metering still doesn't really make sense. Finally, there's an argument that I've seen that increasing solar capacity will displace high-cost oil and gas generators, especially at peak demand times. What I did here was just take a random sample of data from days over the past year for CAISO, and plotted the average solar generation profile against the average demand profile and day-ahead prices for NP15. And if you look, this dark blue is solar. It peaks between hours like 10 and 14-ish, which really isn't when demand is peaking later in the day. It's really not aligned with demand and those higher prices. Only a little bit of solar is actually online and generating then. There's also the issue of curtailment. I think there's kind of an oversupply in standalone solar at California at the moment. Here, each line represents how much solar is basically being thrown away during this peak period, cumulative. So in 2020, over 1,400 gigawatt hours of solar were just thrown away. So on the whole, solar production doesn't really seem to align with peak demand. and continuing to compensate at the resale rate to increase solar capacity would only add to the current oversupply and would fail to incentivize efficient investments in energy efficiency or storage technologies that might be able to sort of mitigate this curtailment. So yeah, all of that in mind, what might be some good policy alternatives? One, you could just compensate at the wholesale rate, because rooftop solar owners only contribute generation, they don't pay for distribution or transmission. This seems like a simple solution. Alternatively, I think this one's kind of interesting. You could change California's rate structure as a whole. Instead of using volumetric rates where the price of energy is set to the average total cost needed to generate that electricity, you could charge the social marginal cost and then recover these fixed costs through monthly fixed charges that could potentially be waived for low-income households. Then there's no longer a cost shift when charging at the resale rate, and that pricing at the social marginal cost would encourage electricity production and consumption that actually maximize social welfare. It's kind of a better price signal. And yeah, so the conclusion is, despite accounting for the benefits associated with reduced grid investments, cleaner generation, and lower marginal generation costs, California's current net metering program seems to be pretty regressive and inefficient. And compensating net metering customers at either the wholesale rate or switching to a nonlinear rate structure, I thought were pretty viable and attractive alternatives. Yeah, special thanks to my advisor, Professor Sofia Villas-Boas, and Professor Meredith Fowley, who both helped me a lot on this project and were also just great mentors throughout college generally.
Great job, Michael. Feel free to ask questions and unmute. Brian, you're unmuted.
Okay. First of all, you mentioned electrification. I think is what you meant, changing gas appliances to electric in households. Is that what you meant by electrification? Yeah,
exactly. That, or similarly changing transportation to more electrified vehicles, public transport, that sort of thing.
Okay, yeah. And yeah, secondly, something like a few hours of battery storage might help shift the demand, right? It'd be interesting to see how that develops.
Yeah, definitely.
And the third thing is, if you really did pay the wholesale rate, would people still be willing at the margin to put more solar on their roofs, or would it become uneconomical?
I think it depends from state to state. I would argue that there's more efficient ways to encourage solar adoption. If your goal is still to have like the same level of residential solar, you could maybe reduce certain tariffs that make it more expensive to bring solar into the US. You get rid of some red tape, just things that don't generate this sort of cost shift. But that's a really good question.
If it's really uneconomical, though, maybe it's an undesirable kind of solar, right? Maybe it's just be interesting to know whether it's really potentially economically viable or only viable if you have subsidies.
Yeah, definitely.
Nice presentation.
Thank you again, Michael, for an excellent point-by-point statement and very carefully designed arguments. I'm really impressed by your work and how you were able to pivot despite the data not cooperating. This is part of research, as we all learn, right, Michael? And you kept going, and it was spectacular. Very proud of you. It was a pleasure also to have been knowing you for so long and working with you. And I cannot wait to what you're going to be doing next, as well as everybody else in this session. Alejandra Marquez is next. And if there are any guests that do not want to wish to be called Sofia Villas-Boas, feel free to rename yourselves and then identify and ask questions. And again, Alejandra, you have the floor.
Okay, thank you. Can you see my screen okay? Perfect. Hi, everyone. Thank you for being here. Today, I will be talking about my research on information disclosure and climate-friendly consumption. And I will be sharing how I assess the impact of carbon labels at Crossroads, which is one of the dining halls here at Berkeley. First, I just want to give a special thank you to both my faculty sponsor, Professor Timothy Bowles, and my mentor, Professor Ricardo San Martin. So I was motivated to undertake this research because animal consumption is a large contributor to climate change, and shifting consumption patterns in developed countries has been identified as essential to meet climate change mitigation targets. Yet the best strategies to actually make this shift in our consumption are lesser known. Despite the large role that food consumption plays in climate change, evidence suggests that most people are unaware of the environmental impact of food products. So I wanted to know how information disclosure through carbon labels can close this knowledge gap and guide us to make better food choices for the planet. In the years since I started this research, climate labels have actually become more common, with restaurant chains like Panera Bread, Chipotle, and Just Salad starting to add climate-related information on their menus, which is all new. So specifically, my research seeks to answer the question, what is the effect of a color-based carbon labeling scheme on food choices at a university dining hall? The first step for this project was to calculate the carbon emissions for every ingredient used by Cal Dining, and those were a lot of ingredients. I led these efforts last summer as part of my position on Cal Dining's sustainability team with the help from David Sanchez, another student, and Sam Lebeau, sustainability coordinator at Cal Dining. Our calculations included all upstream greenhouse gas emissions from agricultural supply chains, except emissions associated with land use change. And we calculated the emissions for over 3,000 ingredients. And then a software program made the calculations to the actual dishes, for the actual dishes. So the research itself consisted of a control phase before the labels were added and a label phase, which both lasted 14 days. And in both phases, the same menu items were offered at the dining hall, allowing for the observation of students' food choices of the same menu before and after the labels had been implemented. So these are some images showing the labels that were added at Crossroads. As you can see on the left, each dish had a label next to it except for pre-ordered items such as daily soups, desserts, fruit, and drinks. The image on the right shows an educational poster that was also put up inside the dining hall to show students what the labels mean and put the emissions in the context of miles driven with a visualization of the impact of each label color. The labels were also added to the online menu, and we created an informational page on carbon footprints within Cal Dining's website. As for the design of the labels themselves, I chose this traffic light colored labeling scheme based on evidence from the literature indicating that this type of labeling scheme makes it easy to compare between food categories, and it's also preferred by consumers very often. So here you can get a sense of the scale of Crossroads operations and the size of the data set that I was working with. The meal count is representing the number of swipes, which is a proxy for the number of people coming into the dining hall and swiping in. The data I looked at for my research were the number of individual servings. So this is 100 grams of each dish, which you can see here in the middle column. The number of servings per swipe is higher during the baseline period, showing that students were getting more servings on average during the baseline phase at the start of the semester than later on in the semester. And this might be explained by the tendency to eat more in a buffet style setting when there are many new food options, as is the case with first year students during the start of the semester. And I just wanted to show this data to see if the carbon labels had an impact on how many students were coming into the dining hall. But since there are a lot of factors for why more students would have come to the dining hall, it was just something that I wanted to look at. So I will now present the findings regarding the impact of the labels on food choices. First, I will go through the distribution of choices across label color. Then I look at the distribution and selection of dishes. And lastly, I will talk about the effect on greenhouse gas emissions. So when looking at the distribution of label colors, we see how this changed between the baseline and the experimental phase. And we can see that green labels decreased by around 4.5 percentage points, yellow labels increased by roughly 7 percentage points, and red labels decreased by 2.5. I expected red labels to decrease as people moved away from the high emission labels, which is exactly what happened. However, for the green labels, I was expecting them to increase as people chose more of the low emitting options. The green labels moved in the opposite direction from what I expected. This could indicate that the labels only motivated students to move away from red dishes and choose more of the yellow dishes instead, with the option between the yellow and green dishes not really impacting their choices as much. And this is plausible because from the visualization of the label's emissions, we can see that yellow dishes are much closer in emissions to the green. So students might feel like they are already making a sustainable choice by going for the yellow. And you can see here that students were looking at this poster and they could see that low and or that green and yellow were much closer, and then red dishes were a lot higher in emissions. So for the second part of my analysis, I wanted to look at food choices in dishes with direct substitutes, by which I mean dishes that had a high emission version as well as a low emission version. For example, for this first one in the graph, the high emitting dish was chicken tenders, while the low emitting dish was vegan tenders. You can see that 5 out of the 7 substitutes in the data moved in the direction favoring the low emitting dish. So in other words, between the high emission dish and the low emission alternative, students were choosing the low emission alternative more often when the labels have been added. And lastly, I looked at the impact of the intervention on overall greenhouse gas emissions. This graph breaks down the greenhouse gas emissions for each label color and shows the average emissions per serving for the baseline phase and the experimental phase separately. The gray bars at the bottom show the overall emissions for each phase. And you can see that there was a significant decrease of 0.21 kilograms of CO2 equivalent in the average emissions per serving, with a confidence interval of 0.18 and 0.25. And this represents a 13% decrease in average emissions. From the bars with colors, you can see that the red dishes are really the ones driving this reduction in greenhouse gas emissions, since the emissions for red dishes decreased by a lot between periods. This tells us that a reduction in red labeled dishes is most important to lower overall greenhouse gas emissions. And the changes in green and yellow dishes really had no effect on emissions. When we think about the scale of Crossroads and Cal Dining in general, a decrease of 13% in average emissions really does go a long way. Crossroads serves around 1,300 meals per day, and if we estimate that each student is getting around 5 servings per meal, that would be a reduction of 1,365 kilograms of CO2 equivalent in one single day. So to conclude, I think that the intervention was successful because greenhouse gas emissions decreased significantly, which is ultimately the goal of adding carbon emission labels. While there are limits to the impact of carbon labels, this research shows the potential for information disclosure to shift food choices toward sustainable options. I also presented these findings to Cal Dining managers who are excited to move forward by adding physical labels to the menu cards of every dining hall, not just Crossroads, and also collaborating with other dining operations at other universities to share our process models and findings. And one limitation of this study is that it focused on only the short-term impacts of the labels, and there's evidence to suggest that these impacts might subside with time. So further research is needed to look at the long-term effects. And lastly, I want to give a huge thank you to the Cal Dining team who helped put up the labels and gather data, and also taught me how to use their menu software and walk me through all the logistics of their operations. So Sam Lebeau, Chef Marcos, Neil, Peter, and Liam are really the heroes of this project. Thank you all for listening.
Well done, Alejandra. This is a spectacular project. Data collection to classify the products, really, really impressive. I have one question and maybe a suggestion. I used, first of all, the findings that you see that the movement is drop in red is fantastic, and also drop in green, and then an increase in yellow. It's really puzzling. and kind of interesting. And I have a similar study where I did something with fish and sustainability, and where we find exactly the same thing, that yellow moves and red and green move really weirdly. So this is one comment I want to give you, and a suggestion that the data you have, I know people use student cards, but there is no way for them to track anonymous student usage, right? It's just the totals that you use, correct?
It's just the totals, the number of servings that, so the servings that are given out.
But you have it at Crossroads. That's the data you used, but you just told me they didn't implement it everywhere. So would you consider adding, for example, Pat Brown or one of those others as a control group during the same time you did everything? They have the exact same products probably in the menu. And so then you don't have just a comparison within Crossroads, but you also would net out anything else that could be happening with that control group. Just a suggestion.
Yeah.
And I'll follow up with by email. I'm really interested in this research, and I'm very excited. Thank you for presenting.
Thank you.
Feel free to also comment on if you had any other points to add.
I'm just looking at the chat.
Yeah.
Thank you, everyone.
Between yellow and green labels, could flavor might explain the difference?
Between yellow and green labels. Yeah, I did want to, you know, when I saw that difference, like I wanted to look back on like specific dishes, and that's just the specific dishes, and seeing if there might have been, you know, something that impacted why the yellow labels increased so much. But I think that, you know, whenever you have a change in the margins of a category, so we see a decrease for green and for red, we would expect the middle part to increase. So I think that the important parts here is that green did not increase, so it decreased, and the red labels decreased. And then the yellow is more explained by like what's happening in the margins.
Well done. And so I'm assuming you did this all before the pandemic.
We actually, the labels were added during the pandemic.
So then the representativeness is the guys that stayed on campus?
Yeah, the unfortunate part was that I wasn't able to, you know, go into the dining hall myself because I wasn't even in Berkeley. But yeah, it did happen during the pandemic, and that's also another limitation that I noted, that, you know, like food choices are impacted by a pandemic and by, you know, things that are happening. So also looking at these results in the context of a pandemic and not generalizing to everything.
But if Cal Dining is willing to do this, let them know to do it not everywhere at the same time, so that they can have at least a control group setting where they can measure what would happen in the absence of the labels. Well done. Any other questions? And then if not, we'll switch to Selena, who I will ask to please share your screen. Selena Wang. Great job, Alejandra. All right, Selena, you have the floor.
Sorry, am I unmuted? All right, awesome. Oh, no. Sorry, wait.
You are, we could hear you. We cannot see you, but we could hear you a little bit.
Okay. Sorry.
Now also we're not seeing the presentation. We're seeing, I think, your background.
A tent and a sunset or a sunrise. We have time. You're running ahead, so.
All right. I don't know where my video came.
You can't share the video, that's also fine.
Yeah, and I can try to see if I can make your video go. Let's see.
Yeah, that would be great.
Ask to start video. I just asked you to start video.
Okay.
Yay, we can see you.
Great, yeah.
Okay. Hello, everyone. So my name is Selena, and I'll be doing my presentation on whether Gen X is a safer alternative to PFOA. And so before I start, I wanted to just give some special thanks to my sponsor, Matt Small, who kind of helped mentor me throughout the way, as well as my faculty sponsor, Professor Roland Holst. And so this is the agenda for today's presentation. First, I'll go over a background information. Then I'll be explaining my comparison framework. And then lastly, I'll be doing a discussion and giving some of my concluding remarks. So what is Gen X and PFOA? Well, these are types of PFAS chemicals, and PFAS, which stands for per and polyfluoroalkyl substances, are a family of man-made chemicals used for its water, heat, and oil-resistant properties. It's used in all sorts of applications ranging from non-stick to firefighting foams to waterproofing. And there are actually hundreds of these compounds, but today we're probably going to be focusing on PFOA and Gen X. PFOA is one of the two most widespread and well-known PFAS compounds, whereas Gen X, a trademark name for the chemical, was made specifically to replace PFOA, as it was, quote, a safer alternative. So PFAS has found its way into our carpets, household items, and food packaging. So surprisingly or unsurprisingly to people, according to the CDC's surveys, PFAS is actually detected in the blood of most Americans. And so this is especially problematic considering that PFAS are persistent in the environment, meaning that they don't break down in the environment, and they are also bioaccumulated. In terms of policies, there are actually very limited regulation on PFAS chemicals in general. And there are actually no federally enforceable standards for PFAS in drinking water, which is one of the main exposure pathways within the US. Regulatory structure is still being developed by the EPA through its action plan, though it still is generally focused on two PFAS chemicals, one of which is PFOA. In absence of federally enforced water standards, a couple of states have kind of taken the initiative and adopted and or proposed its own drinking water standards, though it is usually focused on more long-standing PFAS compounds, such as PFOA, and oftentimes Gen X is not in that conversation. And so I'll be talking about my comparison framework that I'll be using to kind of analyze the risk. So hazard times exposures is the framework that I'll be using for today's presentation. Hazard is the potential for a chemical to produce adverse effects, whereas exposures is how a chemical is able to move through the environment and enter the human body. Oops. For hazards, we'll be looking at chemical toxicities, toxic kinetic health effects. And so toxicity is in terms of cancerous and non-cancerous chronic health effects. And these are indicated by the cancer slope factors and reference doses. These values are extrapolated through dose response studies done on mice. And these values are also used to help derive screening levels for drinking water. On the chart on the right, the column in bold represents which chemical is quote unquote better. But in general, we see that Gen X does appear to have lower levels of toxicity. But in all honesty, the differences between the two are fairly small. For toxicokinetic health effects, studies found that high doses of Gen X display similar health outcomes when compared to that of PFOA. And so it is important to kind of mention some limitations. One is that we're making extrapolations. The first extrapolation we're making is that we're using studies that look at high dose, long exposure periods, and we're extrapolating that to low dose, long periods of exposure. And the second extrapolation is we're comparing the rat effects and extrapolating that into human effects. And then the last thing to note is that a lot of these Gen X's toxicity values I have listed are still draft values, whereas the PFOA values are published values. But in summary, GenX seemingly is better when we're looking at hazard factors, but not much. So next I'll talk about the exposure. So exposure pathways are how chemicals travel and move through the environment. And the figure on the top right depicts this. GenX and PFOA have very similar pathways simply because GenX was meant to replace PFOA. However, there are differences when we're looking at the potential water treatability when we're comparing GenX and PFOA. And this is really important to note. As seen on the bottom right chart, comparatively, GenX has a higher solubility, meaning that it dissolves in water much more easily. And it also has a lower absorption, meaning that it would be a lot more difficult to clean up using existing activated carbon technologies. As a result, cleaning up GenX may be much more difficult and costly to conduct, which may result in more widespread exposures if cleanup cannot be efficiently and effectively done. Lastly, GenX is labeled as an emerging chemical, meaning that there is less studies and research done on this chemical because it is quote unquote a new chemical. This coupled with the lack of public awareness makes it less likely to have regulations and oversight. And unlike PFOA, GenX is often left out in discussions made about regulations and oversight. And kind of all of this is happening while GenX is being openly used and manufactured throughout the nation. And so now I just want to summarize a little bit about the case study that I'll talk about. The impacts can be seen through the Cape Fear River case study. In 2017, researchers found that there were large amounts of GenX pollutants in discharge downstream from the DuPont and Chemours manufacturing site. And so this polluted over 100 miles of the river. And this led to a lot of human and ecological impacts. For example, with human impacts, over 200,000 people were impacted because of contaminated waters. In terms of ecological impacts, we see that with the striped bass population, there were elevated levels of GenX as well as other PFAS compounds. This event eventually led to a lawsuit against the company's misrepresentation and failure to disclose of the illegal chemical dumpings. And this incident did raise some alarm bells for GenX and PFAS for a while, but as of now, there are still no federal drinking water regulations for GenX on the horizon. So now I'm just going to summarize what was discussed in my previous slides. So there are plus and minuses with both chemicals. GenX typically has a lower toxicity based on initial studies that we've seen so far, but not much. And there are still the concerns associated with it being having issues with drinking water treatment, as well as it being an emerging compound. So in summary, based on the points discussed, seeing that both of these chemicals are toxic, they're both mobile and they're both widespread, GenX is simply not a good replacement. In fact, we're simply just trading one hazardous chemical for another. And so in summary, looking forward, I believe that there's 3 points. First, we need more research, not only in GenX, but in PFOA and PFAS in general. We need to tackle the asymmetries in public knowledge and allow people to become more aware of PFAS. Both of these points lead to my last point of needing more regulation and oversight. With more research and public knowledge, and to extent maybe even public outcry, it can potentially result in faster and increased adoption of the much needed regulations. Yeah, so thank you so much. And this concludes my presentation.
Well done, Selena. Is this issue also being discussed internationally?
So yeah, so PFAS is an issue, kind of, PFAS is being used all around the world. But But so far that I know of, I think I know like in Europe, there are a lot of regulations that are starting on tackling PFAS issues, but again, they are very limited. And again, they're also mostly focused on the most widespread and well-known ones, so PFOS and PFOA.
What are some of the alternatives to using PFAS? Like, imagine if there was a regulatory scheme where we were able to better regulate this pollutant. How would that affect manufacturing, or like, what would we do instead, basically?
So yeah, so that's kind of difficult to say, like if we don't use these chemicals completely, but I think what's more needed is like we need more like regulations and like especially on like levels of cleanup and stuff. Like with its use in pots and pans, I guess using not using as much like non-stick and just using like oil on the pans may work. But yeah, that is a difficult question to answer.
So an example of where that is, it's those non-stick pans would be one such example, because they're very popular. Yeah. And also related to Leila's question, if you regulate something, there's always ways to avoid regulation and use a substitute that's even worse.
Mm-hmm.
And you kind of sheds light into that, which
is
sorry, go ahead.
Oh yeah, so yeah, definitely there is like the issue. So like we're seeing this with PFOA. So PFOA is getting a lot more regulation because it's been in kind of in the public eyes a lot more, whereas Gen X is more emerging and so less people know about it. And so there definitely isn't as much regulation and even conversations about it. And so this is kind of the issue with the substitution effect is potentially like, you know, we're substituting one thing and like leaving out another thing that is equally as harming. And so maybe a blanket statement would be good.
Do you also have a sense of the cost increase that would imply a substitution along the lines of Leila's suggestion on what are the alternatives? Is this actually feasible, or would it need to be subsidized initially? Or do you know anything about the trade-off?
So I don't know about the trade-offs with like substituting just PFAS in general, but I know that if we're substituting PFOA for more Gen X, the cleanup cost in water treatment would definitely increase because of the current technologies. Kind of current technologies of cleaning Gen X may not be the same as ones used previously. And so that can definitely increase water treatment costs.
I was meaning production costs so that firms actually switch.
I don't necessarily know what would be like the alternative chemical substitute and if that would change the costs.
All right, well done. Thank you again, Selena. And next we turn the floor to Jackie. And Jackie, I will ask you to please share your screen and start your presentation. Looking forward to it.
Perfect. Hello. And let me, sorry, just a moment. Okay.
I'm outside because all my family is doing something in one of the rooms and I was freezing, so I moved to the sun.
Sorry, I thought I had this ready. Okay, let me share my screen now. Does everyone see it? Okay, perfect. Well, my name is Jackie. Thank you so much, everyone, for coming. I'm an undergraduate in environmental economics and policy, and my faculty advisor is the amazing Sofia. And I'm going to be talking to you today about the impact of COVID-19 outbreaks at US meatpacking plants on consumer demand for increased COVID-19 safety measures for these essential workers. So as we're in the midst of an unprecedented global pandemic, supply chains in nearly every industry broke, seemingly overnight. With the implementation of the Defense Production Act by the Trump administration, meatpacking plants were officially designated as critical infrastructure, labeling these workers as essential according to pandemic guidelines. The purpose of my research was to further explore pandemic consumer behavior and consumer consciousness regarding COVID-19 and large-scale animal agriculture, specifically at US meatpacking plants. And I measure consumer concern via marginal utility and willingness to pay for increased worker protections through consumer stated preferences. Meatpacking plants have long been considered one of the most difficult and dangerous jobs a person could perform. Workers have complained for decades about horrid working conditions and low pay, as well as limited medical treatment for workplace injuries. COVID-19 has compounded the danger already faced by these workers in a number of ways. According to a report from the BBC, meat processing facilities were already breeding grounds for the spread of respiratory illnesses before the COVID-19 pandemic. Some of the most staggering current statistics that I found is that as of April 30th, 2021, at least 58,798 meatpacking workers have tested positive for COVID-19, and at least 291 have died. And a recent UC Davis study found that the presence of a large beef packing facility increases per capita infection rates by 110%, and that large pork and chicken facilities increased transmission rates by 160% and 20% respectively. For my study, I surveyed 860 residents located in the Midwest region of the United States, where the majority of large meatpacking plants are found via random sample survey. I asked various questions about their demographics, COVID-19 opinions, behaviors, and experience, and ultimately their choice of worker protection levels given price and safety attributes for 1 pound of 3 types of fresh meat, sliced bacon, boneless chicken breast, and ground beef. I was interested in exploring whether or not an individual would value safer options for workers if they had been infected with COVID-19, or if a close loved one had been infected, which I made the treatment group, and the control group is everyone else who has not been affected or did not have a loved one infected. So looking at sample demographics, in terms of reported education, there's considerable variation in the respondents' stated education levels, ranging from 4% having no high school diploma to 14% having earned a post-university education. The graphs on the left show the relative density of the highest education gained by birth year and treatment group. We can see a high frequency of those indicating that they have a master's degree or PhD in the treatment group, which makes sense as many have also indicated that they're essential workers. So likely a fair number of them might be doctors and nurses. The sample has a white respondent share of 82.7%, which is actually on average consistent, if not a bit less than the white population makeup of the states that we sampled from, and is actually is higher than the US white percentage of 76.3%. The sample also skewed a bit towards women, taking 65% of the share. And with I'm sorry, with a total of 545 women, 281 men, and the remaining respondents stating non-binary or preferring not to state. The activities and jobs of the respondents were diverse as well, with 9% of the respondents classifying themselves as unemployed as their current status. Preliminary data exploration into the alternative chosen among groups with varying characteristics show those impacted by COVID-19 in a significant way may in fact prefer the safer alternatives. The table in the top left shows us that the treatment group tends to select safer options more often than the control group overall. And according to the data, those who identified as being an essential worker actually tended to choose the safer option less often than those who were able to shelter in place and those who are unemployed. This would make sense despite the high number of essential workers obtaining their obtaining a higher education that we saw in the sample. The majority of essential work tends to be lower paid. Lastly, respondents seem to agree overall that they would like to see the government and corporations do more to protect workers from risks due to COVID-19. As I mentioned, I explored my research via marginal utility as price and safety attributes changed. This structure allows us to estimate average willingness to pay for safer options given the price. Marginal utilities were found through mixed multinomial regressions, and the regression results show the marginal utilities of safety and price attributes controlling for different individual characteristics and treatment status. Not surprisingly, we see a negative disutility on price, which makes sense. And given that consumers don't tend to like higher prices, these results tell us that on average, consumers don't prefer the safer option. However, when controlling for treatment status, we do find that though the safer options are still not preferred overall, utility does increase significantly for the safer options. Utility also increases with income, which makes sense as higher income individuals have the ability to spend more if needed. In terms of willingness to pay for these safer conditions, we see that consumers, or we find that consumers would need to be compensated on average by about 38 cents per pound relative to the average per pound price of $4.53 for the options displayed in order to choose the safer options for workers. Willingness to pay increases with education and income, but decreases with age and among white respondents. Females are also willing to pay more than those who identified as male. Not surprisingly, those consumers who were personally infected or their loved ones were infected seem to be significantly willing to pay a premium for safety compared to those respondents who were not infected with COVID-19. Younger respondents also have a higher willingness to pay for safety than older respondents, which is consistent with other studies that indicate that younger generations base their decisions off of social ethics more often than older individuals. These findings have policy implications in that they suggest a market-based potential to nudge consumers impacted by some disaster and to purchase products that are made safer for the workers. Additional research would need to be conducted to study possible biases introduced by stated preferences in COVID-19. Revealed preference studies are preferable. And yes, that's it. I wanted to say thank you so much to everyone who has helped me along the way, and I appreciate you all being here. Thank you.
Good job, Jackie. Do we have any questions from the audience? All right. Was there any result that surprised you, Jackie, from what you found?
Yes, I guess I was a bit surprised. It makes sense after thinking about it for a bit, but I was a bit surprised that safety was not preferred overall, well, on average overall, and that consumers would need to be compensated to choose the safer option. It does make sense, you know, as far as budget constraints and whatnot goes, I guess, with everything going on with the pandemic, how much it's impacted our lives, and given the community spread, and especially the community spread seen from counties that have these meat packing plants, I guess I expected to see a higher willingness to pay overall, but it does make sense.
Did you distinguish them between unemployed and not employed? I think it was too fast. I didn't quite understand that, because if you control for that, at least you would control for some kind of income shock due to the pandemic.
Yes. I controlled for, let's see.
Always was the regression where you regress the willingness to pay, right? When you have all those controls. Did I see unemployment there?
Yes.
No, the other one.
Oh, this down here? Yes. Yes, so respondents who became unemployed due to the pandemic do have would need to be compensated a bit. But
my question was more, did you run this regression with and without that indicator so that you could see whether that makes sense, that once you control for unemployment, it affects the point estimate of safety on the willingness to pay. But that's just a suggestion. Yeah.
I haven't, but yeah, I will. That's great.
All right, well done, Jackie. And to keep us on time, I will ask Nika to close the show. You can almost say probably the method that Jackie did. Let me just not go into the details. She already explained it. I'm kidding. Go, Nika. Explain your project.
Thanks, Sophia. Do you see my screen, everyone?
Yes, we do. And welcome, Nika. And we're looking forward to listening to your thesis.
Thank you. Hi, everybody. Thanks for this opportunity, and I'm very excited to share my study. When the shelter-in-place order came in California in March last year, we were all forced to change our behavior and also witness no traffic, experience food shortages and panic buying. We all did that. And from meatpacking plant scandals to rollbacks on environmental regulation by former administration, overall, we had active discussions around the environment and climate change and what the new normal could look like. So I started to wonder if I can study whether a pandemic has increased consumers' environmental consciousness and whether it has affected their food choice and shopping behavior. So thanks to Sofia and a classmate helped me turn my curiosity to a research question. And I modeled one of Sofia's studies using discrete choice experiment to derive likelihood of choosing greener product and willingness to pay for them using online survey. This is a method very similar to Jackie's. My scope was California residents over 18 years old, because the main polluter in agriculture is livestock. Surveys were distributed to consumers who eat meat. The idea here is that if we are after the usage of gasoline and to think about greenhouse gas mitigation, we don't go after the Tesla drivers asking a bunch of questions, but focus on consumers who choose to drive SUV or other gasoline cars. So in my case, beef eaters are SUV drivers, and pork and chicken eaters are non-hybrid drivers. So going back to my study, using Qualtrics online survey platform, I filtered out vegetarians and vegans by simply asking, do you eat meat? And followed by demographic questions such as age, race, and income. Then randomly divide participants into control and treatment group, as you can see in this diagram. Treatment group will be shown additional information about pandemic impact on global emission reduction and environmental impact by food chains. After that, both groups were asked the exact same questions. And part one is a series of questions asking about pandemic and environment to build a score. Then finally, they are asked to choose food products out of two choices, where one has lower greenhouse gas emissions in its description, just as shown in the slide. Those questions also have price next to them, like they're choosing food at a grocery store. Oops, sorry. The scores I mentioned have three categories: environmental and pandemic and COVID-19. These are some of the examples, not all, and participants are to answer across 5 levels, from strongly disagree worth 1 point to strongly agree worth 5 points. For example, in COVID-19 question, if someone answered strongly agree to a claim stating wearing mask helps prevent the spread of COVID-19, then she gets 5 points for that question. And each participant has total score for each category, and I can later use to control in a regression as one of the characteristics of the participants. In the same way, I use her demographic information in analysis. These are the additional information shown to the treatment group. Graph on the left are one of the study done by Professor Kamen of UC Berkeley, showing 8% global emission reduction in May last year, which was the lowest since 1900. The one on the right shows the scale of emission footprint per food category. And as you can see, beef and animal products being the highest polluters. And these information are shown in treatment group, but not the control group. So based on choice made in those food choice questions, I built the panel data along demographic and scoring questions. I created this hypothetical meat tax question based on paper from Oxford University asking if the government introduces the carbon tax on food products based on its emission footprint, which do you prefer? And this question shows their preference and can control in a regression along demographic and scoring information to derive the likelihood of a person choosing greener alternatives in the logic model. Now the results. I mapped the average scores per county. Darker colors are the higher scores, meaning they agree to more questions or claims. I was surprised to see the COVID-19 scores, which is in the middle, scores evenly high across counties, because the questions were something like wearing mask or about public health, which have been rather controversial in the US. So I was surprised to see high score were spread evenly. The other two questions were to do with environmental consciousness, and you can see the coastal region have higher average scores. And hypothetical meat tax at the bottom is my favorite, interesting to see. According to my survey, people are willing to pay carbon tax on meat. However, I need to disclose that my sample was rather skewed to white, highly educated and wealthy population, unfortunately due to the budget constraint. So it does not entirely represent the California demographic. But as you can see in the map on the left, participants are clustered around the major city, but this is due to the population density. Here's my logistic regression to see the likelihood of person choosing the greener choice. The first column in the is the most basic logit that only control for scores and treatment. Price coefficient is positive, which is counterintuitive. As we assume price goes up, the likelihood of choosing greener choice should go down. But the second logit in column 2 has product fixed effect, which treat this price bias. The logistic regression 3 has interaction with treatment variable, listed demographic, and used to derive individual willingness to pay for treatment group. Treatment group relative to control group has higher by 40 cents in average willingness to pay for greener choice. And we can also see that as environment and pandemic score goes up, the chance of choosing greener alternatives goes up. And those estimates are statistically significant. And having additional information of consumer demographic characteristic, I found that there is a heterogeneity in willingness to pay, and income and college education are positively correlated with the higher willingness to pay. And I didn't stop here. I also asked them directly these 3 questions. Since the pandemic, do you think you tend to choose food products that are more environmentally friendly? And 63% answered yes. And if you learn that farming pork or chicken is 10 times more environmentally friendly than farming beef, would that change how much beef relative to pork or chicken you would eat? And 73% of people said yes. And I regressed to test likelihood of saying yes to these questions. And people in treatment group has on average though from 6 to 9% higher change to say yes to these questions. And income, environment, and pandemic score have positive correlation. And those estimates are also statistically significant. So overall, pandemic seems to have had some impact with increasing environmental consciousness based on my skewed sample of California residents. As I found in logistic regression models, information about the emission footprint on food products and the pandemic's impacts on global emission level is likely to affect consumers' behavior. So to me, at least, this is an indication of what it takes for consumers' behavior to change and consumers react to those information. And in line with the study presented by Alejandra, I think earlier about the label, it was really cool. Along with that, this study also indicates more potential for massive improvement with simple yet creative solution and policy, such as labeling and raising awareness. As long as we incorporate holistic view of diversity and social cost and the values, we can reform and sustain food production and consumptions. And I would like to thank Sofia for all the education and encouragement and your patience and support, and friends and family and our college for generous funding to make this possible. Thank you.
Great job, Nika. And I was thoroughly impressed by how you kept your focus despite your very large family that lives with you, and you could still focus and do all that you do. You're just like, congratulations on everybody that presented so far. I'm opening the floor to questions for Nika's thesis. And indeed, these last few theses are very, very complementary in how consumers or the marketplace responds to these changes in attributes of products. So even Selena's, it's kind of interesting how everything fits together. So if anyone has also questions for the other panelists, please speak up. We have a couple of minutes. Well, if not, Nick, I have a question. What's next for you?
What's next in terms of this research?
No, what are you doing? Where are you going? You're graduating, right?
Yes, and I'm admitted to the master program at URG at UC Berkeley. So.
Oh my God, you're staying in Berkeley. We can't get rid of you.
You're stuck with me, Sofia.
No. All right. Well, I think our brains are all fried, so we cannot ask any intelligent questions anymore. Everybody has people's names, and we're all emailable. So if people have questions, please reach out to these amazing students that dedicated 2 semesters of their lives to come up with these. And there's more in the afternoon. After you have some lunch and some coffee, come back. We will start sharp at 1 p.m. with Yeeun Moon. And then we'll finish around 3:30ish with Matt. And lots of great research in the middle between those two that I highlighted. And tomorrow again, heavy hard science tomorrow. So if you are along those lines, just also show up. Thank you for Leila for listening and asking questions, and then your careful judging of the presentations and also the feedback. Thank you again. And of course, Anna, thank you for everything you do to keep us in the honors spirit. We couldn't have done it without you. If there's nothing else to say, we will see each other at 1, right?
Yeah, I think we're ready.
All right. Welcome to the afternoon honors. Everybody has eaten, and we are excited to listen to our first presenter, who is Yeeun Moon. You have the floor.
Thank you. I'm going to share my screen now. Can everyone see this fine? Okay, great. Well, I hope everyone had a nice lunch break, and thank you for all the panelists for the cool presentations all morning. Today, I'm going to present my research project on has drug production in Mexico contributed to deforestation? And before I begin, I want to thank my research mentor, Jay Sayer. I wouldn't have made it this far without his advice and support every step of the way. Thank you, Sofia, for sponsoring me and Rausser College of Natural Resources for the research opportunity and funding. So a motivation of this study was to identify the sources of deforestation, which is critical for climate change mitigation and adaptation, and also for biodiversity conservation for apparent negative externalities associated with forest loss. Many studies have been conducted in the past and present to understand the key drivers of deforestation. However, the environmental impacts of illegal crop productions have been poorly understood despite the large number of countries also rich in biodiversity involved in this industry. In the literature, there has been growing observations of the environmental effects of illegal activities such as illegal logging, land grabbing, and cocaine transits. But the understanding of the ecological impacts of illegal drug production has been limited due to challenges of constructing reliable and complete data. Few narrative studies have been conducted to understand the relationship between these variables, and limited studies have attempted to make causal inferences on the issue, but are in different contexts than my studies and with varying conclusions. Mexico is a significant producer of illegal drugs and among the most biologically diverse countries in the world. As you can see on these graphs on the left, majority of opium poppy and marijuana has historically been growing in a line running down the Sierra Madre Occidental from Sonora, Chihuahua, Sinaloa, Durango, and south to Oaxaca and Chiapas. And on the other hand, the majority of forest losses over the last 2 decades have been happening in southeastern part of Mexico, as you can see on the right, in the states of Yucatan, Campeche, Tabasco. But you can see that some forest loss has happened in a major drug producing part of the country as well, as highlighted in this dashed line circle here. According to FAO, the average yearly rate of deforestation has been falling in the last 3 decades from 16 million hectares to 10 million hectares. This recent trend can be attributed to the increased recognition and work in reducing deforestation along with afforestation efforts and the natural expansion of forests in some countries. As shown in this left graph, Mexico's average yearly deforestation rate across the municipalities, both drug producing and non-drug producing, are closely following this global deforestation trend. In this right graph, you can see the trends of illegal drug production of interest. Opium poppy has fairly steady production with some fluctuations over the years, whereas the production of marijuana has declined significantly. There can be several factors attributing to this recent trend, but one of them is that many farmers substituted away from the crop to opium poppy during the second boom in Mexican heroin market, which was around 2001 to 2006, as you can see here. Although the recent trend of deforestation is declining, forest loss is still happening, and the continuation of work in understanding the conventional and new drivers of deforestation is critical. My research objective, therefore, is to contribute in overcoming the difficulties in data collection by improving on aggregated data and dynamic information on deforestation in Mexico. Another research objective of mine is to quantify the relationship between drug production and forest loss in Mexico by asking, has drug production in Mexico contributed to deforestation? The annual forest loss data is obtained from a time series analysis of Landsat images made publicly available by the University of Maryland. It has a moderate spatial resolution of 30 meters and includes 7 files in each 10 by 10 degree tiles. The dynamic information of drug production is then obtained from the Mexican Secretariat of Defense, Sedena by its initials in Spanish. Due to remaining challenges obtaining complete drug production data at the municipal level, opium poppy and marijuana eradications undertaken by Sedena was then used as a proxy for production. To calculate yearly deforestation rate using these data sets, I used tree canopy cover for year 2000 as a base year tree cover across the country for our analysis. It is defined as the canopy closure for all vegetation taller than 5 meters in height and encoded as a fraction per output grid cell in the range of 0 to 1. As you can, 0 to 1. Year of forest loss event file encoded as either 0 or else in value in range 1 to 17 was then used to detect the change from a forest to non-forest state during the study period. With these data sets, we used equation 1 here to recover actual forest cover for municipality I for a given year T, assuming the uniform reduction of the base year coverage until the loss event was detected. Then we calculated the yearly deforestation rate at yearly deforestation rate for municipality I for a given year T using this equation here, equation 2. For drug production, which is measured in hectares, we simply normalized the variables by the size of each municipality and scaled it by 1,000. We used fixed effects for municipality and year in regression to account for time invariant observable variables that may correlate with forest loss. Then we controlled for a conventional driver of forest loss, which is an agricultural activity. Total crop planted measured annually in hectares was then used as a proxy for crop production. The model down here you see here is the main regression for the analysis. Dependent variable is the natural logarithm of the percent change in forest cover for municipality I in year T, and poppy and marijuana variables are the variable of interest in municipality I in year T. Delta is a set of indicator for each year T, and gamma for a set of indicator for each municipality. As you can see on the regression table here, this is an OLS model indicates that 1 hectare increase in opium poppy production leads to 0.04% increase in yearly deforestation rate, whereas 1 hectare increase in marijuana production leads to 0.05% decrease in yearly deforestation rate. Once controlling for municipality, year, and crop production, the estimate of opium production goes down to 0.02%, and the estimate of marijuana production remains the same. This estimation is statistically significant at conventional level. Few limitations on our research project that I want to discuss are the following. Reverse causality. Opium poppy in particular could be driven by forest loss, which in other words, the variable is correlated with the error term. To test this hypothesis, I lagged deforestation variables by 1 and 2 years and regressed them on drug production. There is no statistically significant effect of deforestation on drug production. Then there's measurement error. So we aim to use the local estimate of opium poppy and marijuana production initially, but it took a longer time than expected to construct complete annual validly dated data sets using satellite imagery. Instead, we used eradication data sets as proxy for drug production, and it is most likely be incomplete as activity is intentionally hidden from the government officials. Then there's omitted variable bias. So beyond agriculture activities, population, urbanization, economic development are a few conventional drivers of deforestation we didn't get to include in our regression analysis. These two limitations are in the scope for the improvement of this research project in future. So in conclusion, in this study, I've shown that opium poppy production have a negative effect on the forest extent. In the case study of Mexico, 1 hectare increase in poppy production on average is associated with 0.02% increase in yearly deforestation rate. This suggests that the apparent negative consequences of opium production expands to sustainability by its contribution to the deforestation problem. Marijuana production have a positive effect, on the other hand, on the forest extent. In the case study of Mexico, 1 hectare increase in marijuana production on average is associated with 0.05% decrease in yearly deforestation rate. This positive effect could be explained by how marijuana production is following the same trend as deforestation in the last 2 decades, as you saw in the earlier graphs. And also could be by, according to UNODC, marijuana production requires less direct sunlight as compared to opium poppy. So they tend to be grown in more forested areas. And another conclusion that I made is that eradication may drive deforestation directly by degrading the soil quality and forest remnants, and also pushing farmers out of these traditional hotspots of drug production into the new lands of Mexico. Thank you for listening to my research project.
Well done. We have time for 1 or 2 questions. Jay, without putting you on the spot, do you want to ask a question?
No, I'll leave it open to the field.
What was the most surprising result you found, Yeeun?
Well, opium poppy production was kind of showing the result as expected, but marijuana was showing the opposite direction into deforestation. So that was pretty surprising. And I think, well, I think the most challenging part of the research was definitely constructing data sets using satellite imagery, because I was not well exposed to Python before, and Jay was a huge help in constructing these data sets.
Any other questions? Well, feel free to send a chat to Yeeun to keep us on schedule if you have some questions, and we will switch to Wesley Tam that is going to give his dissertation, his thesis. And you have the floor. Feel free to share your screen, please.
All right, sounds good. Thank you. Yeah, can you all see that? All right. I never know when the screen is being shown or not. But yeah, hi everyone. Good afternoon. My name is Wesley Tam. The title of my thesis is Access to Street Greenery During the COVID-19 Pandemic in Oakland. And so my advisor was Dr. Bill Stewart, and my faculty sponsor was Professor Matthew Potts. And before I began, I just, again, want to really recognize the amazing work that everyone has done so far and just acknowledge everyone that's been able to make it out today. So thank you all so much. So to start, I want to touch upon the benefits of urban street greenery that really framed my research. I define street greenery as the vegetation located along a street, including street trees, shrubs, lawns, gardens, road verges, and other forms of vegetation. And so then below this, I just have a short list of health benefits pertaining to street greenery. So this can include benefits for physical health, such as motivating others to do physical activity and raising pedestrians' perceptions of safety. Benefits for mental health, such as reducing stress and improving one's perception of their physical and mental health, as well as benefits for environmental health, these more abiotic factors, which include air filtration and cooling the urban heat island. So my research really focused on the Slow Streets program, which was announced in April 2020 by the city of Oakland. And these are essentially street corridors closed to car traffic for pedestrians and cyclists who physically distance while outside. Those of you who have visited Oakland may have seen these kind of road closure signs at the bottom left here, along many of the residential streets in the city. And then here on the right is just a map of the distribution of slow streets that I've labeled numbers 1 to 22, as well as splitting them up into 5 different regions of Oakland, which I'll talk about in a bit. But this all leads me to my research question: how is urban street greenery distributed across the slow streets corridors in Oakland? And my sub-questions are: what are the mental, recreational, and environmental benefits of urban greenery for residents, as well as what ideas and strategies can be adopted to better inform future slow streets planning for greenery access in Oakland? So in terms of my methodology, I really looked at 2 types of empirical data. The first type being Google Street View data processed via a computer algorithm called Treepedia, which is from the MIT Sensible Cities Lab. I can share that with anyone who's interested later on. It's really quite fascinating. But essentially the algorithm generates points at least 30 meters apart along all of the slow streets. And then it gathers the metadata at each point for these Google Street View images, and then extracts the images to calculate this green view index, or GVI percentage, based on the number of green pixels in an image. And so this will kind of be the main point that I talk about, this GVI algorithm-based data that I will be talking about in this presentation. And then the second type of data I collected was observational data via field work. And so this involved me walking along all of the slow streets corridors, stopping every 100 feet, and for about 15 to 20 seconds, and observing the environment, recording the data under these 3 indices: the greenery abundance index on a scale of 1 to 5, 5 being the most abundant greenery, greenery condition index on a scale of 1 to 5 as well, with 5 being the best condition or best quality of greenery, and then the sky view index, which is on a scale of 1 to 3, with 3 being the most obscured view of the sky. And so then with this, I obviously established reference images for each criteria to ensure consistency in my methodology. And so then afterwards, I used Cal EnviroScreen 4.0 data, which is from the California Office of Environmental Health Hazard Assessment, that quantifies the pollution burden of each census tract in California. And I overlaid this with the other 2 types of data to determine any correlations between the observed data as well as this calculated environmental burden value. So this is just a map, again, of the slow streets overlaid with the Cal EnviroScreen data. You'll notice that there are some gaps in the Cal EnviroScreen data, but this was just me filtering out the census tracts that the slow streets did not pass through. So it's really only looking at those that the slow streets were contained within. Also to note as well, in the Cal EnviroScreen data, the red and orange represent higher environmental burdens, and this is often associated with lower amounts of greenery. And then the green and yellow represent lower environmental burdens, which are associated with higher amounts of greenery. So as I mentioned before, I divided Oakland into 5 different regions: North Oakland, West Oakland, Downtown Oakland, Fruitvale, and East Oakland. And then you can see here kind of the points that I kind of produced for the GVI values, and where darker points are more greenery and lighter points are less greenery. And so you can see that there's a great variety in GVI values across the slow streets corridors. It's quite difficult to necessarily discern any particular larger scale patterns or trends necessarily. And so this kind of compelled me to take a closer look at each one, more so at a street-based level or a corridor-based level. And so I'll continue to talk about that in my next slide. So in terms of my findings, I first found that Cal EnviroScreen data does not correlate well with this calculated GVI value. If I go back 1 slide, if you look here in West Oakland, you can see that there is quite a high number of darker GVI points, which represent more greenery. And yet these are found in West Oakland with much higher environmental burden. And so again, this kind of compelled me to look at each point or each kind of section at a closer kind of observational level, which was definitely supplemented by my field work. Another finding was that levels of street greenery are most influenced by 3 factors. So the first one is private greenery. this is really homeowners' lawns, gardens, or yards, things that you can just see from the street. Another factor is proximity to green spaces, which includes parks or community gardens that can obviously increase the amount of greenery visible along the street. All right, thank you. And the third factor is topography, which I found quite interesting. For example, if you're at the top of a hill, you can see more greenery down a hill, maybe tops of trees from across kind of further away, versus being at the bottom of a hill where much of the greenery may not necessarily be at eye level. I also found that these patterns or larger distributions of greenery are more closely related to neighborhood norms and private actions. So neighborhood norms really kind of hinge on, for example, one street just may happen to have more greenery than the next. And so this requires more, maybe more knowledge on local history or kind of historical trends, rather than, this is not something you can necessarily just understand from observational data, for example, as well as private actions, which, as I mentioned before, kind of includes residents who maybe have more time or resources to maintain bigger gardens. And so there is kind of this unequal distribution along streets as well. So to conclude, I first concluded that street greenery access must be assessed at block size scales or smaller. As I mentioned before, there could be one street that has high GVI values, the calculated algorithm-based data, but my observations may show that there is much lower levels of greenery. And so this really requires a much more multifaceted approach and different types of data. It's not really enough to just use the algorithm-based data or the observations. It's really important to be able to compare both and to have a better understanding of how greenery is distributed. And then in terms of the policy implications that I suggest, the first one I suggest is more community and resident involvement. And so this can look like initiatives such as subsidies for California native plants, for example, that maybe thrive better and require less maintenance in Oakland's climate or the Bay Area's climate. Or another example may be just disseminating information about greenery and how to grow or maintain it better. Maybe things like culturally specific plants or crops, things like that. And then another policy implication I found was that programs that support neighborhood and private greening initiatives, local residents tending to their yards and gardens, is more effective than having the city manage things like sidewalk trees or public parks, since these public spaces may not necessarily be representative of the experiences that people have when walking along slow streets. Not every slow street has parks or sidewalk trees, and so it becomes much more important to look at private actions and private greenery, because those are really found everywhere. There's really much more common along the slow streets, since these are residential streets. So yeah, that's the end of my presentation. Thank you all so much once again, and I'd be happy to take any questions if anybody has any.
Yeah, we have time for questions, so feel free to unmute and ask or put them in the chat.
I have a quick question for Wesley. Thank you for that really nice presentation. I was wondering if you could tell us a little bit more about the Cal EnviroScreen data and what exactly is meant by environmental burden. Like, do you know how that's calculated and what that means exactly?
Yeah, I can go back to the slide that has some of this data. Essentially, it's quantified by 4 different factors. I believe one of them, I can't remember exactly the 4 indicators because it's kind of been a while since I really looked into it, but I know that part of it is obviously socioeconomic factors, so like median household income, racial dynamics, and then the other 3 include proximity to things like landfills and waste sites, proximity to highways, train lines, things like that. The Port of Oakland, obviously, in West Oakland is a factor in that as well. But essentially the score is quantified by obviously the office that creates it to kind of come up with a I believe ranking that kind of aggregates all these 4 types of data into one score.
Yeah, so it sounds like definitely something that would capture things like air pollution, given what you were saying about proximity to freeways and that sort of thing, right? Thank you.
Yeah, definitely.
Thank you again, Wesley. And Next up is Renee Sirota, and I will ask, please, Renee, to share your screen, and we're looking forward to your presentation.
All right, let me share my screen. Can everybody see? Good? All right, awesome. Well, thank you so much for everyone who's here and also my friends and family, everyone who's tuning in as a guest. My thesis presentation today will be on low-income solar in California, an analysis of some of the incentive programs that have been developed in California to help rectify the problem of low-income people not really having access to solar. So thank you so much to Professor Sofia for being my advisor for this thesis, and I have some more thank yous at the end. So first I want to have some motivating statistics. So this first one is 8%, which is the percentage of income that low-income households tend to spend on energy across the country. And this is about 2 to 3 times the amount that other income levels tend to spend on energy, meaning that low-income people tend to have a higher energy burden, which really means that they have a huge opportunity to benefit from alternative energy sources such as solar. However, the next statistic shows that only 15% of the huge growth in solar adoption across the US has gone to these low-income houses, meaning that about 85% of solar development goes to middle or higher income households. And then this is a really disproportionate effect that low-income people are not able to access the kind of solar benefits that would really help them lift their energy burden. And then the next statistic is that 42% of US solar potential is actually located in these low and middle income areas that really struggle to achieve the kind of solar independence that we really need in this country for everyone to be able to benefit from solar, and also for us to move away from fossil fuels and meet our reduction targets. We actually really need to incorporate low income solar into the equation. So it's really a problem that every single state in the country is trying to address, and California has chosen to address it through a number of these programs that I'll be talking about today. So 3 programs that I'm talking about are all part of the California Solar Initiative, which is like the wider umbrella under which these 3 programs exist. So the first is called SASH, MASH, DAC-SASH. Those are like the fun acronyms. But the first one is the Single Family Affordable Solar Homes Program, and that's meant for low-income homeowners. The second is the Multifamily Affordable Solar Housing Program, which is meant for multifamily buildings like apartment buildings where people don't own their individual buildings. And then DAC-SASH is very similar to the Single Family Affordable Solar Homes Program, but it's specifically targeted at disadvantaged communities, which I'll mention in a second. This is actually really related to what Wesley was just talking about the Cal EnviroScreen scoring framework, but this is specifically having to do with those communities that score in the top 25% of the overall metrics, meaning that those are the communities that are considered the most disadvantaged across the state, having to do with environmental impacts, economic impacts, socioeconomic additional metrics, some of the things that Wesley was mentioning, pollution. These are also communities that tend to be, greatly low income, but also have a large percentage of minority populations. And so those are considered to be those that are most disadvantaged in terms of the environment and economics. And so those are the communities I chose to focus on for my thesis. So that when I reference disadvantaged communities, this is what I'm referring to. So basically what I did is I wanted to see whether these programs helped increase the likelihood that a solar project was in a disadvantaged community, because these are areas that will really struggle to be able to enjoy the benefits of solar without some of these incentive programs. So I wanted to see whether these programs were actually helping these areas be able to go solar. So what I did is a linear probability model in R. So I'm looking at the presence of whether these programs affect the likelihood that a project is in a disadvantaged area. And I'll talk a little bit more about that in a second. So first I compared the programs against each other to see if there were ones that were doing particularly better than others. And then I compared the programs to a control data set of projects in the same time period that were performed under different programs that were not targeted at low-income people or were just done independently by people for their own households to see whether there was a significant impact in the amount of projects being done in disadvantaged areas with the programs or without the programs to see if there was a difference. So the 3 main utility areas that I covered are PG&E, Southern California Edison, and San Diego Gas and Electric, and I consider these both separately and all together. So the first summary table I want to introduce is this one, and these check marks show when this variable was highly significant. The positive or negative show whether it was a positive effect or a negative effect. So whether it increased the probability that a project was in a disadvantaged area or decreased it. So here you can see, comparing the projects to each other,
that the DAC/SASH program definitely had a larger impact. And the presence of a DAC/SASH program involved in a solar project was associated with a 32% greater likelihood that that project was in a disadvantaged area. So that really shows a strong association between that program and projects being in disadvantaged areas, which makes sense because it's the focus of the program, but it's good to see that they seem to be achieving their goals. And the second little table here shows some of the other variables that I considered and how many times those variables were significant. So how many times in all the regressions that I did, those variables were significant. So here you can see one of the ones that was actually almost always significant was projects. And this counted the number of projects in a given zip code. So basically what this means that an increase of one project was associated with a 0.12% greater likelihood that a project in that zip code was in a disadvantaged area. So that's kind of a complicated way of thinking about it, but basically it means that there was a good amount of projects being developed in disadvantaged areas, which is definitely a win overall, given that this is the treatment data. So this is the data from all the programs all together. And here we have the treatment data compared with the control data. So this is the treatment data, all the projects done under all the programs, and then the data done of a bunch of projects done at the same time, not as part of the programs. So here you can see again, the treatment variable is highly significant and positive, and this effect ranges from about 16% to about 53%, meaning that the presence of any one of these programs is really strongly correlated with a higher probability that the project will be in a disadvantaged area. So that's a really good positive outcome for all of these programs. One of the things you can see is that the DAC/SASH program, SASH1 and SASH2, which are two versions of the same program, are pretty different from MASH1 and MASH2, which I'll touch on in a second. Here again, I addressed some other variables, including third-party cost, or third-party ownership and total cost. These two were also very highly significant, and third-party cost was actually also usually significant for the previous data set, and this is a really significant result for something that I'll mention in a minute. Here we have some graphs that help illustrate what I was talking about before. These are made on JTools, which is a really great plugin for R that I'd very much recommend. But what this shows, this is one of the regressions for DAC/SASH, where it shows the DAC/SASH variable and then as well as all the other variables that were included in the regression, and shows the relative importance of those coefficients in determining the impact of the DAC/SASH variable, and then also variables like third-party owned and projects, which were tended to be significant for most of the regressions. And then the second graph here we have showing the impact of the treatment variable, third-party owned, and total system cost. As you can see, third-party owned and total system costs are both significant, but you can see that the coefficient for third-party is much higher than total system cost, so that's something that has an overall greater impact. Here you can see also I made some GIS maps as part of my project as well, and here you see the overlay of the disadvantaged census tracts with the projects per zip code. So it's not a one-to-one association, but you can definitely see here that a lot of areas with a lot of projects are also very close to disadvantaged census tracts, which is definitely a strong sign of success for the program. It's not a one-to-one association, of course, but then there's some of the things that cause that to be true include the issues with the MASH1 and MASH2 programs, which again, I'll talk about in a minute. But this really helps to see kind of visual representation that there are a lot of projects occurring in these disadvantaged areas. So some of the key results, as I mentioned, the DAC/SASH program has, compared to other programs, has a really high association with projects being completed in disadvantaged areas, which is super positive. Third-party ownership is often a significant variable, and this is really key because SASH2 and MASH2, which are the newer versions of those two programs, introduced the option for third-party ownership. So it really looks like that introduction has been successful in creating more projects in disadvantaged areas comparatively to SASH1 and MASH1, where third-party ownership was not allowed. This projects variable, as I mentioned before, is really helpful because it shows that a lot of projects are happening in these disadvantaged areas. And then the treatment variable is probably the most significant of all because it shows that these programs are really impacting the amount of projects being completed in disadvantaged areas, and that without the treatment variable, meaning without the programs, there really aren't as many projects happening in these areas that really need them. As far as MASH1 and MASH2 not necessarily being as successful, this is a really serious issue because in disadvantaged low-income areas, lots of people tend to not own their homes. So there really needs to be a new way to try to get more multifamily housing projects that have solar on them in disadvantaged areas. Thankfully, there's a new program that I didn't actually address that's called SOMA, which is Solar on Multifamily Affordable Homes. And that's actually going to try to address some of the issues that MASH had in targeting disadvantaged communities. So hopefully that's more successful in the future. And then as far as SASH1 and SASH2, similarly to DAC/SASH, very strong association with this presence in disadvantaged communities, meaning that this program, even though it wasn't targeted at disadvantaged communities, is still really associated with the likelihood of a project being in one, meaning that it's really having a measurable impact on the amount of projects being completed in low-income areas. So that was my project. Thank you to Professor Sophia again and Professors Fowley and Anthoff for inspiring me to do an energy-focused project and kind of pushing me into the energy-focused area of my major. Thank you to my solar club, Revolve Solar at Berkeley, and my friends and family and everyone who came out to watch this. Also for this presentation, a special thanks to the people who created the JTools package and helped explain it to me, and also Slides Carnival for my fun slides.
Well done, Renee. And the graphics were indeed spectacular. You made me want to learn it, that package. And maybe you can explain it to me. We have time for some questions. If you want to unmute and ask some questions, that would be great.
I'll ask a really quick question. Thank you, Renee, for your presentation. It was really nice. I'm wondering, and I apologize if I missed this in the beginning of your talk, but is there evidence that the adoption of solar through some of these programs, like actually reduces the energy burden for the low-income households?
Yes, so these all are incentive programs that provide upfront incentives, meaning that low-income people don't have to pay you have to pay very little for the installation of solar programs, and so it really depends on people's conditions, like where they live, like how much sunlight they're getting. But in most places in California, programs like this will lead to an overall lowering of costs for people, meaning that the energy burden will go down. That's not necessarily true for everywhere in the country, but in California specifically, like the amount of solar that we have, the amount of sunshine that we have in most areas of the state does mean that if they're able to offset some of the costs of the initial installation, it will lower their energy burden and their energy costs like in the long term.
Great, thank you so much.
I have one follow-up question. I asked this some presenters and I want to ask you. What's next, Renee? Do you know what you're going to do finally?
Oh, yes. For myself, like in my life or with this research?
No, post-graduation. I'm curious.
Oh, yes. Yes, I actually, so I will be attending Berkeley Law in the fall with a focus on energy law. And I will be, I'm interested in working in renewable energy law in the future and, working on the legal side of how to implement programs like this and how companies can work together with state and local and federal governments in order to make things like this a reality for more of the country.
Very good. We're happy to keep you here.
Thank you.
All right. Any other questions? Well, we are running a little ahead of schedule, so I hope Eva's fun group is in the house. Eva Manjares, you have the floor, and if you want to share your screen, we would be delighted to listen to your honors thesis. And maybe, Renee, I think you need to unshare, maybe.
Great, thank you.
Thank you.
Get this started, sorry.
Looks good. You just presentation mode and you're all set.
Great. Okay, everyone, hi. Wow, we've had so many nice presentations, so congratulations, everyone. I'm Ava. I'm going to be going over the environment and neoliberalism. My work really focused on a historical perspective of Mexico for climate change mitigation. My mentor was PhD candidate Nain Martinez, and my advisor is Dr. Kate O'Neill, both in the SPEM department. The aim of my paper was really to examine the link between the environment and neoliberalism by providing a historical analysis of Mexico to highlight the obstacles of a developing country when addressing climate change policy. My summary of findings are pretty much a lot of what I expected, which really just implies that a financial capacity is not the only obstacle that developing countries face when addressing climate justice. There's a lot of other things in the backdrop of the political rhetoric that are a really big burden to these countries, and understanding was something that was really interesting to me as someone who's interested in international development policy. And I also found that when it comes to climate change, a lot of these solutions and root causes were even more difficult to untangle, right? Was it the chicken or the egg? What came first? What is going to really be the solution to some of these social inequalities that are being exacerbated by climate change impacts? And really this really close examination of Mexico historical events really emphasize how complicated this conversation is. And this is largely due to the legacy of colonialism in a lot of developing countries in the global South. There's a fractured relationship with global powers that I anticipate is also probably problematic, not only for economic development, but when we talk about climate change adaptation and technological transfer of certain projects. And in general, there's also this complicated relationship with neoliberalism in the global South. That is a very important thing to address when thinking about mitigation strategies. What I did really is actually quite complicated. It's complicated to summarize in a 10-minute video, but I really looked at the history of Mexico and that's really, I went back 500 years to the pre-colonial era. And I used that to understand these political rhetorics that are standing in the way of a lot of progressive climate change policy. And in some, really, it really just kind of showed how again, complicated this is. In the pre-colonial era, which is characterized by the era of the Aztec Empire, what that era really showed me was the indigenous expertise surrounding sustainability that I think is very important for acknowledging in the climate justice area. The colonial era was really an exploration of the social structures, especially when it comes to racial dynamics in Mexico, but it's also applicable in a lot of global South countries. Eventually, we get to the independence of Mexico, which happened in 1821, and this era really denoted what the national sentiment toward foreign investment is. And it's also an era that explains a lot about the Mexican nationalism and anti-neoliberal sentiment that is prominent in the dominant discourse surrounding climate change policy right now. Eventually, we get to the Mexican Revolution, and that kind of is really encompassing kind of current political struggles. And neoliberalism here is kind of this tug of war between administrations and trying to figure out the next step of the country. I think it's all very entangled, but every single piece of these eras are kind of really create this like all encompassing picture that I think is very important. And all, what does that have to do with climate change mitigation efforts? And really that's kind of what I'm tying it together with, right? In 2015, we have the Paris Agreement, and really we have national determined contributions targeting greenhouse gas emissions to limit global warming to at least 2 degrees Celsius. We have some principles of climate justice. We have talks about technology transfer and climate finance. And really that's when Mexico kind of began to be in my peripheral. Because from COP21, Paris Agreement, we see a lot of headlines that were looking at Mexico for their very ambitious targets to limit greenhouse gas emissions. So environmentalists, we were very excited. We thought, well, Mexico could be a climate leader. It could set the stage for Latin American development. And if we support them enough in the right way, maybe it could be just the piece to the puzzle so that we can start seeing these changes in other developing countries. But what was really interesting actually about Mexico and that climate change mitigation era was that the progressive targets from the Paris Agreement were done by an administration who I guess could be characterized by as a centrist, right-leaning administration. And to their detriment, this actually led for very low approval ratings for this president that proposed these targets. And he left office with very bad ratings. And it also created a vacuum for a socialist president, which is what Mexico has now. And this socialist leftist president is not necessarily against climate change, but really is focusing on economic development. And that really intrigued me because why? Why is what I guess we could consider a conservative administration having progressive climate change policy when a administration that's more of a left-winging socialist president is backdropping in a lot of the global agreements. And that really kind of led me to think about neoliberalism. And is this the key and the main objective to tackle when dealing with climate change in countries like Mexico? Because neoliberalism for a lot of people could be seen as a way to greenwash some of these initiatives that are supposedly combating climate change. And I think that's a lot of the resistance that Mexico sees with climate change mitigation. And what really worries me about all of that is something that's very important to me, which is climate justice. And it's also very interesting to me to see how this plays out during COVID. Because what we're going to see is less ambitious NDCs, less ambitious goals in the face of, well, a lot of turmoil in a lot of countries. And this here on the right is a greenhouse gas emission. It is from 2019, so it would be interesting to see how this is going to change now considering 2020 and even 2021 with COVID in the background, but for me, it's all really connected, and this kind of serves as a framework of how we can develop strategies to support these countries that are disproportionately affected by climate change. And so the historical perspective for me really kind of ties together with everything that has to do with climate mitigation, right? The global cooperation, the climate justice initiatives, research even, right? When we talk about, having projects that are not only have good intent, but will be supported by local and regional organizations. When we talk about economic development and of course, social equity. Climate mitigation in some is just so really complex, and the root issues really need to be centered in climate justice initiatives. When it comes to global cooperation, and technological transfers, I think social economic understanding of the region could really be the difference between successful projects and a failure. So it's really just important to interrogate kind of the perspective that we're looking at when we are creating global and international cooperation methods. And it's something that I really hope to push anytime that I have, even if you come from a background of a more science-heavy field, because that's often the burden of marginalized communities, is to really understand the status quo of who is poor and why they're poor to begin with. And so I know it's a lot, and if there's any clarification that's needed, please don't hesitate to ask, because I know that I try to squeeze so much in 10 minutes. Maybe not everything was very clear.
Thank you very much, Eva, for your presentation. And we have time for some questions, so feel free to either chat or unmute and ask a question directly. I'm curious if you were to do this analysis in another country, whether you would find something similar, and is Mexico kind of an outlier, representative for someone that doesn't know a lot than you do? What would you answer that?
Yeah, actually, that's kind of one of my next steps. I think this general topic is something that's very interesting to me. What I anticipate being different are essentially some of the details, probably what certain historical eras would tell me about the political rhetoric, I think would be probably different. But there is a lot of similarities, I think, especially in Latin America, with the legacy of colonialism and the history of exploitation of labor and biodiversity. There's just so many similarities that I think it would definitely be an interesting add-on to this topic.
There is a comment and a question. So the comment first from Wesley: really appreciate the focus on indigenous knowledge from the Aztec Empire too. And then Matt asks, what specific resource industries do you believe Mexico would should nationalize, and how imagined they should be run? And fantastic presentation, also a comment.
Yeah, nationalization is actually something that's super interesting to me. Right now they have the oil industry, for the most part, is nationalized. Recently, there was, I think, 17% that was privatized, but the energy sector is nationalized. The railroad system was nationalized up until 1990. But there's always that's kind of like the big argument within administrations: is privatization going to boost the economy and in turn allow us to have more environmental protections, or is this the issue that we need to get rid of, right? So I think that's probably up to people's interpretation. I think if you're an economist, you might disagree or agree that it's just mismanagement, but I think that just differs with fields of study.
Thank you for your answer. So welcome to those that showed up. If you have my name when you do show up, please change your name. I apologize for my wrong Zoom link that I sent. Welcome, Professor Chapela. Selena Melgoza is next, and I hope I didn't completely butcher your name. And if I did, please tell me how to pronounce it well. You try doing my last name and then we're even. So Selena, you have the floor, and we're looking forward to your presentation.
Great. Okay, I hope you all can see that. And so, hello, everybody. I am Selena. I am a senior now studying society environment with a minor in public policy. And today I'll be presenting to you my CNR honors thesis on the topic of analyzing intersectional policy through Milwaukee's declaration of racism as a public health crisis. So first off, putting Milwaukee's declaration into context. In 2018, the Wisconsin Public Health Association had declared racism as a public health crisis. And following their public health declaration, they established a racial equity working group that was set to meet the demands the resolution called for. A year later, the county of Milwaukee was actually the first county in the United States to declare racism as a public health crisis. And soon after, in April of 2019, the city of Milwaukee adopted the declaration as well. And this makes Milwaukee's city, county, and public health association to be the first ones to adopt this declaration, setting racism as a public health crisis. So in my research, I was really interested in analyzing this new framework of racism as a public health crisis, and using Milwaukee as a primary example, and using the pandemic as an example, as well as the American Medical Association's similar declaration as a subcase. I went through a literature review of environmental justice theory and analyzed Milwaukee's declaration along with AMA's declaration. And to do so, I conducted several interviews with people who were directly involved in creating the declarations. And this included county members, council members, and department members, including delegates of AMA as well. And secondly, I interviewed community members and organizations in Milwaukee specifically who were involved in environmental or anti-racism organization work in Milwaukee. And so in the interviews, here's just a sample of my interview questions for the creators of the declarations. And essentially all the interviews, the goal was to understand what was the impetus for this declaration. Was it internal or internally driven, or were community members involved? How expansive was this declaration? Was it able to tackle issues that were social, environmental, health-wise, or sustainable-wise? Additionally, we asked what were the impacts of this declaration? What happened after this declaration was made? Did it aid in the pandemic? And those kinds of things. In my second set of interviews with the community members, I asked them if they knew about the declaration. Were they involved in the creation of the declaration? If they saw this declaration being impactful? And asked them what their perspective was. Was this symbolic or actionable? And through these interviews, I gained a huge context for Milwaukee's social challenges, and that really made me understand further why Milwaukee was making such a declaration. And just to put this into context, to this day, according to 2019 statistics, a white person lives on average nearly 14 years longer than a Black person in Milwaukee. And in interviews, council members and community members have described the disparities as such. And on the slide, I have a few quotes for you. Marciela Nicholson, who I interviewed, is a council member, and she pointed to the mass incarceration that's happening in Wisconsin. 50% of Black men who come from the zip code that I grew up in have been touched by the incarceration system at the end of their lives at some point. And as a whole, Milwaukee incarcerates more folks in the state of Wisconsin than the entire nation. And if you know, the United States itself is the largest global incarcerator. And to put that perspective in our nation, Wisconsin is driving a large amount of those incarceration rates. And Reggie Jackson, who was actually hired as a part of this declaration to be the equity advisor and trainer for the employees, described Milwaukee as Milwaukee is considered the worst place to live in the United States if you're Black. It's the worst place to raise a Black family. And I point this out because it's just to put in context the kind of social challenges that Milwaukee is experiencing and what is driving this, this declaration. And now, looking at the impact of the declaration, Milwaukee was one of the very first cities and counties to recognize the racial disparities between who is dying from the COVID-19 pandemic. And this was even earlier than the United States government had released and earlier than the CDC. And that just goes to point that having this kind of racial conscious policies allow them to address racial issues faster. Additionally, the city of Milwaukee established an ordinance which was to institutionalize racial equity. And in this, they trained over 4,000 employees on racial equity. And this promoted the acknowledgement of racism as an issue because that was something that has not been normalized in Wisconsin, although there were many lived experiences to show. They established Public Health Week as a yearly form of education to happen within the community. They created an ambassadors program as well as a racial equity budget tool and audit committee. And these are very important because there are another ways other than the declaration to institutionalize racial equity practices within the city. And now looking at this, them being the first, there are now 146 jurisdictions and 28 states that have made this declaration. So we can see that this bottom-up approach of starting policy and local politics actually has a large effect and is spanning over the nation. And so with this, there are a number of challenges in Milwaukee's declaration. And after these interviews, I learned about these things. And starting from the top, I learned a lot about the importance of having community members and organizations being on the table and developing this language for declarations. So in specific for the county, there was minimal community involvement. It was more of county representatives coming together and then making this declaration. In contrast, the city of Milwaukee was more in community involved. And this is important because we can see that the city of Milwaukee's declaration and their following actions were a lot more progressive than the county because of that community involvement. Additionally, the county declaration is voluntary. So cities, a part of Milwaukee and Wisconsin, do not necessarily have to adopt this declaration. And that means that areas that may need it the most, including the suburbs which are wealthier and wider, may not have the same urgency to adopt these kinds of resolutions, even though they may be needed in those areas the most. Additionally, there was a lot of challenges with using the term racism and race. These conversations were not normalized, and people from different backgrounds were coming to these equity trainings with Reggie Jackson and not being comfortable because they were saying, I am not a racist. And that is in fact a part of the larger problem of normalizing these discussions. And I think it's very important to have these conversations normalized because it allows for better policy. Another large challenge that we've seen is more structural, and that is in regards to the county and the city budget. So the city of Milwaukee has actually been struggling financially as their shared revenue has been declining between 30 to 40 million, which means every year that they're actually already in a deficit. And that makes advocating for budgets to be into racial equity are harder to do so. And then with the county, it's a little harder too because their budget is still largely tied to the police budget. So community members are asking if this racial equity is serious if they're not actually readdressing their budget to allocate for these services. And I'll touch briefly on the American Medical's resolution. And this came from delegates who are actually current students of color and a part of AMA. And they decided to make this resolution to hold AMA accountable. And the reason why I wanted to look at this is because AMA is actually the largest physician organization, and they passed this resolution in November of 2020. And with all of this, I know it's a lot, I'm talking very fast, there's so much to say. The key findings are that having representative leadership and leadership of color promotes race-conscious policies. In the city of Milwaukee, all 5 major council positions were held by Black people, and 4 out of 5 of them were Black women. And that directly translates to the amount of progressive policies in addressing these racial issues that are present in Milwaukee. Additionally, declarations are a strong starting point to promoting accountability and promoting racial equity. There are still structural challenges. However, we've seen with the city of Milwaukee, ordinance in addition to the declaration have a lot more teeth and are a lot more proactive. Additionally, the voluntary adoption allows for easier passage at the county level, but it does have less teeth in terms of actually promoting racial equity. By looking at budgets, you can see that there's a large scope for feasible systemic change. And lastly, through the interviews, we've seen that the perceptions of the declarations are kind of split. Community members are those who are not involved in the process, seeing these declarations as more performative than actionable, whereas the council members see these as good wins and a step in the right direction for actions. For those who may be looking to do this in the future, some recommendations I wanted to put forward was working to build alliances and partnerships with other organizations that are already doing this work. It's important to work with them and use their language and be responsive to their demands in promoting local or countywide policies. Additionally, at the county level, consider requiring entities to adopt the declaration as a requirement rather than voluntary to extend its reach. And additionally, in the last one, to ensure the declarations are action-oriented rather than symbolic. And I think this goes along with the lines of ensuring that there are ordinances or policies that follow declarations to ensure that racial equity is being pushed forward. And then lastly, in my last few seconds, some concluding statements is that I think that this declaration process has a great opportunity to spur systematic change, and especially in the time that we're all experiencing now. It has the ability to influence environmental justice policies by using a public health framework to push those policies forward. And the biggest thing here is defining a problem impacts how you respond to a problem, and calling racism out for what it is what is needed to make solutions. So thank you all.
Thank you so much, Shalina. Don't unmute because I hope we have some questions from the audience and the attendees. I'm curious, how did you do the interviews? Was it online or did you send surveys? How did you sample who you're interviewing?
Yeah, I actually was able to meet with everybody via Zoom. And that was actually a great experience. I was actually hoping to be in Milwaukee and do this research. But it was fortunate enough that we have Zoom set up and I was able to meet with everyone via Zoom. And then just was able to record and transcribe and then collect all of this data.
Well, we are very thankful you did this and shedding light into ways to highlight it through a lens that is being implemented, it's always very helpful to start the conversations and the solutions, as you say. Well done, Selena. And we are gonna go now to, if Mari's ready, to Mari Wilson. And I hope I said your name correctly. If I didn't, please correct me.
You did, yeah.
And Mari, you have the floor, and then we will stop for a break and come back. But Mari, you have plenty of time because we're running ahead of schedule, so no need to rush, and just enjoy.
Oh, great. Okay, thank you. All right, well, my name is Mari Wilson. I'm in the PMB department, and my wonderful advisor, Claudia Carr, is in the SWIM department. And my honors thesis project was on river basin development, and I'm using the Turkwell Dam in northwestern Kenya as a case study. So this is like a brief introduction to my project. The problems with huge dam projects are fairly well known and fairly well researched. And so knowing these problems can kind of drive developers to opt for medium or smaller scale dams, kind of with the idea that this will be able to circumvent some of those issues that arise. But this kind of led me to ask the question of, are these medium scale dams in particular actually better than large scale dams? Are they a viable alternative? And as it turns out, there's really relatively little research that surveys medium scale dams as a whole to answer this question. So I began in Kenya here. You can see the picture of the Turkwell River on the left, and then there's a riverine forest around it. So I began here in Kenya with this river to see if the rhetoric around medium dams kind of matches the reality of this project, and then seeing if this can be applied to medium scale dam development as a whole. And so moving forward, I'm going to be giving some context on this dam project and then systematically going through its effects. And in the end, you will see that it is quite emblematic of development projects as a whole. So this here is a satellite image of the river. And up on the left, this is just situating you. Here's Kenya. This is the region where we're looking in kind of in the red. Right here is the border between Kenya and Uganda. And the river rises in Mount Elgon, which is right on the border, and it flows northeast. So here it flows, this is actually the upstream portion through West Pokot County. I'm sorry about the noise. And this here is the Turkwell Gorge, and the dam is sited right here. And then it continues flowing northeast through Turkana County, where the Turkana live, in this very arid region, and it lets out into Lake Turkana. And just to give you a bit of like a size reference, Lake Turkana is huge. It's probably, I think it's the largest permanent desert lake in the world. So it does make the Turkwell Gorge Dam Reservoir here look very small, but it's really not an insubstantial amount of water. And I will show you that very soon. But first, we're just going to go over the livelihoods of the people that I just mentioned, which are the Turkana and the Pokot. So the Turkana are the downstream pastoralists living in that extremely arid region that I just mentioned. And the Pokot are the upstream pastoralists. And as you saw, it was a little bit greener there. So they practice some kind of like agro-pastoralism, but it's really not a huge amount of their livelihood. So the critical context I want you to have before I jump into the dam is how these people have lived and how vulnerability has been generated throughout the 1900s by the British colonial regime in Kenya. And the three major examples of how this arose is through, firstly, the restriction of movements. And movement is an extremely important part of a pastoralist livelihood. So by restricting these movements, by not allowing them near the river and the forest, they kind of undermined their livelihood and coping strategies. Others were taxation and also confiscation of animals, which is just kind of directly attacking their resource base. So in the 60s, 70s, and the 80s, there were these droughts, and many people were pushed into famine camps. But the unique thing, as I said here, is that this is a very drought-prone region, and they have historically been able to be resilient to it. But paired with this generation of vulnerability, it became a very serious issue. So the government solution was by providing food relief and then kind of discouraging pastoralism, trying to promote irrigation and settle pastoralists. So keeping that in mind, I'm going to jump into the Turkwell Gorge Multipurpose Hydroelectric Dam Project. This is an image of it on the left. This here is the thin arch concrete dam. It's about 153 meters tall. That's like a little over 400 feet. This here is the reservoir. It's about 6,500 hectares. It's pretty large. It was built from 1986 to 1991. And so the history behind it is that the Kenyan government, most notably the then Minister of Energy, Nicholas Biwott, were involved with the inception of this idea along with France. And so the two main purposes of this dam is power generation, because it's a hydroelectric dam, and secondly, irrigation schemes. Although in reality, I think these irrigation schemes were more about social control, because Biwott himself said that he hoped that the dam would help the donkey, which is the pastoralist, catch up with the horse, which are people in kind of more developed regions of the country. And the second thing is that NORAD undertook the first feasibility study of this dam, And what was really interesting and kind of unique actually to this project is that they advised against it. They said it was non-viable. It was located on 2 fault lines. It would rapidly silt up and it would destroy the downstream forest. But the Kenyan government, with BWAT pushing for it, moved forward with this project anyway. In the end, this dam cost 3 times what it should have, about $300 million US dollars. And it was contracted to Segrea as the engineer and SPI as the consultant. These are both French firms. So BWAT then flew to Paris and signed this contract in secret in a hotel room, although he denies it. And much of this information was given to us because this EU delegate named Ashim Kratz leaked a memorandum to the London Times that stated that the Turkwell Dam was shockingly overpriced. It was financed on commercial loans despite offers for more concessionary loans. And he said, and I will just quote, that the French deal was only made possible because of handsome profits for French executives involved and handsome kickbacks for their Kenyan counterparts. And this includes millions of dollars to BWAT and the then president, Daniel Arap Moi. So despite their best efforts, this news got out. And in Kenya, this project is known as a stinking scandal and the whitest of white elephants, which is a phrase for scandalous and corrupt. So it's really safe to say that from the inception of this project, there was only interest in power generation and monetary gain, and there was really very little thought given to the people living near the actual dam site. So now I'm just going to overview the outcomes of this Turkwell Gorge project and see, did it actually even live up to its purported goals? The irrigation schemes have mostly failed. They lack water, there are pest outbreaks, and they have really not been able to provide food security in the region whatsoever. Secondly, when it comes to electrification, within the first few years of construction, one of the turbines actually broke. So for years, it was only operating at most half capacity. Even so, it has almost never been full, and many people say it was very well known that there just was not enough water on this river to make it viable to actually generate the 106 megawatts that they wanted to connect to the national grid. Of course, none of this power has ever gone to the locals, although there's money being poured into a rural electrification program. They have not seen even a single megawatt really in the region. But you know, the other thing is that a lot of locals don't necessarily need power, they need land and a resource base. And even this is being undermined because we're seeing conflict rising in the area with rising cattle rustling, serious environmental degradation as was predicted, rivering forest disruption, decreased groundwater because there's less flooding to recharge, invasive species, and sedimentation and siltation. Also, their welfare is directly being undermined, as again has been seen in many dam projects and irrigation schemes. There's a huge increase in waterborne diseases and also crocodile attacks. There's been a loss of tradition, social networks and ties. So once a lot of pastoralists do try to settle here, they've lost their social ties and networks. So it makes it much harder to ever transition back into other lifestyles. There's also a current overflow crisis that I will touch on in a moment. And there's also ongoing oil speculation that may become another abstraction point for the water in this dam. So this is a very current issue. It might be a bit confusing because I did say that the dam almost never reached its full capacity, but in recent years it has gotten fuller. This is partially due to geological factors and because siltation is increasing, the overall capacity of the dam has decreased. And this is also paired with heavy rains. So a 2019 dam report showed, and this is across dams of the country, showed that the dams have almost never been inspected or maintained. The county themselves publicly stated that they cannot find the emergency contingency plan that should have been created when this dam was constructed, if it had ever been created at all. There's no alarm system in the region, so there's really no way to alert people. And also 300,000 or more people are at risk of dying when this dam overflows. And you can see, I mean, the perspective might be hard, but this is only a few meters before it will overtop. So this is a very serious crisis that arose in 2020 and it's ongoing. So now I'm just going to go over my concluding remarks. Firstly, on the topic of the Turkwell Dam, the main theory here is that it is a corrupt undertaking. It did not circumvent many of the issues that arise with large-scale dams. It has many of the same negative impacts on livelihoods and on the environment. It has not really achieved much of its goals. To give some concession, I believe it has generated some electricity, but it is not as much as they had hoped. And then to answer the original question of dam development as a whole, medium-scale dams can be extremely dangerous. They do not necessarily or even remotely inherently avoid any problems that large-scale dams have. They're not a safe or viable alternative to be investing in. And many other medium-scale dams share these flaws. So I think the Turkwell can be taken as a jumping off point and an emblem of medium-scale dam development. And then the final framework that has kind of guided me through this and that my professor just drove home in one of her classes that really changed my life and how I viewed things was thinking of development of what, towards what, and for whom. And that is how I have approached this project, considering how different people would answer this question. And this is something that I kind of want to leave everybody else with when it comes to just interrogating how development works and if it is viable. So with that, I just want to thank everyone. Thank you guys for hosting this symposium, and thank my professor for being so wonderful.
And your professor is here, so she can congratulate herself. Claudia, are you there? Anyway, well done, Mari. And we have time for some questions, and then we'll just break for oh, there she is. Woohoo! We cannot hear what you're saying.
I don't think you're on mute, Claudia, but I think there might be an issue with the microphone.
You are worth the wait, Claudia, don't worry.
If we want, in the meantime, there is a question in the chat.
Oh,
are you ready,
yeah.
Claudia?
Patricia is asking, what about medium scale dams makes them dangerous? And then Claudia, you can take the floor.
Okay, hi Patricia. Well, the main things that make them dangerous, they kind of operate on many levels. The first one is it's not less likely to have an overflow crisis. So it can still endanger, especially in this case, 300,000 people downstream. And even though it's technically a smaller dam, it's still 420 feet tall. And if it breaks in a narrow gorge, it can shoot downstream and easily kill people. Other problems with that is there's this kind of like false idea that it's going to provide food security. So by settling pastoralists, you're kind of undermining any alternative lifestyles that they could have. And then by failing to provide that food security, it just leads to like food relief and destitution. And on top of that, having so much standing water in the region can seriously increase like cases of malaria and any other waterborne diseases, which is being shown. And because, it's not just that it's medium scale, it's that it's this really non-directed form of development that does not focus on or consider any people living downstream. And when you do that, even with a medium scale dam, it's still seriously harmful. So I guess that's my general answer. Claudia, for your question.
Can you hear me now by chance?
Oh, yes, we can.
Oh, God. Okay, I'm sorry. I've been having audio problems all morning. So, Mari, congratulations. Really lovely presentation. I knew this would pull things together. It was wonderful. I do have a question, which is, I know you're going to go on with this kind of work, and it really is frontier work. And I really mean that for Southeast Asia and Latin America and Africa. This work has not really been done before. And I'm wondering how do you actually, something I haven't had a chance to ask you is, how do you actually see what you need to know next for this kind of thing? I presume it might be in the downstream area, or how are you that in terms of what's next?
Yeah, I mean, I think the thing that feels most important to me, because I'm focusing on livelihoods in a large part of that downstream region, is actually talking to people. I feel that a lot of what I'm getting from this is kind of like secondhand through reports, and like part of why I took this on is wanting to interact. So
right.
COVID has made that difficult, but going forward, I would really love to be able to actually interview people and hear what their firsthand experiences are with the project.
Yeah, I mean, I would imagine that the government is still going to say this is a positive experiment because it was too embarrassing for them to not do that, right? So that really means you'd need some hard evidence in terms of, is it hurting people, is it helping them, et cetera, presumably downstream then, you're saying?
Yeah, I think also to some extent upstream, because I think like around the actual gorge site is where the Poco are. And they actually have had some serious impacts as well, but mostly downstream.
So those would be the Pokot people upstream, which are both agricultural and pastoral, and then the pastoral is being settled downstream.
Yeah.
Right. That's great. I mean, this will be, honestly, I can guarantee you, working in this area in other regions, the officials in this region will be extremely interested in this data. They will be very interested.
Good. I hope so.
Thank you, Claudia. We have an announcement to make.
I just wanted to congratulate Mari on the GPB major citation. She is the GPB major citation winner, as she knows. So congratulations on that. You're the best.
Thank
Literally.
you.
Literally, you're the
Wonderful.
best.
Oh, thank you so much.
Wow, how wonderful.
Congratulations. So we're
Oh.
gonna make a short break.
I think
and
we
we'll-
also have one other question if we
Oh, we have
can,
one
from
question.
Matt.
Never mind. Apologies.
Matt had asked, how is the current administration approach to tackling the oncoming overflow crisis?
Well, unfortunately, they don't really have much of an approach. I was looking through kind of like what their announcements had been. I saw the one that I talked to you guys about, where they basically said they don't have the emergency contingency plan. There also is no alarm system. Like there's just really no effective way to even warn people. And the other thing is that even if you do warn people, like where are they going to go? Like this is a very like flat, wide area. A lot of these people are tied to their irrigation schemes, as I told you, and mobility has just inherently been decreased, which was one of their survival strategies. So even if they can come up with a plan, it's very hard for them to actually implement it effectively. When it comes to like contingency plans, they did contract with the Kenyan Red Cross, and they came up with like some semblance of a plan and analysis of the situation. So they have something, but it's really not as much as what needs to happen, because this is still like a very, very serious life-threatening crisis, and preventable in some ways. Like long in the past, this was known that this could have happened, which is what makes it so sad. But yeah, I guess the sad answer is not enough.
Great.
Are there any other questions? Okay, we can take a break.
That's it. Great. Thank you so much.
All right.
And I suppose should I go? Will you let me know when? Or?
I will ask Anna to introduce you, if that's okay.
Yeah.
Sure. Yeah, so next we have Ariel. Is it Poach?
Poach. Yeah.
Poach. Okay, great.
Yeah, thank you. So my name is Ariel, and my thesis project is titled When Nature Talks Back, Science, Fantasy, and Rationalism in the Alice Books of Lewis Carroll and Margaret Boyson. And so for some background on where I'm coming from with this, because it takes a slightly different form as a thesis, my project explores the ways that language and storytelling relate to science, particularly thinking about how we collectively make sense of uncertainty and doubt. And I'm drawing inspiration from the many disciplinary crossing possibilities and rabbit holes offered by the environmental humanities, weaving together the study of literature, eco-criticism, and the history of science. I begin with the premise that science, like literature, is a historically, culturally, and materially situated way of producing collective knowledge. And I'm interested in how we can think about this, particularly as it concerns ecological and environmental questions, the ways of knowing and living in the world specific to different people, times, and environments, and how we think about nature and our place in it. So, how do you move slides? So, looking at two stories for or about children, Lewis Carroll's 19th century Alice in Wonderland and Through the Looking Glass, together with his original manuscript and his own drawings, and from the 20th century, Margaret Boyson's Alice, the Zeta Cat, and Climate Change, I'm exploring how fantasy is in dialogue with science. What might nonsense have to say about the particular and historically situated ways we make sense of the world? How do the alternative worlds we imagine through storytelling reflect our understandings of truth and reality, and perhaps provoke new ones? Particularly, I'm interested in how these fantasies might challenge a mechanistic view of nature and instead imagine the world around us, its ecology, to be vital, animate, and alive. I found that what's most moving about these stories is the way they personify and ascribe agency to beings that have traditionally been imagined to be inert or unthinking, and particularly in the wake of scientific discoveries, which also challenge such binaries. For Carroll, the theory of evolution by natural selection, and for Boyson, the study of anthropogenic climate change. So I begin with a passage from Through the Looking Glass. The time has come, the walrus said, to talk of many things, of shoes and ships and sealing wax, of cabbages and kings, and why the sea is boiling hot and whether pigs have wings. The humor of Lewis Carroll, which has given many child and adult readers so much amusement, is centered around the proliferation of possibility and the mutation of common logic. It is not just about walruses that talk, but about seeing the world contrary-wise, as if through a mirror or underground. Experience and reason tell us that the sea is not boiling hot and pigs, as of yet, do not have wings, but Alice's dream suggests otherwise. She is continually made aware of just how irrational the inhabitants of Wonderland and the Looking Glass world seem. She is confronted by puns and other mutations of common language, as well as monstrous and hybrid creatures, animals that speak like adult humans, often taking themselves far too seriously, playing cards, chess pieces, and other inanimate objects that come alive and talk back. Charles Dodgson, who is better known by his pseudonym Lewis Carroll, was an Oxford mathematician, and as such was aware and perhaps a participant in the uproar over the publication of Charles Darwin's theories. Wonderland was published only 6 years after The Origin of Species, and Looking Glass was published later in the same year as The Descent of Man. In this context, we might reimagine all of the lively creatures, mutations, and metamorphoses in the Alice books and see how Alice herself is implicated in this lively order when she swims in a primordial sea of tears with all of the other birds and beasts following her, and when she grows and shrinks unexpectedly as she moves through her environment and must come to terms with that she is perhaps not the omniscient creator of her dreams, but rather a bewildered participant in its ecology. Meaning and truth themselves are dynamic, as are the rules of language unbound in Carroll's evolutionary game. So literature here intervenes in scientific debate and contributes to our imagination of evolution as something random rather than logical, of possibility and unpredictability rather than fixity, making the world seem animate and alive, no longer mechanistic. When Darwin published his theories of evolution by natural selection, the idea that humans shared a common ancestor with all other living things, that species did not exist in a fixed hierarchy, caused distinctions between human and non-human, thinking and non-thinking, natural and man-made, to fall apart. As Alice says, you've no idea how confusing it is, all the things being alive. Curiouser and curiouser, these are some of the same ideas that scholars across the humanities and sciences are discussing now in the context of anthropogenic climate change, when the concept of the Anthropocene, of humans acting as geological agents, is prompting us to scale up our imagination of the human, as historian Dipesh Chakrabarti says. 150 years after Carroll's nonsense verse was first published, the walrus's talk of why the sea is boiling hot takes on a new register. As readers in 2021, in a world where the oceans are warming and acidifying at a frightening pace, what was once nonsense or seeming impossibility comes to bear on reality. Margaret Boysen, a trained geologist and the current director of the Artist in Residence program at the Potsdam Institute for Climate Impact Research, reimagines Carroll's walrus in the era of climate change. In fact, she has taken the whole frame of Alice in Wonderland and rewritten the famous nonsense narrative as an allegory for the way that climate research functions. In Alice, the Zeta Cat and Climate Change, published by the Potsdam Institute's Arts Initiative in 2016, Alice does not fall down a rabbit hole, but instead falls down the ventilation shaft of a supercomputer. She does not pass through a mirror into a looking glass world, but instead enters a climate model through the screen. Inside the climate model, she meets a cast of characters, including a pig with wings and an arithmetic society of nobles, who are personifications of the model's mathematical functions and metaphors for Earth system's processes. So what I have been doing is looking at these two stories and seeing how they are in dialogue with the scientific discoveries and practices of their times. And this is, of course, much more explicit in Boysen's Alice than in the original, where evolution always lies just below the surface. But like Carroll's Alice books, Zeta Cat is a story about what a child can know and come to know, an allegory for dealing with the proliferating uncertainties and contradictions of growing up as a logical creature in an unruly and unpredictable world system. In Boydson's book, animals, flowers, and snowflakes talk because they are representatives within a climate model meant to educate the human Alice about the causes and effects of anthropogenic climate change. Both authors, who inhabit a creative space between fiction and mathematics, show that uncertainty is fundamental to science, but also the imagining of alternative worlds in the future. In a time when the fate of the planet seems intimately entangled with what is possible for humans to imagine, fiction plays a critical role in how we make sense of uncertainty, what stories we tell children and what stories we tell about children are intimately entangled with our imagination of the future and what is possible. It is not just about communicating scientific knowledge of some sort of objective truth, but also about recognizing how science is a way of knowing particular to humans in particular times, and is situated within the language and metaphors we use to understand the world. And I would just like to end by saying how incredibly grateful I am for the help of Professor Turner and Professor Chapella, without whose patience and support and good humor, this project would not have been possible or nearly as fun. And also thank you to the college for making this kind of research possible for undergraduates, and for undergraduates to do work across departments is really such a gift. So thank you.
Well done, Ariel. Congratulations on honors from CNR. And I would like to open the floor for some questions. Feel free to unmute and ask the question directly.
Thank you, Ariel, for your presentation. It was a really lovely presentation. Really enjoyed it. I'm just curious, would you be willing to share like what inspired this for your honors thesis and tell us a little bit of just like the development of working on this?
Yeah. So really this was inspired by a series of coincidences. I think through the conservation and resource studies major, I've designed my area of interest around the history of science and our imagination. It's a word that comes up a lot for me, but our imagination of nature and ourselves as a part of it. And I was really interested, I still am very interested in the history of botany and Linnaean classification. And I had gone to Uppsala in Sweden, where the Linnaean gardens are. And I was reading about how some theories that Lewis Carroll's work was inspired by Linnaeus in part, all of this sort of naming and people asking Alice if she's animal, vegetable, or mineral. And I just thought that was really fun. I'm also interested in children's literature and childcare. And then I was at a lecture at Uppsala University by Kevin Anderson and Isak Stoddard, who work on climate change. And they were quoting Alice in their lecture on the Paris Agreement. And there were just all these strange coincidences leading me to want to do a project about Alice specifically and try to think about it across time and how is botanical classification, and then I found evolution related to also what we're thinking about climate change now. And finding Margaret Boyson's book was really such an amazing coincidence and gift that she also is thinking about such things and tried to repurpose Alice to communicate climate science, but also think about how climate science has produced and sort of humanized that process. Thank
Very
you.
Cool. Thank you so much.
That was really lovely. Someone says, thank you so much, but this kind person is listed as me, so I'm not taking the credit. If you at some point tell me who you are, I'll change your name. Well done, Ariel. And to keep us on schedule, I will ask, please, Cam, to take the floor and present your work.
Thank you. I'll share my screen now.
Oh, it's Maggie. Were you the one that were nice? Okay.
So good afternoon. I'll jump right into it.
This is my research presentation on analyzing uneven vulnerability to urban heat islands using GIS-based techniques, and I focused on Oakland, California. My name is Cam, and I am a conservation and resource studies student, and my faculty mentors for this project are Maggie Kelly and Charisma AC. Before I get into my research, I wanted to start with my motivations for pursuing this project. As a CRS student, I was able to design my coursework to pull from these three fields: city planning, environmental science, and public health. Throughout my undergraduate education, I have developed a passion for researching and understanding environmental problems that lie at the intersection of these three fields, and specifically for approaching these problems through an environmental justice framework to get a better understanding of the social context these environmental issues are rooted in. For my research project, I studied the issue of urban heat islands in Oakland, California. For those of you who are unaware of the term, urban heat islands refers to the disproportionate rise in temperatures in urban areas due to the retention of heat by impervious surfaces, buildings, and other urban materials. This is an increasingly growing problem due to three key factors. The first is a rapidly growing urban population. Currently, 80% of the United States resides in urban areas, with that percentage expected to grow, making the effects of urban heat islands more widely experienced. The second factor is climate change, which threatens a temperature increase of 2.5 to 10 degrees Fahrenheit globally, further fueling urban heat islands. And lastly, historical redlining practices intentionally clustered low-income minority populations, and due to continued disinvestment of these communities, they are now at the front line of today's environmental climate threats. The image here shows the red line map of the East Bay area from 1935. This brings me to my research question and objectives. My driving question for this research project was, how can spatial modeling of urban heat island vulnerability better encompass the wide range of vulnerability factors defined across varying fields? The first half of my project was an extensive literature review, and what I found is much of vulnerability mapping of urban heat largely focuses on the physical occurrence of heat and some social demographic factors. However, the literature also describes other factors that increase vulnerability to urban heat as well, but these are often excluded from the GIS portion of research, which brings us to my project. The objectives of my research are to analyze the occurrence of urban heat islands in Oakland, assess varying vulnerability to urban heat islands based on different aspects, and identify areas most suitable for intervention. My methods for this project came in two parts, the first being a literature review, as I mentioned previously, and the second being a GIS analysis performed in ArcGIS Pro. This flowchart shows the data sources I use in my analysis process to produce the 6 figures I'll be sharing today. As I mentioned earlier, urban heat islands are largely caused by the replacement of vegetated surfaces with impervious urban materials with lower albedos, which allows them to absorb, store, and release more heat energy. This chart shows the relationship between the two maps to the right, mean land surface temperature and percent vegetation cover. Both of these maps were calculated using a USGS Landsat 8 image and aggregated at the census tract level. As seen, there's a very strong correlation between observed land surface temperature and vegetation cover, which is why urban parks and vegetation are the most common solutions to address urban heat. However, before implementing vegetation cover as a solution, it is necessary to examine vulnerability to this issue. The first type of vulnerability I analyzed is social vulnerability. This layer is based on the social determinants of health literature, which refers to social and economic factors that affect a wide range of health, functioning, and quality of life outcomes and risks. The inputs of this layer are listed here and include income, poverty, age, educational attainment, race, and language barriers. This map, along with the rest of the vulnerability maps I will be sharing, is aggregated at the census tract level and displayed using z-scores, which shows standard deviations from the city average. Positive values represent more vulnerable census tracts and negative values represent less vulnerable tracts. I'd like to draw your attention to the dark red census tracts in the middle of the map, which shows a clustering pattern of vulnerability with similar boundaries to the redlining map I showed earlier. The next aspect of vulnerability I analyze is health factors vulnerability. This layer shows health conditions and the factors contributing to these health conditions using data from Cal EnviroScreen. Based on my literature review, I found that various underlying health conditions can increase an individual's susceptibility to heat-related illnesses associated with urban heat islands. The inputs of this layer include asthma rates, cardiovascular health condition rates, diesel particulate matter emissions, and toxic release emissions. The most vulnerable tracts based on this layer are clustered around industrial sites and truck highway routes, which produce harmful emissions. The third aspect of vulnerability I analyzed is adaptive capacity vulnerability. This refers to an individual's ability to respond to environmental threats and alter their local environments to cope with these problems. This layer was the hardest to gather data for, as there are many nuanced micro factors that can contribute to an individual's adaptive capacity in regards to urban heat. These include, but are not limited to, having fans or air conditioning units, or even considering electric bill rates as an indicator of a household's ability to afford using their AC unit, even if it is available. With that being said, the inputs of this layer I was able to include are traffic density, distance to public cooling centers, car ownership, and the year homes were built as an indicator of air conditioning, as AC units were included in home construction after 1950. The data in this map is the least clustered of the 3 vulnerability maps, showing that areas of low adaptive capacity can be found all throughout Oakland. After defining these 3 aspects of vulnerability, I calculated total vulnerability to urban heat by combining the 3 layers. This brings me to my main findings, the total vulnerability map. This map offers a much more comprehensive view of urban heat island vulnerability rather than solely showing the occurrence of urban heat or just social factors. The 2 census tracts outlined in white in the map represent the neighborhoods most vulnerable to urban heat. These were identified by querying the tracts that are in the 90th percentile for both land surface temperature and total vulnerability. The first tract listed is in the Fruitville neighborhood, and the second tract is at the edge of East Oakland, bordering San Leandro. These are the areas where interventions would be most impactful in addressing urban heat islands for the communities that are most vulnerable to them. However, when addressing urban heat islands for these vulnerable communities, the standard intervention of urban parks and vegetation could be more harmful than beneficial. The scatter plot diagram here shows the relationship between vegetation cover and mean income, which has a very strong correlation. This is due to a process known as environmental gentrification, where greening efforts lead to the displacement of the low-income communities they are implemented in. This creates the idea of the sustainability class, the idea that sustainability has to be bought and is only accessible to those who can afford it. As I strongly believe sustainability should not come with a price tag, I want to introduce an alternative solution to avoid this negative outcome, the just green enough solution. This is a planning practice that focuses on social justice and works with communities to identify their goals and come up with solutions that would allow these communities to remain in place after environmental greening efforts. Since there are no clear and easy solutions to this problem, there's currently a lot of research in this area, and this would be the next phase in this research project. Now that I have identified the most vulnerable communities, the next step would be researching how to design and implement green spaces that address urban heat islands while mitigating the potential for gentrification. Looking towards the future, I am proud to announce that I will be continuing my education as a Master of City Planning student at Berkeley, where I hope to continue researching problems at the intersection of environmental science and city planning, and potentially even furthering this research project. I would like to say a huge thank you to the CNR Honors Department for allowing me this opportunity, and a massive thank you to my mentors, Maggie Kelly and Charisma Azy, for all the mentorship and help they provided me with this past year. Thank you.
Hey, we get to keep you. Congratulations, Cam. Hi, Maggie. I have a quick question, and then did you ever think about, you have census tract information. Did you ever think about not just looking at income, but other demographic characteristics of that correlation map? I'm just curious, other socioeconomic characteristics would be really interesting, and not just in a graph, maybe in a multivariate like regression setting, because you have very interesting mapping across these census tracts. So it's very micro. It's very interesting to find not just the marginal correlation, but correlated with more factors together as part of a, a joint analysis.
Yeah, so all of these inputs here are all from census data, and they give a more complete understanding of demographic information that are included within social determinants of health literature. The reason I only included a chart comparing mean income to vegetation cover is that mean income is the best indicator showing environmental gentrification. It made my point the best. In my full paper, though, I will include in the appendix more of the similar maps showing the relationship between both heat and vegetation cover to the rest of the variables.
Yeah, I was urging you not to just do individual graphs for each one, but
Oh.
put them all together. So you say, when we control for income, how is this related to something else? Just a thought, but well done.
Thank you.
Charisma and Maggie, do you have any questions that you would like to ask?
Yeah, well, first I want to congratulate Cam on an excellent project. He's worked so hard over a year, and it's just been amazing to work with him, and not surprising that he'll be starting graduate school this fall. And Kim, I was wondering if you could talk about some of the factors that lead to displacement. Maybe elaborate a bit more on environmental gentrification and perhaps some, I know that's the next phase of your review, but examples of just green enough strategies.
Yeah, so there's a lot of debate around whether greenery is causing gentrification or if it's just a factor in the process of gentrification. But the fact of the matter is that when you have parks and more green space and trees, neighborhoods become more desirable. And that goes for bike lanes and protected infrastructure and any public investment. And it makes neighborhoods more desirable, which allows homeowners and property owners to raise their rent prices and just make the area more expensive, which often drives out the existing communities. And then for examples of just green enough, the one shown here was actually done by I don't know if it was by Jennifer Walsh in CED or one of her students, but it's an example is just showing how you can implement green spaces in sort of the cracks of the urban fabric. So this is a
right. So Anna,
So we're going to take a break and then we'll have 4 more presenters after we come back in about 15 minutes.
Train track, and it's just putting in native plants and vegetation in a way that's not really changing the landscape in a large extent, but a lot of smaller interventions that can add up and really solve the problem without bringing in this investment that causes gentrification.
Thank you.
I would just like to say, lovely job, Cam, and nice to see you, Charisma, too, and Sophia. It's lovely to see everybody. Great job. Back to center spotlight where it needs to go. Back to you, Cam. I would like you to just comment on your design sense. You're a fantastic cartographer and design. You pay a lot of attention to design. Where did you get that from, and how do you think about this when you weave maps into a story?
Yeah, so I got passionate about cartography and mapping and design in general after taking my first GIS class, and it was required for my other simultaneous degree, but it inspired me to pick up the GIS minor, and so I just took more classes in there. And I think maps are really special because you can only say so much with words, but when you can see something laid out spatially, it tells a larger story. And so I love that with GIS, you can show things at different scales, you can show things interact with each other spatially, and it just gives a broader picture. And so I just fell in love with that instantly. And in the last 2 years, particularly, spent my time really honing in that skill.
I was muted, sorry about that. Wonderful job, Cam, and thank you both, Charisma and Maggie, for coming and supporting Cam, and we're very happy that you're staying at Berkeley. Go Bears.
Go Bears.
And I am with you, Maggie. I see people, I'm like, people, and nice people. You know, this is a selective group that comes to the Honors Symposium, even nicer. So I'm gonna ask if please, Catherine Stanton, you're up, and we would love to hear your work. And you have the floor if you want to share your slides.
Thank you. Okay, let me just get it set up.
Okay, so hello and good afternoon. My name is Catherine Stanton, and my research topic was on slow fashion, extending the life of apparel products, and encouraging circularity through textile choice. And I would first like to thank Professor Dara Rourke for being my advisor my first semester, and for Professor Ignacio Chapella for being my advisor this semester. Their mentorship means a great deal to me, and I'm very grateful for the insight they've been able to provide. So to begin, to understand slow fashion, there needs to be an understanding of sustainable fashion in general. And this is something that the industry lacks, as there's yet to be a clear definition of what sustainable fashion looks like. However, Green Strategy has established one definition that, for the sake of my research, I utilized, and this is being clothing, shoes, and accessories that are manufactured, marketed, and used in the most sustainable manner possible, taking into account both environmental and socioeconomic aspects. What I wanted to investigate is how we can utilize the tenets of sustainable fashion to tackle textile waste and other issues within the industry, but specifically using the concepts of longevity and circularity to accomplish this. What I realized right from the start was the amount of consumer influence that was tied into these issues, and mainly due to current purchasing decisions. Whether we realize it or not, consumers have a great deal of power and have shifted the apparel industry from a previous product-driven chain to a buyer-driven chain, essentially allowing us to decide the trends and leave them to playing catch up. This in turn is what has led us to what is known as just-in-time and quick response manufacturing. And we see this typically as fast fashion brands attempt to replicate what has now been decided as the trendiest item. However, there's hope that our consumer influence can be used to our advantage as we become more educated and aware of the consequences of our actions. To move on, fast fashion, as previously mentioned, is essentially a business model dependent on creating the most profit for the industry with little to no regard for workers or the environment at any stage of production. And we see this as it's a system that relies on quick response times, frequent product assortments, a short product life cycle, and focusing on only current market trends. Slow fashion, on the other hand, is a direct response against this, pushed forward by both consumers, designers, and companies hoping to seek and create items with minimal harm. This model pushes forward the use of organic materials, recycled materials, the purchasing of old, used, and/or vintage garments, and consumer behavior that extends the life of a product, such as repair or regifting. And this directly leads us into how we can encourage circularity and longevity within the system and establish them as essential characteristics for production. In order to do so, the role of designers is key, as they are the decision makers of the initial phase of production and have a say as to what textile will be utilized throughout the supply chain to some extent. However, a stakeholder approach and partnership is still incredibly necessary for this work and to transform the linear model into a circular one. Circularity within the fashion industry utilizes all four of the slow fashion characteristics to succeed. And ideally, we would get to a stage where recycling apparel products is a standard instead of ending up directly in the landfill. But designing with circularity in mind can make the biggest impact, as this is the stage where 80% of products' environmental impact is determined. Additionally, longevity. There's two types of durability that it's dependent on, and this is physical and emotional durability. And some may not realize this, but they are both equally of importance, specifically as unwanted yet serviceable goods will still potentially be discarded. And currently, physical durability is decreased due to the quick turnaround that we see, along with bad craftsmanship. But by investing in good materials and slowing down production times, it's possible to work against this fairly easily. As for emotional durability, it's necessary to engage with the consumer again in the design process, either via some form of customization or decision-making, in order to form these intimate connections that lead one to holding on to items longer. It should go also without saying that textiles have a multitude of impacts on the environment, some of them being listed down below. However, most prominently tying into longevity and circularity are the issues of sourcing and waste. Currently, an estimated $192 million worth of clothing are thrown away each year, causing 92 million tons of textile waste. And this is due to the fact that our most conventional materials are polyester, leather, nylon, acrylic, and cotton. And a majority of these are non-biodegradable. Not to mention that synthetic fibers, being polyester, nylon, and acrylic, are man-made and formed out of non-renewable fossil fuels, such as petroleum. Surprisingly, they can present some benefit as we know the most about their life cycles, and this gives us the opportunity to look for improvements within them, but this is very little in comparison to the damage that they create. As for our sustainable materials, these being the materials the industry should be utilizing across the board, they are either recycled or grown and made responsibly, and they're going to follow one of the 4 categories listed on the screen. Cellulosic, referring to those made out of plant fibers, protein, being sourced from animals, regenerated, which is either going to be from previous cellulosic or protein sources, and then recycled, which is directly made out of old textiles or potentially plastic. For more specific examples, we have cotton, either organic or recycled, unfortunately not the conventional cotton, wool and hemp, linen, among others, fall into these categories. And one characteristic that makes these sustainable is their ability to biodegrade and actively work against textile waste issues. Synthetic fibers, although their sourcing is unsustainable, if properly recycled, have the potential to outweigh their harms to a certain extent. As the most sustainable materials are the ones that already exist and do not require any further energy or water use for their sourcing. And recycling and reprocessing work directly towards the goal of a zero waste or circular economy within the apparel industry. And that brings us to our next section, which is the role of textiles relating to longevity and circularity. For both of these concepts, an analysis of the textile cycle is required in assessing whether they fall into biological or technical cycles. Biological cycles follow the principles of biomimicry and nature as a manner of regeneration. Technical cycles rely on products retaining their value and quality even through varying levels of use. And both of these cycles have the potential to work within circularity. However, arguably, the most sustainable choice of textiles would be those that are organic and biodegradable, essentially those natural fibers as opposed to any man-made ones, as these lend themselves to a circular model due to the fact that they have a biological compatibility with nature. This is in part is what is necessary to improve and expand slow fashion overall to a larger scale, but there are also additional things that we can apply. And these are things such as addressing and fixing consumer behavior, having companies and designers emphasizing a holistic approach and view of the supply chain, establishing solid and agreed upon sustainability indicators, not just by one industry, but by both, and establishing strong partnerships among both industries with other goals, and allowing brands and designers to have the ability to determine trends. And hopefully this will all allow us to lessen fashion seasons and overproduction overall. And these are in part some recommendations, but others include the transition to a product-driven chain instead of a buyer, embracing the power of consumer influence, but to the advantage of sustainability, and pushing for corporate social responsibility within these brands, making it a joint effort between brands and consumers, encouraging consumers to treat clothing as an investment, and finally, investing in proper infrastructure to allow for sustainable progress, such as recycling on a larger scale, which currently doesn't exist to the point that it needs to. And in conclusion, it's important to remember that textile waste is a result of both bad consumer behavior, but more importantly, a design flaw and a fault of the system. The role of designers is key to progress, and so is their choice of textiles and how it establishes its ability to be recycled or mended. And above all else, the concept of longevity and circularity are interconnected and necessary tenets to create a new, sustainable, and slow fashion future. finally, I do want to give last thanks to both of my mentors this year and to the CNR Honors Department. Working on this project has been very hard, but also very fun. And I'm extremely grateful for everyone who has contributed to it. Thank you so much.
Very, very well done. Congratulations, Catherine, on honors from CNR. I have a question that was asked by Charisma. She had to leave. Great work, Catherine. I am so sorry that I have to go to another meeting, but I wanted to ask you to expand on the social issues you found in your research, especially on the production side: labor, lack of regulation, unsafe working conditions, price and race to the bottom. Again, great presentation. And then I also have a question.
Yeah, definitely within sustainable fashion, there are so many social factors to take into consideration. Unfortunately, due to the scope of my research, I definitely just focused on the environmental side of that. But as I was creating my research proposal, that was something that I took into consideration because, there are so many tragic examples, such as like Rana Plaza, that real people are being hurt, workers in factories are being hurt. However, that just wasn't something that I was taking into consideration in terms of textiles.
The question I wanted to, or not a question, but I gave you a reference of a very interesting study, experimental study, where a group of researchers introduced a label on working conditions in the apparel industry and wanted to measure stated, but also then they actually see what people do when they vote with their feet through their purchases when they are presented with these information changes. And I give you the link. It's a really interesting study that you may want to find also relevant. And our Haas School of Business Dean, Ann Harrison, has also an established work on sweatshops and how firms react to a bunch of mechanisms through information or other trade pressures on their practices of labor conditions. Just some suggestions for you.
Awesome. Thank you so much, Sophia.
Again, well done. And if anyone has any questions, feel free. We have 1 minute for Catherine, and then we'll turn it over to Matt to close that day. Was the term, actually, I'm very curious, was the term preceded the food, or was it the fashion that preceded? Slow food, slow fashion?
I'm not entirely sure. I
was just like, it's around the same theme, right?
Yeah, it's definitely around the same theme. I would probably honestly guess that food probably came first, just because I think slow fashion in general is more of a more recent trend or concept, but I'm not entirely sure.
Well, thank you. Bye, Ariel. And Matt, if you want to share your screen, you have your advisor in the house. So thank you for joining us, Ted. And if you want to show your, yay, we can. Just because you did this, Ted, you're going to get a screenshot by email. You know me so well. All right, Matt, you have the floor.
All right. Oh, wait, that's might be, sorry. Should be this one.
We're running early, so you don't.
For some reason, it's doing the wrong. Can you see it or?
So what we see is a purple thing, but not in presentation mode.
Okay. Okay, here we go. So I thought it was on the other window with many tabs. All right, so my presentation is about cobalt mining in the DRC. The title, How Cobalt Mining in the DRC is Neocolonialism in the Name of Decarbonization and Digitalization, and how neoliberal interventions will exacerbate this situation. Before I get started, I really want to give a big thanks to Professor Miguel and Professor Villas-Boas for sponsoring and serving as research mentors on this project. It's been a long journey, and I've really learned a lot about what's going on. Okay, so a primer before I get started, just so everyone kind of understands some of the jargon that I use. Artisanal mining is a form of subsistence mining carried out by individuals or small groups not officially employed by a mining company. Usually they have very rudimentary equipment and do it by hands. Among the global south, artisanal mining is a very common subsistence livelihood. And then on the other hand, there's large scale mining that also exists in the region right now, which is mining companies that we generally associate with mining products. They're very capital intensive. They're usually open pit, meaning that they remove a layer from the top of the ground. And they're almost always run by incredibly wealthy multinational organizations. The region that I study in this paper is specifically Katanga. It's the southern subregion in the DRC. In 2015, it was split into 4 different provinces, but for the sake of continuity, I just referred to the entire subregion as Katanga. However, if we want to get more specific about it, currently most of the mining operations that are taking place are in the very southern portion in Haut Katanga and Lualaba. But there's also the Haut Lomani and Tanganyika districts that have other mining operations as well as more agriculture economies. And so while I jump in, I had in my paper, I had three separate sections. I do an extensive historical review, and then I talk specifically about the 2002 mining code and the failures that the mining code and neoliberal policy has created in the region and in the country as a whole. And then I talk about these corporate dominated reforms. So the reason why I had a historical review was that international corporatism, whether it be colonial or through multi-party investors, they've always had a special interest to keep profits extracted from Katangan subsoil. So when we examine certain policy solutions, we should be able to adopt a more skeptical review amongst corporations' auspicious motives to solve the quote unquote poverty, to solve the poverty problem in the region. And so the second reason why I wanted to include it was that also developmentalism in neoliberal contexts, this has been seen in many other countries within the global south. They often provide very technocratic solutions to try to create inclusive labor markets that are generally ahistorical and apolitical. And so therefore, because they assume that they're operating in a vacuum, they don't, the policies often fail. I want to also just state that it's a grave miscarriage of justice to try to fit 100-year history into a sub 5-minute section of the presentation. So instead of going through line by line and trying to cover everything at light speed, I've prepared 5 takeaways that are important to keep in mind from the history. So the first takeaway is that mining started in Katanga, specifically with the creation of the corporation Yeeun Moon, which was a multi-party stakeholder Belgian group that slowly implemented inclusive labor practices and social domination out of necessity in the mid-1920s. This created both level of sympathies with the colonial regime and only specifically Katanga, and also increased production of the UMHK into an international copper producing powerhouse at the time. So my second takeaway is that in Katanga's attempted secession in 1960 and 1963 happened right as independence was happening. This was because there was a clear necessity for the UMHK to hold on to its colonial asset, and so therefore they tried to leverage their influence by also pairing with colonial sympathizers who were trying to reunite the Lundan Empire, and basically created a really terrible crisis throughout the 1960s through 1965. The US is also implicated in this time with McCarthyism basically murdering the first president and then eventually installing Mobutu, because the US also had this basically proxy war happening at the same time in the Congo crisis to take over power of specifically the mining industry in the region. So the third point is that Mobutu nationalized the UMHK and turned it into Geka mines, but he also he wasn't a state communist, he was more state capitalist. He still allowed foreign investment, especially US influence and help to develop mining sites. And what is usually lost in history is that Mobutu also was a strong, Mobutu actually was a strong nation builder for about 10 years. We saw there was 6% growth in the entire nation as he continued to develop Geka mines and Katanga. However, he also underdeveloped nationwide agriculture, national infrastructure. This lopsided economic spending on the economy would slowly prove to be its demise, because in the 1970s, the global recession that started with the oil crisis and the collapse of copper prices really hit the DRC hard. The country was dependent on copper revenue and imported most of their other goods. And so as the country was falling apart, Mobutu was also leveraging that to strengthen his political connections. He becomes despotic in power. He basically he embezzles major funds from the World Bank, and the World Bank in the US, when they give these commercial loans, they don't really care that the debt burden is on the people. It's really just so that the strategic value of the minerals doesn't fall into Soviet hands, which is what they were really afraid of for a really long time until the Soviet Union collapses. And then when the Soviet Union collapses in the early 90s, the US basically cuts off all interaction with the DRC. The state is experiencing massive collapse at this time. And so the fifth takeaway from this is that Mobutu took advantage of the economic collapse and promoted ethnic division in Katanga against Kaijins who also were recruited in the UMHK days to come work at Geka mines, and basically used that as leverage against his political opponent Etienne Tshisekedi, and committed it's unknown to the global north that there is this massive genocide in this region, but that it happened. And so anyways, there's another civil war that happens in 98. Laurent Desire Kabila gains power. He partially does this by privatizing mineral assets and selling them to allies in exchange for arms. He comes into power. He's a quote unquote Marxist, which scares the US. However, he's just as despotic in power as Mobutu was. And much to a lot of speculation that still exists today, Laurent Desire Kabila was murdered to a lot of happiness of wealthy Western elites who were looking to further invest in the region. He was replaced by his 29-year-old son, and the World Bank basically approaches this failing sector and talks to them about how, and pushes urgently that we need to privatize the mining industry. And so that leads us to 2002. And so this next section of my presentation, I talk about the 2002 mining code. I talk about three distinct failures, both on the national, on the regional, and towards the specific artisanal mining community. But before I get there, I also want to talk about why cobalt is so important and what we're getting so wrong about it right now. And so we used to only think of the UMHK as a copper producer, but at its height, it was 8%. It contributed to 8% of global copper supply. However, what's more important today is the 65% of cobalt that comes from Katanga. Cobalt was initially used as pigments for catalysts in plastic production and as super alloys for jet engines and also other high temperature things. However, today, and rapidly growing, the 62% for battery components actually might be obsolete at this point. It changes on a yearly basis. And so battery demand is growing rapidly. We use cobalt in all of our major batteries. We use it for electric vehicles. We're using it for grid storage for renewable energy. We're using cobalt for laptops and cell phones, and we have been for that period, which also raises a lot of, I think, suspicion from my end that the assassination of Laurent Desiree Kabila coincides with the cell phone and laptop boom. That's something that I want to do a lot more research in the future. However, looking forward to battery demand, we have very large expected increases in demand for supply. Sovacool had said that there's a paper said there's 87,000% potential supply increase. Another article states that there might be a 4,500% increase in price itself. Okay, so the 2002 mining code, it was a neoliberal mining reform that basically existed in a vacuum. The reform dismantled Gecko Mines as a state monopoly mining operator and laid off 10,000 employees. Those 10,000 employees were supposed to be remunerated. They weren't. It gave permits to specific mining concessions granted by the Ministry of Mines off technical criteria, off of technical criteria. I'll talk about that more later. It establishes ZEAs, which were artisanal mining places that, artisanal mining could take place there. They legalized that. And then it also established a royalty system for concessions that mining companies would have to pay the state. All right, first mining code failure. Financial corruption of large-scale mines is rampant in the DRC, as one, may cynically expect. Gecko Mines very early in the process took the role of Ministry of Mines because they held all the exploitation permits at the time, all the best ones, and basically gave those permits in these backdoor deals. The CEO of Gecko Mines, who's still the CEO now, but hopefully will be removed soon, would only answer to the president. And so through multiple embezzlement, there's been kickbacks, there's even been with Glencore, which is probably the worst of the worst. They also became a mining concession seller in the DRC because Gecko Mines would give them permits and then they would give them again. Over this entire course, Joseph Kabila's net worth now is like $2 billion. Money is continually missing through state revenue coffers. Failure number two, talks about on the regional level, there is numerous environmental health externalities, as well as a complete disregard for communities surrounding mining sites. There's dangerous pollutions in lifeline rivers. For example, the Basse Kondo, the hunting preserve, it was established in 1957. There's now a mine and a refinery on the protected hunting reserve, violating national law. These impact local waterways that are lifeline for everything needed from agriculture to drinking to washing. On top of all this, those who live near either transportation roadways or sites experience massive amount of air and noise pollution. There are some studies that are, looking at the level of lung disease in and around mines, as well as radiation poisoning, because Katanga has high uranium reserves that are being upended. The worst part about this entire portion is that there's massive community upheaval. Mining companies come in, they don't really do any due diligence. They remove the state basically removes these towns and puts them in places with non-arable land, and they are either not compensated or severely undercompensated. They have no agency in this program. And on top of this, there's like a 0.3% corporate social responsibility that large-scale mines are supposed to participate in. Of course, they don't do that on top of everything else.
Matt, could you please summarize the third failure briefly and then try to conclude? We're just running a little bit over time. Thank you so much. I know it's really unfair to summarize all of this in 10 minutes.
I'm all over the place. Okay, so the third failure basically is that the artisanal mining supply chain is fully exploitative in every sense of the way, in every sense you can imagine. There's no social programs for them. The ZEAs that were produced are too small. People always talk, there's child labor present, and just rampant systemic poverty in the area. Okay, so I'll quickly go over my kind of, just being upset about these corporate dominated reforms. The main one that's gaining a lot of traction is, quote, cleaning up the supply chain. NGOs have done a good job reporting on the conditions that are happening. However, their calls for change are always, let's bring Apple, let's bring Huawei, let's bring Daimler into this conversation to do due diligence and make sure that these mining supply chains are sustainable. However, sustainability really is going to come down to what they decide is can and can't pass while the demand is still so high for cobalt. On top of this, there are numerous anecdotal evidence that talks about the contradictory nature of downstream efforts. Samsung SDI had one of the best ratings for supply chain due diligence, but at the same time, they're buying tons and tons and tons of cobalt from Glencore, which is the worst offender. The second portion of corporate dominated reforms talks to supply chain transformations and the leverage that downstream companies have. The price of cobalt is demand oriented and not necessarily deflation of price, but price shocks leading from specific policy changes, and also innovation could create supply chain shocks that will be felt downstream. There's evidence that very similar things that have been done in the north-south Kivu provinces, and as well as Tanganyika with the 3TG and Obama's Dodd-Frank law had really negative effects on artisanal mining in the region. The recycling and circular economy is somewhat of a more long-term worry that has ostensible good benefits to it, but also has a lot of negative, also could do a lot of damage. In terms of basically closing the cycle without any form of new policy intervention for artisanal miners. It's not necessarily about having, having cobalt that is, conflict-free, but rather developing the region around it so that there isn't conflict to begin with. And so then the potential hope is that the first democratic election, actually since 1960 happened in 2018, electing Felix Tshisekedi. I have high hopes for him. He enacted Kabila's outgoing mining code reform, which moved up to a 10% royalty on cobalt concessions. He is creating agencies that will buy artisanal cobalt as a monopoly, and he's slowly weeding out Kabila's network of power. We're hoping that Albert Yuma will be the next, the head of Geka Mines will be the next to go. And so going forward, industry nationalization, I believe, in the formalization of that industry should be the goal. I think that the neoliberal intervention into Katanga was one sought on opportunity for this new digital and decarbonization revolution that we have going on. And over the course of the next 20 years, over the course of 20 years, large-scale mines have repeatedly abused the mining code with no recourse. They should have to pay for the crimes against humanity that they have committed. Thank you so much, Ted, for everything that you've done. I really couldn't have gone this far without you. And thank you so much, Sofia, as well. Also, congrats to everyone in Honors. This has been a really amazing day listening to all your presentations.
Thank you. You spoke so long and eloquently that we have no time for questions to keep it with time. So if anyone has questions, Matt Arena at Berkeley, if you just start typing, his email will be there. I give the floor to Anna.
Yeah, thank you, Matt. And then thank you again to Ellen for being one of our judges. And yeah, and then also Leila from this morning, who's not here, but thank you to her. And thank you so much for Sophia, who's also been judging all of you as we're going along and helping keep track of the time, asking you great questions. Thank you so much for that. Once again, we're going to be coming back tomorrow for the biological sciences. So that will be kicking off around 8:45 tomorrow. So if you want to see that, that'll be another full day of a second symposium. But congratulations to all the people who went today. I think there's still a number of you here. So congratulations on finishing your presentations today. And also congratulations to those of you who are graduating, which I know is many of you who will be graduating in the next few, either this spring or in the summer. So congratulations on that. We really appreciated your research and hearing all about it today.
Yeah, thank you so much for having me. Great presentations, everyone. Congratulations.